Hello? Okay, great. So it's always on?
Okay, Assalamu alaikum. Good morning, everyone. We just gave a few minutes extra for people to come. We know it's a Friday morning and it's a basic science session, so not many people are interested in this. It is really my pleasure to co-chair this session with my new friend and colleague, Dr. Mona Al-Atibi. As the name indicates, she's a Saudi Arabian lady. She's a scientist from San Diego, where she has her lab there, and she did a lot of work, and she is one of the task force members of the World Symposium. And we will have 3 talks this session, and followed by another session with three talks, and given about half an hour, 1 hour and a half for people for the Friday prayer. So it's my pleasure again to co-chair this session with you, Mona, and please introduce the first speaker.
Thank you so much, Dr. Al-Balan, for the kind intro here, and really my pleasure to be here. Good morning, everyone. Thank you for coming. We have exciting lineup, and it's my pleasure to introduce Professor Soni Palamsedi, Professor of Cardiopulmonary Institute, and she's from Gießen, Germany. She's going to talk to us about unraveling the genetic and epigenetic blueprints of pulmonary arterial hypertension.
Can I start? Thank you, Mona. And also thanks to Professor Abdullah for this kind invitation, and salam alaikum. And then I think I would like to change a little bit my title towards the epigenetic reprogramming in PH, whatever the ongoing work from aging to mechanics and to the therapeutic opportunities. So when we all know that the pulmonary hypertension, it has the diverse etiologies. So one is with the pure form, which is the pulmonary arterial hypertension, the PH due to left heart disease, the PH due to lung diseases, and the chronic thromboembolic pulmonary hypertension, as well as with a hematological or unknown multifactorial disease. At the end of the day, could be different etiologies. They all result in pulmonary vascular remodeling and leading to right heart hypertrophy and right heart failure. So the question always is, what leads to these pulmonary vascular changes? So it was well studied in the group 1, that is the pulmonary arterial hypertension, and people has described more about the genetic predisposition. Yesterday, what Paul Yu was mentioning, a lot of BMP, TGF-beta signaling, activin signaling, related molecular pathways related to a lot of mutations has been identified. The BMPR2, for example, takes the central stage with 70%. However, when you now look at not every BMPR2 patients leads to have a pulmonary hypertension. So the pathological variants explain only 20 to 30% of PH cases, and most BMPR2 mutation carriers remain asymptomatic. The Delphi 2 study has shown that it takes only 2.3% per year. They need an additional hit, a trigger at a second hit, what you call it, that can lead them towards the BMPR2 or to develop symptoms. And that's all about group 1 pulmonary hypertension, but when you take about group 2, group 3, and group 4, there is no major genetic mutations that has been identified. This clearly tells you that you need mechanisms that are regulated by the environment and that responds to the environment. And that's called is the epigenome. So epigenome is the one which is affected, the genetic architecture, which is affected by the environmental stimuli, the drugs, the aging, the lifestyle, and so on. In a simple terms, if you can say the genetic changes are like a hardware for a computer, but the epigenome is like a software. So it can write the things, it can remove the things, and it can architect in a way so the genes can be on or off. So without changes in the DNA sequence. Yeah? So this is, I think, could be a DNA, different mechanisms are known, such as the DNA methylation or histone modifications like histone acetylation and methylation. So at the end of the day, they try to change the chromatin, which you can see in these loops, so the DNA that could be opened up so that the gene transcription can go on. So the epigenome, why we think it's an important player? Because The genetic factors, I told you that not every people whoever has a BMPR2 mutations or TBX4 will have a disease. So it has to be combined with the different environmental stimuli such as hypoxia, inflammation, drugs, aging, and so on that could be translated into the epigenetic changes or the modulation of the genome. And so in a very simple terms, I can say genetics is the one which loads the gun, but the epigenetics is the one which pulls the trigger. So in that way, these epigenetic changes may determine what is the penetrancy, what is the timing, and the severity of the pulmonary vascular changes that are happening. So I think in my talk, I would like to focus on 4 different aspects. One is that epigenetic landscape, which we try to delineate from the human pH vascular cells. And then what could be the drivers of these epigenetic changes? So we explore the mechanosensing and the aging aspects, which we will also talk about. And where are we with regard to the therapeutic opportunities, which is maybe more interesting for you in terms of epigenetic therapeutic targeting of the epigenome. So we started this a few years ago in understanding the epigenetic landscape. So what we did is like when we get a transplanted PH lungs and also the health donors which has rejected, so there I think we started isolating the PH vascular cells. The first thing you observe is that these cells are different. They are hyperproliferative in nature. They are resistant to apoptosis. So they are out of their environment, but they have a different phenotype. And this phenotype persisted for several passages. And also, when we did the RNA sequencing to see what are the gene expression changes, we saw more than 2,000 genes are differentially regulated. This tells you there is some addiction going on for these cells, and there is some memory. They keep a memory in their cells, so they are out of their environment, but still they can do their job. So they are tuned in a way to become more hyperproliferative and so on. So that's why, which gives you an epigenetic memory. So we went and checked for the histone changes that are happening by doing the chip sequencing. And so what you can see here from the D panel to the P, which is from PH patient samples. So you have a lot of histone acetylation changes that are happening in the PH vascular cells. Fine, but then I think what we also observe is that we see more than 35% of the genes are related to developmental pathways from PH vascular cells, which are from the adult, which, where you don't expect such kind of activation of the developmental genes. And a lot of transcription factors, which has to shut down. They are very highly active before birth, and after birth, their expression has to go down. Their function has to go down, but now they are reactivated or re-expressed, and this is all modulated by the histone acetylation changes, which you can see here. So this made us to think, okay, there is some reactivation of developmental program going on, and this is epigenetically modified. And putting all these things together, the big story of our paper, so where we said like, okay, when we combined RNA sequencing with the epigenome, the chip sequencing data, this is all that makes your vasculature to, when it's exposed to different hypertensive stimuli, which I will talk later, but this is all leads to re-expression of developmental transcription factors and developmental genes like T-box domain, SOX domain, and so on, which gives the cells a memory. So they can live in any environment, they behave in the same direction. So, and what we also and this could be epigenetically reverse reprogrammed, so I will talk about that later too. But then what we think is like the programs, the developmental programs, which has to be shut down after birth, they're now reactivated in the adult disease, and so they came in wrong time of your life and wrong environment. This may be behaving like a fetal-like circulation, and this leads to pulmonary hypertension. And then we asked, can we reverse these changes? So because when you now look at the histone acetylation, the acetylation marks are written by the writers, the enzymes called histone acetylase transferases, HATs, or removed by the enzymes called HDACs, histone deacetylases, and the acetylation mark is read by bromodomains. So, and what we did is like, since we see more histone acetylation, so we try to block that by using P300 CBP HAT inhibitors. So what we did, we took the human pH precision cut lung slices, So we make unto 400 micrometer thick sections, and we established a protocol that you can keep them alive in cell culture dish for 2 weeks, and you can treat them with different inhibitors. So when we treated with this P300 inhibitors, and then for example, when you use sildenafil, you don't see any reverse remodeling, but here definitely with these molecules, you can see there is a decrease in medial wall thickness. And beyond the human relevance, but we also checked this in 3 different animal models of pulmonary hypertension, and in all 3 animal models, we see a decrease in the right ventricle systolic pressure and increase in the cardiac output and so on, clearly suggesting that this epigenetic phenotype, what we observe in PH, can be reversible. So then, but the question is, what drives these epigenetic landscape changes? There's a huge change that is happening out there, but what could be a driver? So we explored 2 concepts. One is like the matrix stiffness that comes very early, and also about the aging aspects. So with regard to the matrix stiffness, because I think what we all know that because we have a very early stiffness that is happening in the distal or the proximal pulmonary arteries before even we identified the severe hemodynamic changes. We all think that initially, okay, this is only changing the pressure and the flow, but what they can do also, they can give a mechanosensing signals. So they can modulate a lot of biological pathways. In that way, there is a mechanobiological feedback that goes on, and the work from Steve Chen's group has elegantly showed that very early the stiffness of the matrix happens in animal models, and this drives the vascular activation of the hyperproliferation of the cells and so on. So this is an early pathological change, and there is also literature where it shows that I told you that it's not only a physical barrier or modulating the flow and pressure, but the stiff matrix or changes in the matrix, how it is architecture and so on, they can also give signals to the cells, like mechanosensors are there, like hyposignaling molecule, and then via the Rho kinase signaling and so on, they can also alter the epigenome. So this is what we try to explore, whether even in pulmonary hypertension, the mechanical stiffness, what we see within the vessels, whether they are transmitted to the nucleus and whether they change the nuclear architecture and the chromatin changes. Indeed, when Samuel has done this work, what we see is that compared to the top panel from donor to the idiopathic form of PH, you see even the nuclear change, architecture is changed. The circularity of the nucleus is decreased. So that means all the architectural changes, because you can also see here, the nuclear circularity is changed. That means the nuclear proteins and how the nuclear dynamics and actin cytoskeleton within the nucleus, this is all altered. And that can have a huge impact. And then I think further, I don't want to go in detail because this can change the whole architecture of euchromatin. That means the chromatin that is active versus the heterochromatin where the gene is shut down and so on. And then I think we try to modulate the mechanotransduction pathways like hippo and Rho kinase signaling, what gives the senses of the mechanosensing. And then what we can, in short, can say, when you try to modulate those ones, you can see a decrease in the histone acetylation, this H3K9 acetylation on the extreme panel, as well as you can reverse back the nuclear circularity, clearly suggesting that the mechanotransduction pathways are one major pathways that drives these epigenomic changes, and that could be modulated. The next thing would be asked, another thing that is important here is also the stiffness happens upon age, the pulmonary vascular stiffness and then compliancy loss and so forth, but upon aging. And another thing is like now we see that the mean age in the PH diagnosis more than even group 1 is shifting. So it's more than 55, but in group 2 and group 3 is of course about 65. And from the PH diagnosis over time, from NIH registry to Compara, also the age is increasing and more than 45% of the population is above 65 years. And what is known that the aging can influence the epigenome. So they can really modulate by transposon elements and whatever the DNA methylation changes, the whole architecture can be influenced. So together with Kodatello's team, who collected the PBMCs from the PH patients, what we have done, we performed RNA sequencing. Interestingly, what we see from these PBMCs, when we did this sequencing, we see a lot of changes related to aging. The pathways relating to aging came up. So this was not, it's an unbiased study. We did not check for aging molecules and so on, but a lot of aging pathways came up. And interestingly, even in group 1, we see 6 to 7 years, we did like an aging clock. You can predict an age, not a chronological age, a biological age. So here we see that 6 to 7 years of accelerated aging is going on in the immune cells from the PH patients, clearly suggesting that beyond chronological age, there's also accelerated aging going on. So now the next question is, how will it influence the immune cells? And so for that, I think what is known is like what is coming up in the field is that when you become older, you gain a lot of mutations. So these mutations are not germline, so they are not coming from your birth, but they are coming upon aging. So age drives these changes. So they modulate some of the driver genes, and that gives a clonal advantage and can lead to hematological malignancies. So when we started looking, these are called chip mutations. So I think when we screen for in our PH patients cohort, so what we see, 30% of them, they have these somatic mutations that were gained upon aging. And this is majorly related to two enzymes, which is related to epigenetics, that is the DNMT and TET2, so which modulates the DNA methylation. So indeed, these age-associated somatic mutations we gain, and Stephen Archer's group also has confirmed in a big cohort. So this leads to change in the epigenome. So the next question is, what does it do to the cells? So when we take these PBMCs and try to mimic these mutations, like DNMT3 and TET2, which is a loss of function mutations, what you see, there is an increased, this leads to, the moment you change them, it leads to high inflammatory cytokine profile. And you put this conditioned medium onto smooth muscle cells, cardiac fibroblasts, or vesicular organoids, this all results in increased proliferation state. So in a way, age, by driving the somatic mutations in the epigenetic regulators, modulates the inflammation, and this inflammatory phenotype drives the vascular remodeling, clearly suggesting that age is one major factor that driving the epigenome. So then next, I think at least we know what could be the factors that can drive this epigenetic landscape, and then it could be also reversible. But where are we towards the therapeutic opportunities? A lot of preclinical work has been done focusing on the histone acetylation landscape. It could be HATs, it could be histone deacetylases, the erasers, or the bromodomain inhibitors. All the preclinical work except one study clearly shows that they can do the reverse remodeling. And there is a pilot study has been done by Steve Provencher and Sebastian Bonnet's group, where they use the BRD4 inhibitor in a small population of the patients, where you can see a decrease in the PVR and an increase in the cardiac output. And there is a recent study where we all, like the work from Martin Wilkins' group and our work, as well as the work from Kurt Steinmark's, we all have shown that the class 1 HDAC inhibitors can be a potent way of doing a reverse remodeling or reversing the pulmonary vascular changes. And there, I think Serena and Scientific, which is the CS1, which is now a controlled release formulation of valproic acid, which is a class 1 HDAC inhibitor, and they have shown, which is led by Rebenza, so they have shown this is a primary endpoint of safety and tolerability, met successfully, and there is compelling data available on the REVEAL score functional data set and reduction in mean PAP. And then it is now approved as a compassionate use, and there is a phase 2b clinical trial going on. With this, we are at a baby steps towards the clinic. But I think one other thing which we have to be careful with the epigenetic modulators is that because you are modulating a lot of genes, so you should know which ones are you targeting. You should be specific about that you are not blocking the off targets and the genes which is of your interest. So that's why you should be careful about undesired side effects. But I mean, luckily, so from the cancer field, we know that they are trying to use, working a lot on this direction to improve the next generation inhibitors and also to precisely target the epigenome on the genes where you want to have the direct effects. So this is a precision epigenomic therapies which we are also working on. And then I think, of course, we need to know what is the influence on other systems that are affected, like right ventricle, the immune cells, and so on, and how to really find out the responders for the non-responders if it comes towards the clinic and so on. So this is still a lot of things that need to be improved, but clearly it gives you a point that I think this is something that could be reversible and targetable. So at the end of the day, I think what I told you, that in the PH pulmonary vascular cells, you see a whole epigenomic landscape, even the nuclear architecture is changed. And it could be modulated by the stiffness, the pulmonary vascular stiffness, via modulating the nuclear changes and epigenome or alterations may be happening. On the other hand, aging, by changing the somatic mutations on epigenetic molecules, can lead to epigenomic instability and again leading to epigenomic shift. And this epigenomic remodeling is dynamic and reversible, because with the specific therapeutic targets, you can reverse back these changes. And definitely this creates a new opportunity for the biomarkers and targeted therapy in PH. With this, I would like to thank all the people who are from my group who has contributed to this work, and the clinical work from Codatello's team and Sebastian Bonet's team from Laval University. Thank you for your attention.
Thank you, Soni. Please join us. The questions will be at the end of the session. Now it's my pleasure to introduce Dr. Muna Al-Ataibi from San Diego again. She will be talking about deep phenotyping into phenotypes-based ontology. Muna, the podium is yours.
Thank you again, Dr. Abdullah. Really honored to be here. You know, Jeddah is my hometown. So I'm thrilled to talk to you about some of our work on, specifically on metabolomics for deep phenotyping in pulmonary hypertension. All right, so we're gonna start with a clinical vignette. I'm sure most of you clinicians, this is very common presentation for you. So you have two patients, two young females presenting with chronic shortness of breath. You know, general workup was negative. Echocardiogram showed evidence of RV dysfunction. Referred to the PH clinic, who did a right heart cath, showed elevated pulmonary artery pressure at 46, PVR of around 8, normal wedge cardiac index is low. They're around functional class 3 to 4, so you highly suspect that they have PAH. You cath them, you rule out other causes, and then you start them on dual therapy. So same presentation, same phenotype, you know, phenotypically similar patients. Patient A, when you start them on dual therapy, they improve, their functional class status improve, their RV function improve on echo, and they do great. Patient B, however, despite, again, similar presentation, they have progressive symptoms, they have declined exercise capacity, they have RV dysfunction on repeat echo. You escalate therapy, multiple hospitalization, and again, it just seems that it's not working. So I'm sure this sounds somewhat familiar, you know, seemingly similar phenotype, but something is missing. And what we're missing here is the underlying biology. So instead of thinking in terms of labels and only hemodynamic, what we're missing is thinking about the molecular phenotype of these patients. So one patient might be, for example, dominated by bioenergetic stress or by immune activation, inflammation, versus other patient might be dominated by other molecular profiles. And those are not captured by hemodynamics and clinical metrics we use. These biological patterns are what I'm going to refer to as endophenotype. So the endophenotype basically means using molecular programs such as any omics, metabolomics, genetics, transcriptomics, or even a combination of the above, and then reclassify patient based on their molecular patterns into different categories. And these patients, these endophenotype could explain the differences in patients' function and outcome in response to therapy. So in the next few slides, I'm just going to take you through different endophenotype using metabolomics. So for the rest of the slides, we're going to go over two different categories. So using high throughput metabolomics to define endophenotype for disease risk and endophenotype for drug response. All right, so first off, why metabolomics and what is metabolomics? So metabolomics basically means measuring the small molecules in the blood. You know, calcium, potassium, alanine, all the other metabolites. And the reason I got interested in metabolomics is because it is the most sensitive to any change in the physiology or changes in your bodies. It reflects your genetics, it reflects what you eat, it reflects your drugs, your exposure, really all the risk factors. The interesting thing about metabolomics as well, it's very dynamic, meaning if you adjust something, you remeasure the metabolomics, you see a response right away. Now, there are two types of metabolomics. There is something called targeted metabolomics, which basically you measure known metabolites or known pathway. So you go in with a predefined hypothesis, knowing exactly what you want to measure. And a lot of people did great work with targeted metabolomics. And there is untargeted metabolomics. And when we say untargeted metabolomics, you basically measure high throughput, either a class or multiple classes of metabolites, and try to discover novel mechanisms or novel metabolites in relation to disease. To give you an idea what untargeted metabolomics means, in our body, only around 20% of the metabolome is actually known. The rest is just black box. It hasn't been identified or pinned to a pathway before. So it makes it challenging, of course, but also makes a great opportunity for discovery. So for our work, we focus on both untargeted and targeted metabolomics. And again, I'll give you examples of how we utilize this in our work. All right, so first off is examples of using endophenotypes, metabolomics for endophenotypes of disease risk. So we ask the question, can we identify at baseline using metabolomics which patient at high risk of dying? Because right now with PAH, we use metrics of disease severity, we use 6-minute walk test, functional class, and all of that, but then obviously it has their own shortcomings. So we wanted to see if we can use metabolomics to do that. So if you think about PAH and really other non-communicable, non-genetic disease, the issue is for every change in PA pressure, an increase in PVR, there is small and maybe clinically undetectable change in RV dysfunction or RV function and decline in cardiac output. And this change starts before any clinical risk factors or any clinical risk scores really are apparent. So what we're trying to do is we're trying to capture this through biomarkers or metabolites. So to do that, we started by assembling one of the largest cohorts for PAH. So we had around 3,000 patients with group 1 pulmonary hypertension across the US. Those were assembled from many centers, and we had 4 cohorts of PAH, 1 cohort of control. These patients were well phenotyped. They had right heart cath data. They had, functional metrics and whatnot, but they were also followed for up to 5 years for mortality. So then we performed high throughput metabolomics, and we focused on the bioactive lipids. We're interested in the bioactive lipids because those could function as signaling molecules. They're highly tied to pulmonary hypertension, but we did it in high throughput fashion. So we measured around 20,000 metabolites here. We did some regression analyses to identify metabolites that are associated with mortality after adjusting for multiple metrics, including disease severity, duration, and lots of others. We identify a set of metabolites that were significantly associated with mortality, even at a metabolome-wide threshold of 10 to the negative 5. Again, most of these metabolites are just unknown, so we decided to focus on a convergence approach, meaning we wanted to identify a pathway that is related to mortality instead of just focusing on single 1 or 2 metabolites. So to do that, we did several approaches, including molecular networking and clustering and others, and we were able to identify a very interesting cluster of metabolite. And when we did Mendelian randomization and we did association of the genes and the metabolite, we were able to pin these metabolites into the oxysterol to bile acid pathway. And we validated this finding in a community cohort of just normal individuals to link these metabolites to this pathway. In fact, when we also tested some of the transporters for these metabolites, they were also significantly associated with mortality in pulmonary hypertension. Those metabolites were not your, upstream oxysterols or downstream bile acids, but they were intermediates. And, there's a lot of interest now in the bile acid and oxysterol, not only as inflammatory or obviously liver-related metabolites, but also as, related to signaling molecules and upstream inflammation. So then we wanted to validate these findings in human tissue. So we took lung samples from human with PAH and controls, and we also saw this pathway and these metabolites were dysregulated in human tissue. We also validated in preclinical models. So we saw these metabolites were dysregulated in the Sugen hypoxia more so than the monocrotaline model. We wanted to take that back to the clinic, so we built a metabolite risk score using a weighted score of these metabolites. And when we compared this metabolite risk score to clinical metrics that we use in clinic, this metabolite risk score actually outperformed all of these clinical metrics in predicting mortality for these patients. Then when we compared it to the Reveal light risk score, for example, it was non-inferior to it. But then interestingly, when we combined these risk scores, the clinical risk score and the metabolite risk score, it definitely improved prediction and for mortality for these patients. When we classify patients using the metabolite risk score into low risk, intermediate, and high risk, you see in red here in the Kaplan-Meier curve that patients with high risk definitely have higher separation and high risk of mortality. This is up to 5 years. So you see that at 2 years, they had around 45% risk of mortality. Now, interesting to the right, you see in this forest plot that patient in the higher risk score has around 10 times risk of dying compared to patient on the lower risk score. When we looked at the reclassification, so we looked at the agreement between the clinical risk score and the metabolite risk score, you see that in the graph to the left, there is some agreement, especially on the high risk score, which I think it makes sense that when you see patients that are super sick, you don't really need a risk score to tell you that they're at high risk of dying. But if you focus on the plot to the right, only patient who died, and they were classified at lower risk at baseline using the clinical risk score, the metabolite risk score was able to reclassify around 40% of them as high risk to start with. And it was able to reclassify up to 50% of the low risk for the clinical risk score as high risk when we used the metabolite risk score. Then we ask the question, now this all is static, prognostication, but how does the metabolite risk score does as a dynamic marker? Like after therapy, when patient improved, what happened? And in a subset of patient, we had follow-up blood samples. So we saw that when patient improved, the metabolite risk score actually is dynamic, so improve with therapy, versus when patient don't improve, it just stays the same or worsens. We were then interested to see how early can the metabolite risk score predict disease. Specifically, how early could it detect any dysfunction in the RV? So for that, of course, as you guys all know, with PAH, it's like very tricky to have longitudinal samples. So we utilized community cohort of around 6,500 individuals. Those are normal individuals. We had blood samples at baseline, but then they were followed for more than 20 years, and we had echo data, and we had estimated RV pressure. And then we tested our top metabolites from the metabolite risk score, and we were able to see that our top metabolites were associated with RVSP even 20 years in advance, which it was really interesting to us because there's potential for early detection and not just mortality prediction. All right, so switching gears now to looking at metabolomics endophenotype of drug response. Like a lot of other people, I was really interested in trying to predict which patient would respond to which therapy, which PAH-specific therapy. And then the other thing, when we start someone on PAH therapy, are they actually responding or not, more than just doing the right heart cath and asking them. So to answer these two questions, we utilized longitudinal or prospective data of two different cohorts. So in cohort 1, they were randomized to different PAH therapy. There were new diagnoses of PAH. At time 0, we collected blood samples. They were randomized to pH therapy. Then we repeated right heart cath, got blood samples again. In the other cohort, they were already on baseline pH therapy. Again, we collected blood samples at baseline. They started on other pH therapy, and then we got blood. In the second cohort, interestingly, patient got cardiac MRI at baseline and follow-up. So to answer the first question, when you start someone on PAH medicine, are they actually responding or not? Basically, we're trying to identify pharmacodynamic biomarkers for pH therapy. So to define a pharmacodynamic biomarkers, we had to have two criteria. Criteria number one, the biomarker has to change with the pH therapy, but not with other specific therapies, so it has to be specific to this pH therapy. And then second, it has to be associated with improvement in pH metrics. So in this case, we chose RV function and PVR. So for this cohort, you see here in this volcano plot that these biomarkers increased consistently with this pH therapy, both at 26 weeks and 52 weeks. So we then took these metabolites and we did some Bayesian analysis of these potential pharmacodynamic biomarkers. And we specifically chose the biomarkers that A, changed with therapy, B, associated with improvement in RV systolic volume by cardiac MRI, and also improvement by pulmonary vascular resistance. So if you look at this green right lower corners, those are the potential target pharmacodynamic biomarkers. This is just a forest plot showing these top metabolites. So in the first one, you see that metabolites which increased with therapy consistently associate with improvement in the RV systolic volume and reduction in the PVR. And the opposite is also true. Sometimes these biomarkers were actually reduced by therapy, so they were quote unquote bad, and they were associated with worsening of RV systolic volume and worsening of PVR. Then when we took one of these specific biomarkers and looked at within that cohort of patients who were started on therapy, patients who responded versus not responded, what are the differences in these pharmacodynamic biomarkers? So if you see this plot here, those patients at baseline, they started on the same therapy, but some of them had increase in the pharmacodynamic biomarkers, and those are the patients who responded, versus patient who did not respond had a flat or like really worsening of this pharmacodynamic biomarker, which is really interesting because potentially you could follow this and see perhaps like these patients have some, hypermetabolizers or like, some antagonism or whatnot, something to kind of think about. Now, of course, we're not powered to do this analysis, but again, when we look at mortality and all outcomes, including hospitalization, by these biomarkers, you do see a separation here. It's not statistically significant, but again, it's interesting to think about. We next asked, can we predict at baseline who is going to respond to specific pH therapy? So for that, we utilized this cohort. Again, those patients, some of them were on pH therapy, so when we did our analysis, we adjusted for whatever therapy they were already on. So again, we measured this high throughput metabolomics, we focused on the bioactive lipids, and we did we took the metabolites at baseline, we performed some fixed effects analysis, and we identified a bunch of metabolites at baseline that were predicting improvement in RV systolic volume and improvement in PVR. When we perform prediction, so this is just an example of using the metabolite risk score, a prediction of improvement in PVR, and we used bootstrapping in this case, and you see that the R2 square is 0.82, which is like pretty good. We didn't have external validation, so we used just the bootstrapping, but again, limited data, but interesting potential that we could be utilize this approach in the future. So back to our patients that we started with, the clinical vignette. So again, similar phenotype, different biological state, utilizing omics and different omics, including metabolomics and deep phenotyping. Potentially in the future, when we see these patients in the clinic, we could perform the clinical risk scores, but we could also tell If patient number one, for example, had a low metabolite risk score, perhaps patient number one was responsive to this specific pH medicine versus the second patient was not maybe responsive to the specific pH medicine, so would deserve to be started on something else alternatively. If we knew that patient is high risk at baseline, then perhaps early referral to transplant or thinking about triple therapy and whatnot, from the beginning. So with that, I really, this is a picture of close to our lab in San Diego. I welcome you if you're around to give us a visit. Of course, the main challenge with omics is the need for collaboration. You need external validation, you need data, you need biospecimen, you need biosamples. So it's really not a one-man's job. So to summarize, metabolomics enable deep phenotyping by revealing molecular signature that extends beyond traditional hemodynamic classification. We can use metabolomics to biology-defined endophenotype. Those could refine risk classification. It could uncover mechanism, and it also could help predict treatment response. Of course, this approach requires rigorous external validation for some studies. The first one, we had that. For the second study, we didn't. Longitudinal data, integration with other omics platform, I think that would be really interesting. Our progress really depend on large-scale collaboration, shared biobanks, and then strong clinician-scientist partnership. So with that, I'd like to thank my lab members at UCSD, my pulmonary vascular group, and my collaborators, Chief Chan, Bill Nichols at Cincinnati, Anna Hemnes, and Mo Jane. And thank you very much.
Thank you so much, Mona. Both talks were very elegant, very deep, very thoughtful. Thank you so much. We should steal you back from the States and come back to your hometown. Great. So the last talk today is going to be about GDF2 gene mutation in high inbred population with PAH. This is a work done at King Faisal Specialist Hospital, and it's going to be presented by Dr. Lama Abdi. Lama is a scientist from King Faisal Specialist Hospital in Riyadh.
So good morning, everyone. It's a pleasure to be here. Thank you very much for the kind introduction. It's a pleasure to introduce you to what we do in our lab, specifically when it comes to gene discovery and the type of work that we do in our lab. So I'll get started. So pulmonary arterial hypertension is a relatively rare disease that looks at elevation of blood pressure, specifically in the pulmonary artery system, and it has many different factors, many different things that affects or leads to the progression of this disease. But my take-home message from the slide is the very last section, which specifically talks about the different types of PAH, and it could be idiopathic, but it can also be hereditary, and that's basically what I'm going to talk about today. So why genetics in PAH matters? Because genetics can confirm the diagnosis that we perform in the clinics, and it can also guide prognosis, a refined clinical diagnosis, and also offer more defined screening for the families. In these instances, idiopathic can become genetically explained. So this can open doors for genetic counseling, can offer families preventive measures, and can also help refine the clinical diagnosis. So just a very brief introduction into why does it matter when it comes to offering the family counseling is if we deal with an autosomal recessive versus autosomal dominant disease. With autosomal recessive, we typically tell the families that there's a 25% chance of having an infected child. But if we're dealing with an autosomal dominant disease, then we tell the families that there's about 50% chance of having an affected progeny. So incidence rates of heritable PAH, and this is one of the main slides that I would like you to pay attention to because it really drives home the message of why is genetic testing very helpful when it comes to diagnosis. Is for many of these diseases, clinical testing itself or by itself is not really sufficient to have a definitive diagnosis for these patients. We do observe this in a wide range of disorders, not only PAH. Actually, there is a very recent study that was published last year where we looked in an unbiased manner of all the reports that came for genetic testing, and we compared the molecular outcome with the clinical indication, and about 40% of those reports had the wrong clinical diagnosis. So this really drives home the message of the importance of molecular testing for these patients. So we're going to talk about data now. So this is 7 families, all from the same Saudi clan, that were referred to pulmonary hypertension clinic, specifically to Dr. Dal'an's team, and they were examined. So their ages were between 14 and 38 years. So they had all the amazing workup that they can get in the clinic, and also had biochemical assessments. All the investigations were performed, and all the imaging were also performed. And as part of the standard care in our hospital, they were also referred for molecular testing as well. So in molecular testing, we've identified a single variant in GDF2. It's a presumptive loss of function variant. It's a homozygous deletion in exon 2 that we were able to molecularly confirm. And basically what we thought is, since this variant truncates the protein in half, could it potentially lead to complete loss of function of the protein? But basically this was not indicated in the experiments that we performed. So all of these patients had the same exact variant, and thanks to Dr. Dal'an's and his team's help, we were able to stratify the patients into high risk, low risk, and intermediate risk, and I'm gonna walk you through the data now. But basically, why GDF2 and why does it matter? So we know that heterozygous variants in GDF2 are associated with hemorrhagic telangiectasia, and they're not necessarily linked to PAH, but it can be part of the complications, although the pathway is not really fully understood. So although OMIM only lists this particular association for GDF2, we also think that there is enough body of evidence to link GDF2 with pulmonary arterial hypertension. Specifically, there are 3 different studies that reported on the association between GDF2 with PAH. And these 3 studies also reported in younger populations and all had homozygous variants. So the first one, with Upton et al., basically reported a deleterious missense variant. Hodgson also reported a potential loss of function variant, and the last one reported a complete deletion of the gene in a younger patient that had association with PAH. Also, biologically, GDF2 also makes sense, because if we look at the pathway again, we see that GDF2, and I don't know if this would show. So GDF2 here, it sits right over here, and we see that downstream of it, there is BMPR2 as well as MAD8, and both of these genes are associated with a pulmonary hypertension. So we think that we do have enough, we don't have the indication in OMIM. We think that we have enough evidence that this is potentially the causal variant for this family. So if we look at the stratification of the patients, if we look at the first group, which is the more severe group of patients that had severe functional class 3 and class 4, reduced 6-minute walking, their labs and their examinations also reported very severe indications. They were consistent with advanced right ventricular dysfunction and poor short-term prognosis. And if we looked at these families very closely, so these are the 3 families here with their extended members as well, we see that the first one had a homozygous GDF2 variant, but the other 2 had a heterozygous variant. So we thought that was odd. We thought it would always segregate with the genotype. Whenever we had a homozygous, it would always be severe, but we were surprised that that was not the case. So if we move to the intermediate, which had moderate function of class 2 and class 3, and also their labs were indicative of ongoing pulmonary vascular disease, partially compensated by right heart function. So if we look at the 3 families that were classified as part of this intermediate group, what we observed is, although the first one was homozygous, the other 2 were also heterozygous. So we thought that was odd as well. For the low, which had maintained class 2 function and had a stable disease and favorable outcome, we were also surprised that he was homozygous for the variant. So basically what this tells us is there is no clear genotype-phenotype correlation, and we'll get back to that in a second. But first, we wanted to make sure that our cohort did not have any features of HHT, which is associated with GDF2. So these patients were examined, thoroughly examined, and we did not really see any defined features of HHT. In the, for transparency, we would like to also mention that only one patient had some sort of imaging that indicated maybe there is a hepatic artery branching, but it was not specifically linked to HHT. So as I mentioned, GDF2 has no clear genotype-phenotype correlation. What we saw and what we observed is that our cohort is relatively young, so there is very young, this manifestation appears in younger cohort, which really suggests that this could be an aggressive nature of PH for this particular genotype. Excuse me. So I mentioned that all of these 7 families were from the same tribal affiliation. So this basically gives us the opportunity to call this a founder variant. So a founder variant, I'm going to explain it in a second. Basically, there is a founder effect. There is an isolation of a community. It could be due to geographical reasons, for example, as we've observed in Finland. It could be due to religious, where it's not really permitted to marry outside of a specific religion. It could be cultural. Some cultures like to preserve their heritage, so they do not permit the marriage from outside of specific cultures. And we've quantified this in Saudi Arabia. Previous studies in my lab have showed that there is clustering, meaning that there is tribal clustering, so some certain variants remain clustered within a specific tribe. And very rarely do they jump to a neighboring tribe. So couple that with consanguinity, which we know is very highly prominent in Saudi Arabia, we get a very powerful combination. What we see is an enrichment of autosomal recessive diseases compared to other modes of inheritance. So for example, this was quantified by my lab as well, where we looked at neurodevelopmental defects or neurodevelopmental disorders in Saudi Arabia. So with the help of consanguinity as well as the founder effect, we see that about 75% of autosomal recessive, 75% of neurodevelopmental defects are actually due to autosomal recessive forms of this disease. If we compare that to Finland, where there is a founder effect, but there is no consanguinity that we know of, we see that only about 20% of neurodevelopmental defects are explained by autosomal recessive diseases. So basically, this drives home the idea that we have 2 powerful factors that are really enriching for recessiveness in our community. And it's really unfortunate because if we look here in publicly available databases, the Middle Eastern population is very underrepresented. I believe only 3,000 exomes and genomes are deposited in public databases. So the contribution of the founder variants are very specific to the Middle East is not really out there for people to mine and understand. So these founders provide opportunities when it comes to gene discovery, as we're discussing right now. So it helps us make connections, make us think that these genes are causal, or maybe these genes are dispensable. We don't really need them for day-to-day functions. It also helps us with variant interpretation, meaning calling a specific variant to be causal, or maybe not so causal, or maybe benign. It helps us also inform public health policies when it comes to disease burden, which diseases are more prominent in our community, and also make projections of what we think is going to be even more prominent in the future, offer preventive measures for families, give us insights into multifactorial diseases and druggable genome discoveries, and also population history, which we touch on also. So this is really important because whatever we discover in a research basis also is moved on to clinical use. So basically all the novel gene discoveries, all the variant interpretations that we make it into the clinical lab so they're able to have a more refined reports sent back to the physicians that order these tests. So with that, I would like to offer a concept of penetrance. This is a genetic concept where usually if you have 100% penetrance, meaning whenever you have the dominant allele, you will always have that trait. However, there is something called incomplete penetrance, where even if you have the mutant allele or the dominant allele, sometimes you do have skipping of that trait, and basically what you present is with a mutant trait. There's also variable expressivity, where you have a combination of the two traits together. So this is very important because with the large cohort that we have, we're able to calculate penetrance for our variant. So what we've done is, with the database that we have, we identified 6 additional patients with GDF2 variants, and we also had them referred to the clinic for thorough follow-up, and we saw that some of them did not really have PAH. So that means that we do have incomplete penetrance for this particular variant, and we were able to quantify it. So we were able to find that 43% of homozygotes have PAH, while only 14% of carriers or heterozygotes have PAH in our cohort. Also gives us the opportunity to perform mutational dating, so we can calculate the age of a specific mutation. Because we saw a large number of patients with this particular variant, we wanted to know how old is this variant, how long has it been in our population. So basically, we can use a specific algorithm that looks at the haplotypes surrounding that particular variant and can basically predict how many recombination events happened over time. We like to compare this particular variant with another variant that we know is trans-tribal, meaning it's present in multiple tribes. But they both have the same allele frequency in our database. And it was not surprising to us that this particular GDF2 variant was about 3.1 generations, originated 3.1 generations ago, while the trans-clan variant was much, much older, with about 65 generations back. So final thoughts I'd like to leave you with is, even though PAH mainly affects patients in their 3rd and 4th decade of life, we do have a younger population here with potentially explained genetically by GDF2-related PAH. So we know that GDF2-related PAH appears to be highly variable clinically. There is no clear genotype-phenotype correlation. We don't always say that homozygotes will present sooner or with a more severe phenotype. We define the characteristics of these patients with PAH and other carriers. We hope that we're able to uncover some phenotypic variability or understand why is the phenotype and genotype do not always correlate. Maybe that would be also explained epigenetically, as has been mentioned before. We were able to show that this variant is ancient, but it's not as ancient as the transclanal variant. And we also highlight the importance of extended family screening and how genetic testing can inform screening going forward in the future for specifically this particular tribe. With that, I would like to thank the people who actually made this work happen. So this was started in Dr. Fuzan Al-Kharayaz's lab. It was spearheaded by Dr. Siban Alif, a postdoctoral fellow in our lab, as well as all the technologists and coordinators that helped. And of course, a special thanks to our collaborator, Dr. Dalan, as well as the patients and their families. And with that, thank you, and I'm ready for questions.
Thank you very much, Nama. This was great, very interesting talk. And with that, we're gonna open the discussion here. So Dr. Irene Wang, please.
Thank you for a very interesting session. It was entirely driven by females, obviously. I have a question for every one of you, and the first one is for Sony. Is there studies on BMPR2 carriers and epigenomics? Because those would be the candidates that I would like to look at, because all you are showing could be the consequence of PAH. It's so hard to distinguish what is due to PAH in terms of epigenetic changes versus what is preceding PAH, right? So what if you were to look at if you were to combine genomics and epigenetics and look at the BMPR2 carriers that are asymptomatic?
No, I mean, it's an excellent question, Irene. So I think at least what we try to do is from the iPSC, which we have from the BMP2 carriers, and try to work with those cells. But my major problem is that we don't see any phenotype of this BMPR2. So you don't see any vascular changes that are happening either with the endothelial cells proliferation or smooth muscle cells, or any abnormalities is not prominent until you add a second hit, which is majorly this LTB4. So if you add this, then you can see a phenotypical changes. That's where I think it was not studied, but I assume that the epigenetic changes will be occurring due to genetic mutations. So we did this now with the TBX4 mutation carriers. And there we can definitely have a phenotype, and we see, because TBX4, it's also like a super enhancer and a transcription factor, so we see a lot of epigenetic alterations that are happening due to a mutation background. So it is there, but I think a second hit in BMPR2, selectively a second hit matters. Yeah.
Thank you.
Thanks.
So in your eyes, second short question to you, what is stiffness in the eyes of a laboratory researcher? It's
So-
a different stiffness, right?
Yeah, it is different stiffness. So I think what we are trying to do is like to apply different Pascals. So different stiffness of different variations. So we are just working with this kind of matrix that gives different Pascals, and then we are working with that. It's not different orientations. That's also the plan which we want to have to work on that. Yeah. Did I answer you?
Well, maybe I ask you afterwards.
Yeah, okay.
So I have another question to the metabolomics, to Mona. So is there a metabolome of pH treatments?
Yeah, I mean, that's what we're trying to, I guess, identify. I think the main issue from our end, and we were able again to do that in like a small cohort, the main issue we find is the cohort, right? So when we reach out to like lots of clinical trials, for example, unfortunately a lot of them do not store samples, at least in the past. So until we have that, until we have large, data set, all what we have is smaller cohorts before and after and whatnot. But what we found here was interesting that yes, we do see, some signal of therapy. I don't know if that answers the question.
I would propose to you to look at parenteral prostacyclin users, how that it must affect the metabolism.
Oh, absolutely it does, yes. Because specifically in this platform, when we focus on the bioactive lipids, of course it does affect that. So we do correct for it in our therapy, and we'll do sensitivity analysis before and after and whatnot, but yes, it does.
Dr. Alabdi, thank you also for your presentation. You talked about corkscrew vascular morphology, and you had an arrow there in your picture. What did the arrow point to? Was this a venous change in HHT, in this GDF2 carriers? Or?
Oh, no. So this was from a previous study that was done, I believe it was done in the earlier 2010s. And they did mention that the particular younger child had an abnormal vascular morphology that was shown in the image.
So the tortuosity, is that simply the consequence of high pressure? Or is it a vascular malformation?
I will have to double check on that study again, but sorry.
To rule out HHT in these patients, we did actually MRI imagings for all of them, look at the deep organs to see if there is any evidence of that. All of them were negative except that patient. There was some tortuosity of the arterial system, but it did not fulfill the criteria for vascular abnormality that we see normally with HHT. So it was just some sort of anatomical variant of no clinical significance.
Okay, thank you.
Hassan?
Thank you, Abdullah. Thank you all for the elegant presentation. I have a challenge for my understanding first, and I hope that you'll be able to clarify it, starting from Lama's. Well, the question is for the three of them. Maybe Lama is just the instigator of the question. You have a patient who has some genetic defect that does not necessarily translate it into disease or phenotype, and we assume that the epigenetics are part of this process. But this is a dynamic process, and how do we know, maybe another rephrasing of Irene's question, how do we know that we're dealing with an epigenetic as a cause or epigenetic as a response to the disease and the genetic defect that we're having. So this probably might be an issue that I just have a problem with understanding it because it's a dynamic process. And how do we translate this into the metabolic part, the endotyping of this metabolome? If we have a group from Saudi Arabia and we do the same basic work on them, do you believe that we will identify the same metabolome? And differences in the genetics, epigenetics, and other things might influence the outcome of this metabolome that you're studying. So how can we link all together? And the other question for Muna is, how easy is it to really, get the metabolome, assuming that we successfully validated the data? How easy it is, is it relevant? It is so cheap, available, and doable that we can utilize it in a clinical grounds. Thank you.
Sure, I can start with that question, specifically since I'm a biochemist, so I can touch on each one of the points that you've mentioned. So it's well known in genetics that there are variants that provide a risk factor, and we believe that GDF2 is the variant that we've identified provides high risk for PAH. Does epigenetics play a role? Yes. Because in all diseases that we've discovered, we know even in early embryonic development, the interaction between the mom, the environment, the uterus, the fetus, the whole of the being that we are does influence progression, does influence phenotype, and does influence the biomedical and the biochemical interactions. Do we think that the Saudi population, if we screen them, will identify the same metabolites that we've observed in San Diego? I would say maybe not, maybe yes. Maybe we'll see some overlap between the two, but given the high genetic diversity in San Diego compared to the difference between what we see here in the Saudi population, I would say I would err on the side of maybe not. But it would be a cool thing if we see that both signals that we get are for the same pathway. That would be cool, because then it would tell us that potentially that's a target that we can develop therapeutics for. I will leave the floor for my colleagues.
So I would say, the epigenetics is both a cause and a consequence, because now a lot of mutations, also a decreased expression of the DNA methyltransferases is known. It's not need to be only a mutation, but there is a change in the epigenetic modulators at baseline. So that's why I think maybe they can drive by themselves the epigenetic changes. Like, for example, if you take TET2 mutants, they develop TET2 knockout mice, they develop a spontaneous pulmonary hypertension where we have a problem of the BMPR2 mutant rats. Some of them develop, some not. Some labs they can reproduce, some labs cannot. So this is where I really think it's involved. It's a causative agent, and it's also definitely involved in the progression in combination with the genetics, because not every patient who has a mutation develops PH. So they definitely need an environmental trigger. It could be an internal or external environmental triggers, which has to modulate epigenome, and then you have consequences of the disease.
In the process. So again, how am I going to tell this particular one? The studies might show something in one area, another lab might discover another epigenetic consequent to the disease. So how easy it is to really say this is a cause and this is an effect, and that's my complexity of understanding.
You are absolutely right. So because I think before even understanding the whole epigenome, we went very fast in going to the preclinical models and trying the modulating HADs, HDACs, BRDs, and so on. That's where we complicated the whole field. We really didn't understand the basic biology, but we went for translation, which gives us an additional value of the epigenomic-based therapies. But I think what we need to understand is what is the changes that are occurring and what are consistent and which stays for a longer time. That memory, I think this is what we need to target. And this is what I feel, which has to come up, yeah.
Yeah, I will just add up. I totally agree that it is very challenging to know the cause or the effect. For metabolomics, what we're trying to do is we're trying to look at patient before the development of this disease, like for example, either looking at this gray area of some dysfunction or some elevation in PA pressure, but not quite pulmonary hypertension. In that community cohort we looked at, they were normal with some RV changes, and then that kind of give us an idea. And just to add up, to answer your question about the regional, I think we absolutely need to have geographical or regional presentation. And unfortunately, at this point, we still don't have it within the same platform, but that's why we want to do it. Because if we don't do it, we don't know. I mean, I could tell you what I think, but it doesn't matter. And then lastly, how easy it is to kind of translate to the clinic. It's not that hard. Once we know that, for example, we have a X, Y, and Z metabolite, we can build like a ship or like whatever it is that you could put a drop of blood and it could tell you high risk, low risk. And so it's not hard to build conceptually, but we need to validate and confirm that, yes, is this universal, is this not, and whatnot. But hopefully through collaboration, we can do that.
Inshallah. But again, the complexity comes from the dynamicity of the phenotype.
Absolutely.
So it might change by anything.
Yes.
The background, the environment, the food.
Absolutely.
Thank you.
But ولك في cancer, like, you know, yes, exactly. We're just behind.
Amazing talks, amazing discussion. We are only 5 minutes behind time, so we'll take a break now. And the next session is, I think, more appealing for mostly clinician attendees. So it's gonna start at 9:45. Please make sure you come back on time. Thank you so much.
السلام عليكم ورحمة الله وبركاته.
Pathogenesis, prevalence of PAH and connective tissue disease, diagnosis, prognostic and risk factor differences between idiopathic and connective tissue disease related.
As I'm part of the SAPH family for many years now, I like to come to this conference year by year. The organization, as usual, is perfect, and the quality of the scientific program is outstanding.
We have bring many people from different countries, and from Europe, from US, and from Saudi Arabia and Dubai and many other countries.
Many colleagues that really worked very well. From the international speaker, I really need to say that the PBRI had a great role in SAPH success. So I'd like to have basically special thanks.
Where you can meet all the experts in the field, get all the updates, and get to know your colleagues and mingle with them. SAPH is an amazing team, which we are really proud of, and I hope it will continue for years and years to come.
ارتفاع ضغط الدم داخل شرايين الرئة وليس الضغط العام. وبما أن يرتفع ضغط الدم داخل شرايين الرئة والقلب تدفق الدم من الجهة اليمنى من القلب يدخل الرئة. فإذا كان هناك ضغط عالي في شرايين الرئة يأثر على عضلات القلب بالذات الجهة اليمنى مع الوقت.
نعم ممتاز. طيب هل هو يعتبر مثل ما ذكرت في البداية أنه هل هو مرض نادر وهل تشخيصه سهل؟ وبرضه يا دكتور يعني كيف تشوف طريقة علاجه؟
هو طبعا نادر لكن لها أسباب كثيرة، بعضها شائع وبعضها نادر، وهو يصنف إلى عدة مجاميع. هناك ما تسمى المجموعة الأولى وهذه هي اللي نادرة، وعادة يعني بالطروحات والنشرات العلمية تقريبا بين 50 إلى 100 كل مليون يصابون في هذا المرض. وهو طبعا بحكم انه نادر وبالتالي يكون تشخيصه في العاده صعب شويه، وعاده التشخيص يعني يتولى عمليه فرز اسباب المرض، لانه هو صحيح المحصله هو ارتفاع ضغط الشرايين الرئوي، المحصله النهائيه، ولكن لها عده اسباب. فدقه التشخيص تكمن في فرز هذه الاسباب، وبالتالي وصول التشخيص الدقيق، وبالتالي ايضا العلاج الدقيق، لان لكل مرض لكل سبب من هذه الاسباب علاج.
استديو رئيس الجمعية السعودية لمرض ارتفاع الضغط الشريان الرئوي الدكتور عبد الله الضلعان. أهلا بك دكتور عبد الله، مساء الخير.
أهلا وسهلا.
دعني أبدأ معك في البداية عن تعريف يعني بهذا المرض.
الريبورتاج المقدم لخص المرض لكن ببساطة تعريفه هو ارتفاع الضغط داخل الشرايين الرئوية وليس الضغط العام للجسم. وكما هو معروف ضغط الشرايين الرئوية دائما يكون قليل جدا ليستوعب تدفق الدم من القلب.
So, ladies and gentlemen, welcome back to the next session. Together with Abdullah Aldalan, I welcome you to session 2 here, or session 3 actually, which is called PAH Management TGF-beta Superfamily Pathway. And it's a great honor to introduce someone who actually doesn't need an introduction here, who I think has been here for probably all SAPH meetings along the years. So Artie, you must have been, this must be your 19th time, because this is the 19th meeting. Welcome again, and the title of your talk today is Sotatercept in Newly Diagnosed PAH, Efficacy and Safety Data from Hyperion. Please.
Assalamu alaikum. Dear Stefan, thank you for the very kind introduction. Dear Abdallah, dear organizing committee, thanks for having me back again, despite the fact that I missed a couple of the meetings, unfortunately, due to my lack of reliability in the past few years. But in fact, you know, I was there when at least some of the very first meetings took place with my friends Majdi and other colleagues that I really cherish. The collegial atmosphere at this conference is really unique, and I very much like to come back as long as I'm invited. So thank you very much for having me. Today it's my pleasure to present the results of the so-called Hyperion study. This is about the efficacy and safety of sotatercept in patients with newly diagnosed pulmonary arterial hypertension at intermediate to high risk of mortality. And this is an encore presentation to the one that Professor Valerie McLaughlin gave at the last ERS. So it is actually the reflection of what has been presented there. And since then, you're all aware, the main manuscript has been published in the New England Journal of Medicine for all of you interested in reading into the details of this study. We have heard yesterday from Paul Yu extensively about the suggested mechanism of action. Without a doubt, sotatercept is a scavenger of ligands of the activin receptor signaling pathway. Whether or not it's really working through rebalancing with the BMP pathway, I think we have increasing doubts about that. It will play a role, but it is certainly not the only simple explanation, and there are many other potential explanations, but without a doubt, it's a very potent scavenger of the ligands of the activin receptors, both A and B, but predominantly activin receptor type 2A. So the benefit of sotatercept had already been established in prior phase 3 randomized controlled trials, actually starting already with a very remarkable phase 2 study, but then continued in the STELLA trial in patients with WHO functional classes 2 and 3 on extensive background therapies. And these patients also were very prevalent patients. And this is in clear distinction to what you're going to see in the Hyperion study population. In this very prevalent patient populations, there was an improvement with adding sotatercept on top of the background therapies that were mostly extensive background therapies by 40 meters and an 84% reduction of risk in clinical worsening events. The Zenith trial, which was initiated later, aimed to include patients who were more severely impaired by their disease. So patients had by definition to have either a functional class 3 or 4 clinical severity, and there it could be shown that even although the trial was prematurely terminated, there was a 76% reduction in risk of major morbidity and mortality event at this interim analysis. The interim analysis was pre-specified, but then the trial was stopped prematurely and nevertheless showed significant reductions in the risk for this morbidity and mortality events. So in this study, the aim was to include patients who were diagnosed within 1 year before inclusion into this study. So they were quiet incident. They were not by definition all incident, but they had to have their diagnosis being made within 1 year. And they could have all respective treatments that were available at that time and were indicated during that first year of diagnosis. And the key eligibility criteria were that they had to be adult patients, obviously, with a diagnosis younger than 1 year. They were in functional classes 2 or 3, and they had to have a REVEAL Light 2 risk score of more than 6 or higher, or a Compara 2.0 risk score of 2 or higher. And then they were randomly allocated to receive either placebo 3 weekly or sotatercept with a starting dose of 0.3 milligrams per kilogram, as it's currently also suggested for the marketed product. And then the target dose remained the 0.7 every 3 weeks, administered subcutaneously. Also in this study, there was a pre-planned interim analysis when more than or equal to 61 events were counted. And the aim actually was to include patients and then continue the study until to the final analysis, which actually didn't happen because of the premature termination, which was at the end of the day a collegial decision between the steering committee and the sponsor of the trial, because there was a loss of clinical equipoise with the wealth of data that were available until then. So these are the study endpoints. The primary endpoint is interesting and worth looking at. It was a composite of death from any cause, hospitalizations more than 24 hours for PAH worsening, atrial septostomy, lung transplantation, and then the deterioration in performance in exercise testing due to PAH, defined as a 6-minute walk decrease from baseline, and at least one of the following: worsening functional class, signs and symptoms of increased right heart failure, addition of a background PAH therapy or change in the background PAH therapy delivery route. So meaning when you were on a non-parenteral prostanoid and had to be switched to subcutaneous or IV, that would be considered a clinical event worth fulfilling the criteria of deterioration. You might stumble over the fact that the deterioration 6-minute walking distance was not quantified as it was in previous trials. So it didn't say it had to be at least 10% or at least 15% as we had in previous trials, but any deterioration could have been 1 meter below the own baseline at time of randomization would be considered a clinical worsening event if it was corroborated at the same time by a worsening in functional class and the other items listed here. There were a couple of secondary endpoints, and these were more or less, so to say, positive or gaining endpoints. For instance, the proportion of patients achieving a multi-component improvement at week 24, proportion of those who maintain or achieve a low risk and reveal, proportion of patients who maintain or achieve a low risk in the French risk score, and or a change in NT-proBNP, proportion of functional class improvements, positive gains in 6-minute walking distance, overall survival, and so forth. Remember the order of the secondary endpoints because they were subjected to a hierarchical testing, meaning that as long as the endpoints in that subsequent order remain to be statistically significant, the next would also be analyzed for statistical significance. As soon as one of the secondary endpoints is not statistically significant anymore, you are not allowed by these hierarchical testing to further elaborate on p-values. The disposition of the patients is shown here. There were 441 patients screened, of which 120 screen failures happened. Ultimately, 320 patients were randomly allocated to either receive sotatercept or placebo, of which in the sotatercept arm there were 17 premature discontinuations and 11 in the placebo arm, with a mean follow-up time of 13.2 months. This is the so-called contemporary PAH formulation showing us that these patients in average were older than in previous trials. They had more comorbidities when they were included, and they were at that time, because of the more recent diagnosis, not as extensively pretreated as patients in the preceding trials. So as one example, the prostacyclin infusion therapy was only given to 16% of the patients, while the other parameters, actually including the 6-minute walking distance, the NT-proBNP levels at baseline, and also the PVR, were quite well matching the study populations that we saw in the previous trials. Also keep in mind that having the diagnosis quite recently, as opposed to the very prevalent patient populations from STELLAR and PULSAR that were on average 7 years from diagnosis when they were included, implies that there might also be a so-called immortal time bias present in the other studies, meaning an enrichment of survivors in the preceding studies, and this is more reflective of real-world patients that are included shortly after the diagnosis has been made, and the assumption should be or could be that they tend to deteriorate at higher frequency as opposed to an enriched population of survivors, which was the case in the preceding studies. Nevertheless, there was an impressive reduction of 76% in the occurrence of clinical worsening events. Kaplan-Meier curve started separating very early, actually after the first 3 injections of sotatercept. There was a hazard ratio of 0.27, meaning that there was a 76% reduction in the occurrence. And then on the right-hand side, you can see what the primary endpoint components were composed of. It's noteworthy that the response or the significance was mainly driven by the last item listed down there, which is deterioration in performance in exercise testing, bearing in mind that losing 1 meter could already qualify you for fulfilling such an endpoint. However, as mentioned before, it had to be corroborated by an accompanying worsening in the functional class and or other clinical events. When looking at the disposition of the patients in predefined subgroups in this forest plot, all the effect is showing to the right direction. Sometimes the arrow bars cross the line of equality. However, this is also mostly present in underrepresented or small small subgroups. Nevertheless, in a forest plot, the most important reading is that it should go all in the right direction, which it does. Regarding secondary endpoints, as I mentioned, they were hierarchically tested. The difference in the proportion of patients with multi-component improvement, the secondary endpoint was positive and was increased by 14.45%. The difference in percent of patients with a low risk reveal light score of equal or less than 5 at 24 weeks was also higher in patients receiving sotatercept. This was significant. However, the study population did not meet the difference in patients with regards to the low-risk simplified French score, and we may elaborate later in the discussion with Olivier, who is the inventor of this simplified French score, what possible explanations might be responsible for it. And because this did not meet statistical significance, the other ones, although showing numerically in the right direction, were not tested for significance anymore. Safety is an important issue with sotatercept. We had discussed and we may discuss in more detail about this, and it was actually reflecting what was observed in previous trials, and we will hear much more about safety and long-term safety from the Soteria trial, which actually invites all patients that have participated in previous randomized control trials to be further treated, and Joanna Preston will tell us more about long-term safety. Here, the signals that were observed previously were also observed, including bleeding events, serious bleeding events, increased hemoglobin, telangiectasia, and also some AEs with an incidence of more than 10%. For instance, epistaxis and telangiectasia. There was no difference in pericardial effusion in that trial, and we were all sensitized that pericardial effusion might be a problem with activin ligand traps, sensitized by the KIRA study results, where this was clearly more frequent than in the active group than with placebo. There is an emerging signal for sotatercept as well, but it is very, very rare. Nevertheless, we have to keep an eye on that. One thing that is a little bit different from the previous studies is that the percentage of the occurrence of these AEs that have been listed here was slightly more pronounced than in previous trials. May it be that we are all more sensitized to look for some of them, or that really this real-world patient population that was included is more prone to experience some of those side effects. This is a matter of discussion, and definitely we will have to look on that very rigorously on long term. So allow me to summarize that in Hyperion, sotatercept initiated in a PAH population within the first year of diagnosis led to a reduction of clinical worsening events as defined by the study protocol by 76%. The translation of the numbers needed to treat is 5 at 12 months, so 5 patients need to be treated to prevent from one of these events occurring. The prompt benefit over placebo was shown very early, as shown in the Kaplan-Meier curve. This study population was slightly older, had more comorbidities, was mostly intermediate low risk as defined by the comparer risk score, mainly on double treatment with a minority on prostacyclin infusion. We can also discuss about the issue of background therapies. There was a consistent treatment effect, as we saw in the forest plot, across all pre-specified subgroups. There were statistically significant improvements in the secondary endpoint of the multi-component improvement endpoint and the Reveal Light 2 risk score. And then the other ones, for the explained reasons, were not tested for statistical significance, although they showed numerically in the right direction. The safety, as it says here, was manageable and generally consistent with prior sotatercept studies and its current label. So in conclusion, Hyperion expands the evidence-based positive benefit risk of sotatercept from prevalent PAH patients to patients within 1 year of diagnosis. There was this rapid divergence of the benefit that we saw, and sotatercept is generally well tolerated in this population with a manageable safety profile. And as I mentioned, we will learn more about its safety profile from the ongoing sotatercept study. So I'd like to close with some references and also with a thanks to my steering committee colleagues in the clinical development program of sotatercept, all co-authors on this publication. And most of all, I want to thank our patients who volunteer to participate in these important clinical trials and thereby move the field forward and allow us to develop treatments for future generations of patients. Thank you for your attention.
Artie, thank you very much for this very comprehensive presentation of the results of the Hyperion study. You may join us here because we'll have Q&A at the end of the session, as always here.
Next lecture will be actually the logical continuation, which is sotatercept long-term efficacy and safety, SOTERA study by Dr. Ioana Briston from La Haye Medical Center in Burlington, Massachusetts. Welcome.
Thank you so much for, wow, this is loud, for inviting me again. It is such a pleasure to meet friends again and discuss very important aspects of pulmonary hypertension. So I will be presenting the new data on SOTERIA on behalf of my co-authors and steering committee. As a reminder, the sotatercept development program includes these specific clinical trials, and all patients who finish one of the 5 clinical trials have the opportunity to stay, to come into SOTERIA for a long-term treatment with sotatercept. Among the 5 clinical trials, SPECTRA is the only open-label trial. The other 4 are the international multi-center randomized controlled trials that you are familiar with, and large, based on our PAH experience. To mention also cadence that Professor Vacheri will be talking about, LIGHT-TRAY is looking at a fixed dose of sotatercept in PAH and the pediatric PAH clinical trial, which is still ongoing. So for this particular analysis, this is the second interim analysis of SOTERIA. We thought of going more in depth and analyzing patients from the beginning of their exposure to sotatercept. So for example, if patients were treated with sotatercept in an active arm in the parent study, the index date of the exposure was day 1 in the parent study, whereas patients who were in the placebo arm in the parent study were indexed as day 1 at their time of rollover into SOTERIA. That's how we thought it's a more accurate exposure to sotatercept. We also had 3 cutoffs analyses, and the last one for this particular data presentation was in August 15, 2024. The median follow-up of these patients was 2.5 years. The primary objective of SOTERIA is safety. We need to learn long-term safety of this not only new first-in-class drug, but also first biologic for pulmonary arterial hypertension, with secondary objectives including efficacy parameters that are clinically important for us and our patients. As of the last cutoff, 431 patients with sotatercept have been analyzed. And I should say, although we have 5 clinical trials that roll patients over into SOTERIA, at the time of the cutoff, both Hyperion and Zenith trials were still ongoing, so we do not have data including these particular patients yet. The next cutoff will include probably, I would say, almost 900 patients, so more information to come. The characteristics of patients in Soteria mirror in a large part the Stellar trial patients because the majority of patients came from Stellar. The age was around 48, majority females. The pH etiology is showed in the table with a small representation of connective tissue disease patients. The functional class patients 2 and 3 were in preponderance, and a little over half of these patients were treated with triple therapy. A third were on double therapy. If we look at the summary of the safety events, after a median of 2.5 years of follow-up, we can see that among the 431 patients, the cumulative exposure was it was 1,200 person-years at risk with an exposure of almost 1,000 days with a range up to over 6 years. Among all adverse events, you can see that although there were significant events that were considered related to treatment, and there were some, almost 40% led to dose delay, 14% led to dose reduction, and I think that's important because over a long period of time, we have to be cognizant that some patients will need adjustment of their dosing. But only 8.6% led to treatment discontinuation in this longer trial, and 4.6% led to discontinuation due to death. Among serious AEs related to treatment occurred only in 7.2%, and to treatment discontinuation 6.3%. This graph on the left-hand side summarizes the frequency of AEs of interest. The most common AEs of interest were bleeding events occurred in 58%, with 9% of these events being serious bleeding events. Among the bleeding events, epistaxis was the most common cause of bleeding. Followed by increase in hemoglobin, thrombocytopenia, telangiectasia in a third of the patients, but none of these events were deemed serious. An increase in blood pressure and thrombotic events in 8% of patients, with 3% being serious. Now, if we look at the time from initiating sotatercept to the first bleeding event, I'd like to focus first on the bleeding events because they're most common. If we look at all bleeding events, it occurred in almost 58% of patients. The median time to event was around 100 weeks, and serious bleeding event occurred in less than 10% of patients. Among the most common bleeding events, there were epistaxis. And if you look on the left-hand side, the Kaplan-Meier curves of epistaxis and serious epistaxis show the occurrence over time, over weeks, with 38% of participants having epistaxis, and among those patients, only 3 patients had serious epistaxis events. But when do these events occur? When do these events of interest occur over the length of treatment? We performed 2 types of analyses. The first analysis was an exposure-adjusted incidence rate, depending on the first, second, or third cutoff of analysis. The first cutoff was end of 2022, the second was end of 2023, and the third was in August of 2024. The first median follow-up was almost a year, the second was 1.8, and the third follow-up median was 2.5 years. If we look on the left-hand side for all adverse events, all three cutoff times showed a similar occurrence or incidence of all adverse events, as well as each of the adverse events of interest, including bleeding events and epistaxis. If we look at the serious adverse events on the left-hand side, we see the same pattern with the same and similar occurrence between the three cutoff times. To look a little bit differently at the event time over time, we did a second analysis, and we looked at adverse events of interest during year 1 versus year 2 versus year 3 of sotatercept treatment. And you can see among all AEs, as well as serious AEs that are in darker green, the telangiectasia, as well as bleeding events and epistaxis, thrombocytopenia, increased hemoglobin, as well as thrombotic events. The proportion of patients experiencing these AEs of interest and SAEs of interest were generally stable by year. So there's no signal so far that there is an increased incidence of adverse events of interest during accumulation of exposure in time. However, we also can conclude from these two analyses that these risks of adverse of interest do not disappear. They do not happen all or most in the first year, and then it goes down. So I think we have to continue to be vigilant to follow these patients continuously and with the same caution and vigilance. Looking at the efficacy endpoints at baseline versus week 24, year 1 and year 2 of sotatercept treatment, we can see in the first graph that the 6-minute walk distance improved from baseline to week 24 and maintained at year 1 and 2. Of course, there are less patients at year 2 cutoff, so this is another limitation of an open label trial. Very similarly, NT-proBNP improved at week 24 compared to baseline and maintained its improvement at year 1 and 2, whereas functional class, in the last graph, the blue is functional class 2 patients and green functional class 3. Compared to baseline, there were more patients in functional class 2 at 6 months, with some patients achieving functional class 1 and non-functional class 4, and then the trend stabilizes or maybe improves at year 1 and year 2 with more patients in functional class 1 and 2. So the conclusions for the second interim Soteria analysis is that sotatercept remained well tolerated after long-term follow-up of a median of 2.5 years with a maximum of 6.2 years. There was a low discontinuation rate. The non-serious bleeding events were relatively frequent, but the majority were epistaxis, and all this has been consistent with previous findings. No new signals of safety have emerged from this interim analysis. In general, AE rates did not increase with extended sotatercept treatment, including serious events and bleeding events. The sotatercept safety profile was consistent over time, and the efficacy improvements were maintained over a 2-year period. So the long-term benefit risk of sotatercept in patients with PAH remains favorable with sustained efficacy and a consistent safety profile. But we can analyze patients in Soteria, and it's wonderful to follow them long term. However, many more patients are being treated in clinical practice around the world nowadays. So maybe Soteria is not enough to understand the long-term effects of sotatercept, especially on the safety profile, and we have to look beyond it too. So beyond Soteria, I would like to talk about the 2 important and rare possible side effects associated with sotatercept that we have not seen in Soteria. The first rare adverse event is of hypoxemia, and this is a letter published from the lead of Mark Humbert and Marius Hopper of 2 patients, one with congenital PAH, one with hereditary PAH. At the entry, at the start of sotatercept, they had a moderately decreased DLCO, and they had significant hemodynamic and clinical response. So they were good responders to sotatercept. But after 2 years of treatment on average, they developed hypoxemia. Their DLCO decreased further, and the investigation, a thorough investigation, showed a positive intrapulmonary shunt. They both, of note, had associated telangiectasia. So the sotatercept was discontinued, and their hypoxemia improved. And then we have to try to understand what is the mechanism behind this hypoxemia that is partially or totally reversible upon discontinuation. If we look at the scheme, we know that the ligand trap acts on the TGF-beta arm, but maybe not only, like Paul Yu was discussing yesterday, and maybe the interaction between different receptors and this ligand trap may be much more complex, that beyond the positive effects that we can achieve with sotatercept, by modifying the BMP 9 or 10, the emergence of HHT-like symptoms may be explained, such as epistaxis, such as cutaneous telangiectasia or mucosal angiodysplasia. And on the other hand, a more vigorous dilatation beyond what is needed in certain pulmonary vessel or regions may be accounted for the effects of hypoxemia in a hepatopulmonary syndrome-like phenomenon. And I think we can say that probably it's not an AV malformation that is developing, because the effects are at least partially reversible when we discontinue the offending agent, but more like patients with hepatopulmonary syndrome, in whom if we transplant the new liver, the HPS resolves in many cases. The second rare adverse event is pericardial effusion, already alluded to. There are emerging reports in the literature. I chose 2 of them. The first one experiences the sotatercept treatment in 24 patients. 6 of these patients had baseline pericardial effusions. All these 6 had enlargement of pericardial effusion at follow-up. 4 of these 6 were symptomatic. The PAH etiologies varied, so there was not a clear signal of a subtype of PAH that may be associated with pericardial effusion. The exposure was around 25 weeks. Interestingly, all had improvement clinically and in hemodynamics and echo parameters with the treatment. One of them had cardiac tamponade that required intervention. And sotatercept was either reduced or discontinued. The second case series is from my former colleagues at Tufts that described among the 10 patients that were treated, 4 had enlarging pericardial effusions. 1 had a new pericardial effusion. Again, the pH etiologies varied, and all had improvement in hemodynamics with sotatercept and also echo parameters. So this is an emerging information that we should be cognizant and integrated when we follow patients that are treated with sotatercept. Now, because of this emergence and signal that has initially been described with other ligand traps, okay. Sounds good, thank you so much. We looked at the 2 finished clinical trials, Stellar and Zenith, that had all patients had baseline and 24-week echocardiograms. So we looked and we analyzed their echocardiograms to see if there's a signal in the first 24 weeks. And this complicated table summarized shows that roughly 25% of patients at baseline have some degree of pericardial effusion, most trace or small. At 24 weeks, there was no signal of an increased pericardial effusions with sotatercept compared to placebo. The number of pericardial effusions stayed small and very similar in the 2 arms in both clinical trials. However, because we recognize this was a short-term follow-up by echo, only 24 weeks, we alerted all the investigators in Soteria, and currently the investigators are prompted to evaluate thoroughly any new episodes of hypoxemia, as well as be alerted and look for pericardial effusions if the patients become more symptomatic. So we hope that with the next data analysis, we will better understand the long-term effects of these 2 rare possible adverse events with the sotatercept treatment. So I would like to conclude by acknowledging and thanking our patients, the study investigators, and the site staff for very hard work on following patients long term. We know it's difficult to maintain patients in a long clinical trials, and we encourage the Soteria investigators to keep and maintain these patients in the trial as long as possible to accumulate accurate data continuously. Thank you so much.
Thank you very much, Joanna, for this very nice overview about Soteria and the other long-term data. So we now switch from Group 1 to Group 2 PH, and it's an honor to introduce Jean-Luc Vachery from Brussels in Belgium. Jean-Luc, you will talk about targeting the activin signaling pathway in CPCPH due to HFpEF, associated with HFpEF. You're here a couple weeks too early, I would say, because we'll hear about the cadence results very soon. But I'm sure you will tell us what we're allowed to say so far.
Assalamu alaikum. Thank you very much, Stefan, and my special thanks to Abdullah and the organizers for inviting us again in this to this very nice meeting. It's really a pleasure to be here and come every year and over. So yeah, the deal here is to talk about how to target the activin pathway in CPCPH due to heart failure with preserved ejection fraction. These are my financial disclosures. In fact, if you look at PAH, of course, this is where we got inspiration from with sotatercept to see how the treatment algorithm has changed over the years. And in contrast, this is how the treatment algorithm from group 2 pulmonary hypertension evolved since 1998 and the Third World Symposium. So there's basically nothing and no recommendation with the exception of course, treating the underlying disorder. There were attempts, however, to treat the precapillary component of pulmonary hypertension over the years, and I'm sure you're well aware of the trials that were done, mostly in heart failure, with a couple of trials which I'm sure you will recognize, and this is, of course, based on the current definition of group 2 pulmonary hypertension. You see that the focus has been mostly on heart failure with preserved ejection fraction for good reasons. As cardiologists, this is obviously one of the emerging diagnoses that we face in the clinic, and it's also challenging in terms of differential diagnosis from PH. And you see that there's quite a lot of trials that were done over a couple of years. However, one of the, I would say, hallmark of those trials is that some of them were interrupted and stopped prematurely for several reasons. That's the case for Soprano, a study looking at the effect of macitentan in patients with persistent pulmonary hypertension after implantation of a left ventricular assist device. It's also the case for SAPH with oral treprostinil, looking at patients with preserved ejection fraction with the primary endpoint being 6-minute walking distance. And that's also the case for Serenade, looking at macitentan in a group of patients with combined post and precapillary pulmonary hypertension, looking at a change in NT-proBNP at week 24. You can already see that the primary endpoint for those phase 2 trials are quite different, PVR, 6-minute walking distance, and NT-proBNP. And of course, we also had the result of the PASSION trial, tadalafil, that Stefan and Marius Upper spearheaded to look at something that was a little bit more robust, which is the primary endpoint of event-free survival. The trials were suspended because, mostly because of, with the exception of PASSION, because of slow recruitment and sponsor decision in the case of PASSION, but Stefan does know better than I do. The sponsored drug was withdrawn halfway of recruitment. It's interesting to look at why those studies struggle to recruit. And we saw that previously in the MELODY trial, and you will see a little bit about cadence in a couple of minutes. If you look at Serenade, the idea was to look at macitentan, again, in patients with CPCPH. It's a large screening group of 437. There was a run-in period where there was a drop by half of the cohort, and 200 patients were included in the run-in. There was a less strict selection, probably a little bit more focused on the heart failure world, not really focused on hemodynamics, and perhaps a little bit less experience in PVD, which might have led to more screen fails. Also, one of the ironies is that the same story repeated again. So patient in the group that was receiving treatment, in the analysis of the patient included, had fluid retention on macitentan despite the run-in phase, and this is something we looked at the same way with the MELODY trial. PASSION is very different. It's a German study focusing on PH center with a strict hemodynamic definition. It's not PVR as the primary endpoint. It's time to clinical worsening. There's a mistake on that slide. There was less of screen failures, and it's really a pity that the study was not completed by sponsor decision to discontinue the medication. So you have two aspects of the spectrum that I would like you to keep in mind when we'll talk about the next step for attempts to treat group 2 pulmonary hypertension. Now, we have 3 more recent trials that were completed. The HELP trial with intravenous levosimendan, looking at exercise wedge pressure. The DYNAMIC study, which is a phase 2b trial with riociguat as a study drug with cardiac output as the primary endpoint after 26 weeks. And the SPHERE-HF with mirabegron, which is beta-3 agonist, looking at PVR as a primary endpoint. Let me show you a little bit of the outcome of those trials, HELP, DYNAMIC, SERENADE, SPHERE, and PASSION. The question of study completion was, of course, raised with SERENADE and PASSION, but the other completed their recruitment and were analyzed. There's only one study that really met its primary endpoint, which is the DYNAMIC study with a change in cardiac output. And based on the available population, the other didn't meet their primary endpoint, with various other considerations looking at the DYNAMIC study. For example, there were more dropouts in the riociguat group as opposed to the group of patients with who were receiving placebo, and of course SERENADE being associated with an increased risk of fluid retention. This is, in fact, to go back to the story of pulmonary hypertension in group 2, I think it's always good to go back to basics and see where we perhaps made a couple of mistakes. In fact, the reasoning behind the treatment of a precapillary component in pulmonary hypertension associated with left heart disease has kind of a strong pathophysiological rationale, and this is what heart failure looks like. Irrespective of ejection fraction, you have several phenomena that would lead to vascular remodeling, that's endothelial activation and dysfunction, leads through all these mechanisms to end organ damage, whether it's in the cardiomyocyte, the fibroblast, you have inflammation, you have an effect of, you have a cardiorenal syndrome. So it's a broad syndrome that is affecting a lot of pathways, or that involves a lot of pathways, again, all independent from the level of ejection fraction, but with different phenotyping expression. And the precapillary component found in heart failure is one of these phenotypic expression of the complication of the underlying disorder. We know that there is vascular remodeling, and we know that not only from this study, from Faiz and colleagues, but from other evidence also coming from animal models, that if you look at the changes in vasculature in patients who are affected by heart failure, pulmonary hypertension, heart failure with reduced or preserved ejection fraction, this is what you're going to find. This is a study that was, I think it's probably one of the very few histological study looking at 108 patients with preserved or reduced ejection fraction associated pulmonary hypertension, 12 control, and 17 PVOD. And this is also very important in the reasoning. The lung specimens were retrieved. There was a hemodynamic correlation that was sought. And you can see here from A to C, a central artery, a control vein, and a control intravascular or intraparenchymal vessel. This is what you can see in normal patients. And this is what you see in patients with heart failure with preserved ejection fraction from G to I, and from J to L, what you see in a patient with PVOD. In fact, there's a remodeling that is affecting all the pulmonary vasculature, from the arterioles to the capillaries and the venulae. And this is probably where the struggle is in this very peculiar indication for potential treatment. These abnormalities of remodeling are more strongly correlated with pulmonary hypertension in a small intimal thickening, similar to the pattern that is expressed in PVOD. So there are commonality in terms of phenomenon or mechanism leading to pulmonary hypertension in group 2 and PVOD. We also have some lessons from genetics, and in an animal model of left ventricular pressure overload, we have an expression, we have dysfunction of KCNK3, which is also involved in the development of pulmonary arterial hypertension. So you have remodeling, you have also a genetic dysfunction. And to come back to the point of whether the TGF-beta superfamily is also affected in this disease. I'm bringing back a lesson from an animal model, and there were several models that were tested in those experiments, looking at the effect of manipulating the activin pathway on pulmonary remodeling. That was favorable in animal model of obese rats who developed HFpEF and pulmonary hypertension associated with HFpEF. And the irony is that there was an interaction that was fine with the SGLT pathway. So it's a very complex phenomenon, more complex than we initially thought, but this would have served as a good reasoning and a good reason to test sotatercept in group 2, in the group 2 population. So here we are with Cadence. Phase 2 double-blind randomized placebo-controlled study to evaluate the effect of sotatercept versus placebo in patients with CPC-PH associated with heart failure with preserved ejection fraction. It's kind of a large trial, 91 sites, 12 countries. The recruitment goal based on the primary endpoint that was PVR was 120 to 180 patients, and there was a planned interim analysis to adjust for the recruitment number. The key eligibility criteria are not really surprising. Adult patients with left ventricular ejection fraction above or equal to 50%. At no point of time, patients were allowed to have less than 45% on 2 stable readings at any time. There was a hemodynamic confirmation of combined post and pre-capillary pulmonary hypertension with a PVR that was set at 4 Wood units. Patients were symptomatic class 2-3. They had to walk more than 100 meters to be stable on heart failure medication, and their comorbidities being managed appropriately. And of course, the patient needed to adhere to the study procedure. It's pretty standard, and as usual in group 2 trials, you have some inclusion criteria and you have a lot of key exclusion criteria. That includes, of course, other forms of pulmonary hypertension, lung disease, valvular disease were excluded, severe valvular disease or arrhythmias, uncontrolled hypertension, etc., etc. The hemoglobin level was supposed to be above, it was not allowed if a patient who had hemoglobin level above the upper limit of normal or less than 10 were not allowed, and as expected, also low platelet counts were considered to be a contraindication or exclusion criteria given the effect of the compound. The study endpoints. The primary endpoint is a change in PVR at week 24. There was a key secondary endpoint. There was a change in 6-minute walking distance and a couple of secondaries, including clinical worsening and T-proBNP. There was also an echocardiographic sub-study that was looked at through a core lab that was, of course, interesting in the context of heart failure. Of course, safety endpoints were included, and the rate of treatment emergent adverse events were also considered. This is the study design. Screening period, 28 days to revise the inclusion criteria, right heart cath at baseline, and then the randomization 1-1-1, sotatercept 0.3 milligrams per kilo, sotatercept 0.7, and then placebo. The placebo arm was the third arm. There was a primary endpoint collected at week 24 for PVR and secondary endpoint for walking distance. But then there was a randomization of patients on placebo because the idea was to look at the longer term effect potentially with a third right heart cath at 48 weeks, where patients ex placebo were randomized to 0.3 and 0.7, and the patient on sotatercept from baseline were kept on the same dose, and of course there was a safety analysis. So here are the study results. 164 patients were randomized. And you really thought I would give you the results. In fact, as Stefan mentioned, there's unfortunately not much we can say. The presentation is going to be at ACC as a late-breaking clinical trial, will be presented by Mari Gomberg on behalf of the steering committee. There was a press release, though, that you probably saw saying that the study met its primary endpoint, which is, I guess, extremely important because this is a population of patients we identified at last World Symposium as a population of interest. It's population one, PH associated with HFpEF with elevated PVR. We can argue whether it should be 5 or whether it should be 4. You need randomized controlled trials if you believe those patients should be treated, of course, and that's the only way we can get away with evidence supporting the benefit of any therapies in this context. There are other trials, and I just want to point out that the paradigm in PH associated with HFpEF is slowly changing. Maybe sotatercept is the last exception to what may be the next rule. The next rule is to look at drugs that may have a positive effect not only on the pulmonary vasculature but also on the left ventricle. And those 3 trials with levosimendan and 2 relaxin analogs in the studies, REFIRE and APEX, are looking really at that population with a broad range of pulmonary vascular resistance. It's not completely stupid to believe that maybe those drugs have an effect at a lower PVR than the one that was included in Cadence, and this is probably something that is a true novelty in our field. Now, some take-home messages. We don't have the full results of Cadence, but there's a couple of things we may say. Cadence was the largest trial performed in a group of patients with a very clearly established precapillary component with a PVR above 4 Wood units. It's a landmark trial in patients with significant PVD. There's no doubt about that. There will be a world before and a world after. You will see the results very soon. This is changing a little bit the paradigm and participating in moving the field forward to a phase 3 trial potentially, but more thoughts are going to be given, of course, to the feasibility and perhaps to other components of the disease and perhaps also to other strategies in group 2 pulmonary hypertension. Thank you very much.
Thank you very much, please, yes. So now the stage is open for questions from the audience, from the moderators. Any questions? Okay.
Run, Forrest, run. Run, Forrest, run.
That's at least functional class 2.
Fantastic session from the 3 speakers. Congratulations. I actually had a question for all 3, but Ardi, I found the answer for my question in table S11 in the supplementary index. So I'll avoid. I've got one for Jean-Luc and one for Ioana, if I'm allowed. Jean-Luc, you took us through the rationale very nicely with pulmonary vascular effects, but since you didn't reveal any results, I'll tell you about my one patient in Cadence who went from post-capillary to pre-capillary. Mm-hmm. Could this be acting directly on the left ventricle?
That's a very good point. I'm afraid I cannot answer the question for now, but we have insights, of course, on the effect on the left ventricle of the drug based on hemodynamics, but also based on echocardiography. So it could be.
Sorry, I didn't want to put you in an uncomfortable situation there.
I'm never uncomfortable.
Ioana, so I've heard you give this talk many times, and well done for still keeping it interesting. But the one thing that I picked out that I've not looked at before is the thrombotic events at 4%. Is that just very similar to sort of background rates? Are these thrombotic events, are they venous? Are they arterial? Is it anything to do with the hemoglobin rise? Or is it just, if you placebo correct it, which obviously you can't in Soteria, then it just doesn't come out as a signal?
Yeah, it's a very good question. So some of the thrombotic events were venous, most of them. Thromboembolic disease, pulmonary embolism in a couple of other cases. There was not a signal or a clear correlation with an increase in hemoglobin. Definitely not. I don't know if it's a underlying predisposition of these patients who are actually also bleeders. So if you look at the the risk of bleeding in the placebo arm in Zenith, for example, or in Stellar, there is a high risk of bleeding in these patients who are treated with therapies that also affect the coagulation cascade. On the other hand, I think there may be a slightly high risk of thrombotic events, but it's very difficult to judge without a placebo arm long term, unfortunately.
Okay, thank you. Once again, congratulations to the speakers for a great session. Thank you.
Dr. Abdullah?
So Joe, look, I was hoping that you break rules and you tell us about the results, but you didn't do that. So thank you so much for at least announcing that the result was actually positive. It met the primary endpoints. This takes me to the question to Ardi. Recently, you presented the data on sotatercept, PVRI, effect on myocardium, LV mass, RV mass. And given the good number of attendees, I would like you to elaborate more on that early data.
Thank you, Abdullah. And I have to disclose, we have not discussed this question before, but I'm very grateful that you give me the opportunity to elaborate a little bit on that. It is actually no speculation, but it is proven that sotatercept has direct effects on the myocardium, left and right. So this has been actually indicated from very, very early preclinical studies throughout what we saw during the clinical development for group 1 PAH, where there was this interesting phenomenon observed for the first time in the field of pulmonary hypertension, that patients who had a massive reduction in the afterload of the right ventricle by means of PVR reduction had no change in cardiac output at all. This actually corroborated nicely what we suspected from drugs like sotatercept to have an impact on the myocardium directly, and now there is work from Rodatello's group that we've just presented in part at the PVRI, as you mentioned, in Dublin, that clearly shows that there is a load-independent impact of sotatercept on the myocardium, left and right side. It goes as far as you see reductions in right ventricular and left ventricular mass of patients who've been treated for PAH. So I would be surprised if anything else is seen in cadence than a direct impact also on the left ventricle. Of course, we assume there will also be an impact on the pulmonary vasculature, but it is without doubt that this drug also affects the myocardium.
I think this is a very interesting point, but you could see that in a positive or a negative fashion. I mean, if we have effects on the myocardium that then lead to a poorer cardiac function, you may think that's a bad thing, but in the context of HFpEF, if you have reverse remodeling on the left ventricle and also a hyperdynamic state in HFpEF is considered a bad thing. So I think when we analyze these data and interpret these data, we also have to look within the normal range of EF, RVEF, LVEF, from where to where is a change. Do we go from high normal or from a hyperdynamic state to a normal state, or does it then go beyond the lower limit of normal? That might be important to look at it, not just simply look at the delta. So important findings.
And if I may add, I think it will also be interesting to study these patients with differential response in the myocardium, maybe as you describe, and their response to exercise. Are they able to increase their flow, their cardiac output, stroke volume, accordingly to exercise, because there may be a good balance in the end.
Thank you.
Okay, Olivier.
Yeah, thank you, all the speakers for the nice presentation. So I just have a question for the panel, perhaps Ardi and Joanna first. But in trials, sotatercept showed very, very nice improvements, but without any distinction between true responders and non-responders. Because in real-world data, we clearly have patients who respond very, very well to the drug, and some others, they do not respond at all. Nothing happened. So first, do you have any hypothesis for that and how to distinguish the responders to non-responders? And number two, what is your attitude if you have a patient who do not have any response and when to assess the response? I have an idea, but I would like to have your insight.
Yep. Shall I start first?
Sure.
Yeah. So thanks for this very relevant question. And I believe that roughly speaking, and I mentioned that yesterday, the rule of the third applies. In our own patient cohort, we see that there are about a third of patients who fulfill criteria of true response. And by true response, I really mean the hemodynamic response that we wish to see in these patients, with a meaningful reduction in the pulmonary vascular resistance and mean PA pressure, with improvements in all functional parameters, and completely independent from the changes in NT-proBNP. That's a separate discussion because NT-proBNP is no reliable biomarker in sotatercept patients and not correlated to PVR anymore. So having said that, 1/3 in our observation are very good responders. There is another third in the middle that has a good response or, not so much of a response. One wouldn't really say neutral, but it is not overwhelming. And then there is a clear third that has no response at all, in whom one should consider discontinuing the drug. This is definitely a drug that has a very robust side effect profile. The longer we treat the patients, the more side effects we see. This drug is an HHT mimicry drug. And with HHT, we know it's not only telangiectasia, it starts with it. But in patients who are born with inherited disease, it takes 50 to 60 years for the full display of the syndrome HHT. So why would we expect to see differently with a pharmacologically induced HHT? And it has all features are emerging. So it's a matter of whether or not the risk to benefit is still balanced in patients who do not have a meaningful improvement. However, all of the patients will at certain periods of time develop relevant side effects, starting with hemoglobin increase, HHT mimicry, more or less occurrence of pericardial effusion, bleedings, and so forth. So we need to keep the balance, and that's why I'm very thankful for your question, because this is no drug that we apply and forget. This is a drug where we have to assess the efficacy and we have to watch the safety, and if it doesn't work after half a year, we should stop it.
So if I may add, thank you, Ardi, for your experience. Like any therapies in PAH, the rule of a third applies. Not everyone responds to one drug. It is the bell-shaped curve that applies, in the real world. Now, what to do with these patients in whom hemodynamics do not show improvement? What do we do in general? We assess the efficacy and safety profile. We see how these patients feel, if they feel better or not, and if they had any side effects or not. I think, it's a little bit too much to say that all patients will develop side effects at some point, because so far the data are not there suggesting that all patients at some point will develop a side effect. So I think the drug overall is tolerated. However, I agree with you that you have to continue to be vigilant, because even though the risk is not of side effects, it doesn't seem to be cumulative, it's still there. So you have to be following these patients closely. If there's no response at 6 months hemodynamics, I think it's reasonable to stop if the patient doesn't feel better and none of the parameters that we think are clinically important.
Yeah, but it is the first time we ask the question of drug withdrawal, because the other drugs, when we don't have any response, we escalate. we do not stop the drugs.
Absolutely. But this is a new therapy. This is a biologic. This has a different safety profile than all the other therapies that we have available. So maybe we need to apply a different approach. I agree.
Thank you.
Merci.
Thank you to all speakers for the wonderful talks. In this part of the world, we are really, really interested in sickle cell disease. And we just had 2 patients with the sickle cell disease treated with sotatercept. And now we are reporting these 2 cases. And the reason we chose sotatercept in those patients because they failed all other medication. They were very, very symptomatic. But for the known data that patient with chronic hemolytic anemia, they have high really blood level of activin B mainly, but also A. So we thought that this is really interesting. Also, they might have some PVOD component where treating PH without really overflowing the left ventricle might be reasonable. So really my question to you as really expert, do you have any really experience with treating patients with chronic hemolytic anemia, where it's really well known that their activin level in the blood is quite high with sotatercept? Because that would really enrich the look of the usage of this drug in this particular patient group.
Yeah, I have to say I don't have any experience.
I have no idea, but I could think of it could be worthwhile to talk to those folks that use luspatercept in malignant anemia, because I'm sure that there is some off-label use also in other hemolytic anemias, at least to give it a try, and then might be there is something to learn from. I think Paul Yu's still in the audience, is he? I've seen him earlier.
Yeah,
Paul, do you know anything about activin signaling and sickle cell anemia?
No? Yeah. Yeah, I mean, our really main concern was what's going to happen to hemoglobin S, because if that goes up, then those people will have more acute really crisis. That did not happen with those patients, and really, at least the clinical data and hemodynamic significantly improved. So just like to really have light on this. Thank you.
Interesting.
Yeah, thank you very much for the great session. I just wanted to comment on the direct impact of sotatercept on the right ventricle. I think we sometimes overlook what is really driving the symptoms of our patients. So not only is the afterload increase, but also the diastolic stiffness of the LV and also of the RV. So on the RV, we are not really able to measure it because E2E prime and E2A are not validated for this, but there is a direct link between stiffness and symptoms. So if you had a drug like sotatercept, which direct impact on the RV, improving this wall tension, diastolic stiffness, then this would explain all the things we see in our patients. The drop in anti-proBNP, the symptoms which really become better after some weeks even. So this might not be deteriorate at that time, but what happens in 2 or 3 years, or in 5 years, or on the long run. So this is something we have to look at, and to look at our patients with sophisticated measurements and for the direct impact on the RV. So I think that this might really explain why cadence is positive, or perhaps positive, and why the symptoms are becoming so good at the very short time. So I think the compliance will be improved. This is what we see in our data.
That's an important comment,
So,
Khadra. So
if I can, a quick point to what you were saying, you're absolutely right. So first of all, cadence is positive. It's not perhaps positive, it is positive. And the second point is that, especially in group 2, you should not underestimate the role of atrial function in the symptoms. Most of these patients with group 2 PH are in atrial fibrillation, and this is what we already see in the heart failure world. There may also be an impact on the regurgitant volume of the tricuspid valve. So you're totally right. It's very complex to analyze, but there's another layer of complexity that is associated with the right atrium and the left atrium, actually.
Related to what you have said and others have said, I think one important issue here also to learn is what is the optimal dose for the long-term treatment. Because if we can identify direct myocardial effects or effects on the atria, then this would be a desired effect. And then, as you mentioned, Rodre, in the long run, whether we could have also a negative impact on contractility is probably most likely dose dependent. So maybe we have to also play with the dose in the long run, and that could help us. Dose is one thing. You have a question?
Yes. So I was so interested in the serious rare side effects of indications of hypoxia and pericardial effusion. So in the hypoxia, a patient with other congenital heart disease, were there any hemodynamic analysis to understand what happened exactly in these patients with the complication, not before, after the complication happened?
No, there was no hemodynamic assessment at the time of hypoxia. But there was a thorough evaluation of the reason for hypoxia with echocardiograms, with bubble study, with 100% shunt estimation. So it all pointed out towards an intrapulmonary shunt physiology. The hemodynamics were not assessed. However, they were assessed initially and at 24 weeks. And these two patients had a good response with a drop in PVR. So again, I think maybe Stefan was alluding to maybe the dose for each patient should be tailored. How to tailor it, we don't have an answer yet, but I think it should be studied. Maybe not everyone needs 0.7 milligrams per kg.
Yeah. So dose is one thing.
Yeah.
Time is another one, and
Yeah.
also the timing until the effect occurs. And in this context, maybe another question for you, Artie. You showed the Kaplan-Meier curves that separate quite early. You told us that the effect is visible after only 3 doses, which is surprisingly fast. So could you maybe elaborate on the potential reasons why we see this effect so early, and relate that also, as we talk about side effects, to the timing or the time until we see potential side effects?
Yes, so apart from what Roda just mentioned, the potential impact on myocardial stiffness, which may in fact be mechanistically explained as a very rapid effect, we know that there is a surprising vasodilatory effect that sotatercept has, and I'm going to show a few slides on that, that shows a very, very early reduction in PA pressure, much earlier than you would anticipate from a presumably anti-proliferative drug. And there is obviously this feel-good component that is related to hemoglobin increase. It is definitely a feel-good drug, and that sometimes masks the absence of relevant hemodynamic improvements in these patients. That is also, but maybe not only, related to hemoglobin increase. We're going to ask something because I wasn't ready with my answer, but please interrupt me as usual.
Maybe just related to that. You mentioned the vasodilatory effect, and I guess that statement is based on rapid effects that are seen on hemodynamics. But do we really know, is it a functional effect, is it vasodilatory, or can we also expect, if we think about the main mechanisms of an antimitogenic effect on the pulmonary vessels and an apoptotic effect? These occur within hours, and one could argue that if in the small vessels you have quite predominant remodeling, and then your orifice is very, very small, and even a small effect, if you then think of the area, will give you a large effect. So could that also explain it, an early remodeling effect versus vasodilation?
So this elaborate question just shows how much Stefan and I are trained from the times when imatinib was first suggested as a treatment for PAH. And we had these same arguments because also with imatinib we saw some very early clinical improvements that were not necessarily explained by a true anti-remodeling as you anticipated, but in fact depriving only a few cell layers in the center of resistance vessels lowers pulmonary vascular resistance by the order of 4.
Yeah.
That we know definitely. There is a possible alternative explanation or another contributor, which could be a sensitization towards the mechanisms of the background therapies, which are all vasodilators. And there's good reason to believe that influencing the activin pathway has an impact also on the complementary signaling pathways. It can augment the complementary signaling pathways, but it can also sensitize again towards the vasodilator. So there are a lot of, it's a mixed bag. We still need to learn a lot about this, but it's remaining interesting.
Yeah. I see Irene, please.
Thank you. I have a very simple question compared to what you were just
You
talking
never have
about.
simple questions, Irene.
Well, think about how difficult it is now in the sotatercept times to risk stratify when a patient comes back. So probin B is down, NYH class is down because probably oxygen consumption is improved in the brain. And the third of the French risk criteria is also improved. Please tell me how to risk stratify. I think for a simple physician without MRI, without, I don't know, modern diagnostics at hand, maybe liquid biopsies or anything we will have in the future in metabolomics, it may become really difficult even to assess non-response.
So definitely it highlights the importance of invasive hemodynamics again. And I have to say I am so pleased that we overcome the blindfolds we all were carrying, that hemodynamics don't play a role. We should only look on clinical parameters. This disease is defined by hemodynamic changes in the pulmonary circulation and the interaction with the right ventricle. And if we assess efficacy, we should look what the presumed efficacious drug does to the pulmonary circulation. Having said that, I totally agree with you that we need to redefine the risk scores because NT-proBNP doesn't work anymore as a surrogate for PVR. It doesn't with sotatercept. It goes all ways. It is massively reduced also in patients that have no afterload reduction. TAPSE-PASP, the very useful ratio, for the first time has changed with drastic reductions in PASP with no changes in TAPSE, which was unseen before with the other treatments, but that is also included in some risk scores. So we need to maybe define sotatercept-treated patient-specific risk scores that may use other parameters than the currently used.
Adi, stroke volume index doesn't work anymore. PVR may not work anymore. What does 5 mean? Is 5 meaningful for a SOTA patient? So basically all your hemodynamic bars may be elsewhere. Please help.
But I mean, clearly we have seen a different also hemodynamic profile of the changes, as you just said. And clearly we have shortcomings with the risk stratification tools that we're currently applying. And we have always considered that work in progress. So how can we handle that? In my view, we have a combination, or should have in the future, a combination of disease-specific assessment in PAH. There, this concept of disease remission may come into play, where we measure things that directly have to do with the disease PAH. At the same time, should probably capture things beyond that, including comorbidities, the influence of comorbidities and other things, or how the right ventricle responds. And that then together should give us a good picture of the patient at baseline, but also the changes in disease-specific parameters, but also beyond that help us in the overall risk assessment and prognostication of these patients, because we have also learned that not only the disease is driving prognosis, but also other factors that may be present in individual patients. So we've used up even the extra 10 minutes that we were given, but we had great discussions, and I'll give...
So unfortunately, we have to end this great session, actually, and the discussion took more than almost 25 minutes, so that's a topic by itself. So the SAPH session is ended, and we're moving on to satellite symposium or session by, again, Dr. Gufrani. And it's about regarding the data-driven rear-cingulate effect on the right ventricle in CTF patients with pulmonary hypertension.
Yeah.
Oh, I'm sorry.
Thank you.
I didn't know everyone else had to clear the podium. Thank you very much for the kind introduction. I think there's some uncertainty on who's going to stay on the stage or not. And again, I'd like to renew my thanks for the organizers of this beautiful meeting. I just spotted my friend Salah Al-Damas from the very early days in the audience. Long time no see. It's a great pleasure to see you again. So, as if I had anticipated that this issue about RV, what medications do directly or indirectly on the right ventricle, may be also interesting beyond what I'm going to tell you about rear-cingulate. I have put some introductory slides that may add to the information needed to better get into this topic on how to focus on RV-specific effects of drugs versus afterload-dependent effects of drugs in the field of pulmonary arterial hypertension therapies. So we have all focused in the preceding talks and the preceding sessions to the upper part of this figure symbolizing the signaling pathways that are of interest currently with some of the newly established therapies, some of the emerging therapies like the tyrosine kinase inhibitors, and so seralutinib is listed here as one of those under development, and the phase 3 is about to come to an end, but there are also other interesting molecules listed there. But on the bottom right, there is a little heart, and I think we need to focus more on the heart to understand the interaction with the pulmonary circulation better, and also to question whether or not our drugs are neutral to the myocardium, whether or not they're beneficial for the myocardium, or whether or not they may in fact have some detrimental direct effects on the myocardium. To better understand the right ventricle, we should remind us about the complex structure of this right ventricle, which is much more complex in addition to its conduit, the main pulmonary arterial branch, the complex interactions with the tricuspid valve when it becomes leaky, as well as the outflow tract. So it should be really considered as one connected system. And of course, we nowadays have very elegant ways to visualize, but also to assess right ventricular functions and dimensions beyond what we used to do before. Also being reminiscent of the fact that the contraction of the right ventricle goes in all three axes. It's a very complex contraction mode that allows a healthy right ventricle to produce output, which is in clear distinction to what we know from the concentric contraction on the left side. So I like to use this schematic to highlight that for a very long time our main focus was to develop drugs that are mainly targeting the pulmonary circulation. Whether they are vasodilators, whether they are antiproliferative, pro-apoptotic substances, they were all meant to target the pulmonary circulation, where in the precapillary resistance vessels, basically the disease initiates and then also progresses and evolves.
The targeted treatments that showed some efficacy, in fact, in improving the pulmonary circulation, lowering the afterload for the right ventricle, and thereby secondarily also improving right ventricular dimensions and right ventricular function. However, we should be aware that many of these drugs have also direct impact on the ventricles. For a long time we thought, and now we've proven that, for instance, prostacyclin derivatives have a direct inotropic effect on the myocardium. And I'm going to show you what we learned about emerging new substances like sotatercept, but mainly also what riociguat does or does not on the right ventricle in addition to its afterload reduction. So we also know that clinically we see patients that have the same degree of pulmonary vascular resistance. However, their ability to adapt to this increased afterload is considerably different. And you can read that from the shape and the size of the right ventricle. Some patients have a very well adapted right ventricle with a good RVPA coupling. They show that they have a homeometric adaptation. They are in a compensated, compensatory, hypertrophic state of their right ventricular myocardium, as opposed to other patients producing the same cardiac output, but with a failing right ventricle that is increased in its dimensions and goes into a maladaptive dilatation. But how to assess cardiopulmonary hemodynamics? And textbooks actually teaches us that we screen our patients with non-invasive imaging technologies such as echocardiogram. They're very suggestive. They are able to increase the likelihood of really pulmonary hypertension being a problem, and it is also a good tool to increase the so-called pretest probability before a right heart cath. But ultimately, the gold standard remains right heart catheterization, also because by using the balloon, it allows us to at least roughly distinguish between precapillary and postcapillary pulmonary hypertension. In addition to the pressures, we can also assess cardiac output by means of thermodilution or by measuring saturation in the mixed venous samples and applying the equation of Fick. So we can assess classical hemodynamics. What it doesn't tell you is how the cardiac function is in relation to the strength of the myocardium. To be able to do that, we still need to borrow experience from our left heart colleagues who've been using conductance catheterization for the measurement of pressure volume loops, from which you can then derive contractility independent of the afterload. And it's been for a long time that we missed those data for the right ventricle until very few case reports with some non-conclusive results were published for a small series of patients from the right ventricle, and then Rodatello from Gießen and his group started establishing this gold standard for the measurement of contractility also for the right ventricle, which wasn't an easy job, I have to say. They needed to adjust and optimize the measurements, but ultimately were able to derive pressure volume curves as you would like to have them. The high fidelity pressure recordings are paralleled by measurements of the conductance, which is dependent on the dimensions of the circumferent blood in the ventricle, and the more the blood is evacuated, the signals in the conductance parts of the catheter change. And what you get at the end of the day are these assessments of volume and pressure, and they are put into relation, where you can then assess from the steepness of the change in PISO. You can assess contractility by means of EES. You can assess the afterload by means of EA. You can calculate the stroke work. You can calculate stroke volume or cardiac output from stroke volume and heartbeat, and also you get an idea about the preload. And when you do that in a multi-beat assessment, you have
Basically, by changing the preload of the right ventricle, you get a direct measurement of contractility. This is very useful. This had to be done at a certain stage. It is the gold standard, and it allowed us for the first time to measure and therefore also to assess the value with regards to risk stratification of our patients, with regards to the assessment of the individual courses that these patients may undergo, but also allowed us to assess the direct or indirect effects of some of the medications that we use in treating pulmonary hypertension patients for the first time with this very elaborate technology. As everyone can imagine, the technology, however, is not generally applicable, and it would be desirable to have surrogates or at least less invasive methods to come up with the same measurements, and therefore there are some complementary imaging technologies you can use for the assessment of the volume changes. So far, cardiac MRI has been the gold standard for the assessment of volumes, but more and more and increasingly 3D echocardiogram is able to provide us with the same important information. And again, from Choda's group, Zvonimir Rako, first author on a paper where they could combine the data of pressure measurement pressure measurement in the ventricles and combine it with volume measurement derived from 3D echocardiogram, and in fact even were able to assess non-invasive or less invasive pressure volume curves by these combinations, and even segment the patients into low, intermediate, and high-risk patients based on the pressure volume loops, and these nicely corroborated with the clinical scores that were also found. And then again, cardiac MRI provides you with much more information apart from the dimensions. It can also allow you to assess functionality of the right ventricle. You can even assess strain with the MRI, but you can also assess the wall thickness and also the mass of the right and the left ventricle respectively. And here's just an example of the discussion we previously had. We were not surprised to see that the right ventricle had major changes in its volume during the CMRI investigations. We were not surprised to see that there was some hypertrophic effect that was seen that could be also attributed to the changes in afterload. So an unloaded right ventricle loses muscle mass that is been known for other drugs as well. But we were quite surprised to see that also the left ventricular mass was reduced in these patients, indicating that this might in fact be a direct myocardial effect as well. So going one step further, the aim of the group of Choda was to further reduce the invasiveness for the assessment of contractility, and thereby they worked still with an invasive device, which is a core-lock device, actually allowing you to introduce high-fidelity pressure catheter into the right ventricle, which is connected to a transducer outside the body of the patient, and then the pressure curves from the right ventricle at high-fidelity recording are transferred to your mobile device. This is considerably different to the CardioMEMS, which only allows you intermittent recordings of the pressure while the patients are sitting on their transducer chair. So here you can collect high-fidelity signals as they are produced in the heart of the patient, at rest, during exercise, during normal activities, while sleeping. A 24/7 recording online is allowed. And from those pressure curves, Niels Kremer from Choda's group was able to derive calculations of the contractility, and these have been validated, in fact, by the gold standard. So this is the way one should do it, not only hypothesize, but really validate what you're measuring with your new technology, with the previous gold standard, and then you can de-escalate the invasiveness. And this is possible now with this device. And we use the device in a series of patients because we were so much interested to see whether or not beyond vasodilatation, beyond reductions in PA pressure, beyond reductions in afterload,
there might be some fundamental differences between classes of drugs. So in this series of patients, just recently published by Niels Kremer, again in Circulation, there were subsequent patients who received sotatercept while carrying the Core-Lock device, and there was a comparator group in patients in whom treprostinil intravenously was administered as a extension of the treatment. And these two groups were observed in parallel, and what you can appreciate in the figure showing end systolic pressures is that with sotatercept, as well as with treprostinil, end systolic pressures tended to decrease over time. However, these are not days, these are hours that you see on the x-axis. So the reduction in pressures with sotatercept happened within 48 to 72 hours, which was much quicker than anticipated. What was also observed was that the P-ISO reflective of the EES was starting to decline with sotatercept and with treprostinil respectively. However, the EES to EA ratio that was reduced over this period of observation in sotatercept, meaning that the RV-PA coupling, while maintained, was not improved, but that was in fact improved with treprostinil as a consequence of a direct positive inotropic effect of treprostinil. So it's a direct comparison in patients using one of these elegant devices. Last but not least, you will still ask yourself, how can I apply this to my clinical routine? And the guys didn't stop working hard and developed a very handy, useful software which every one of you can use. This is the time everyone should pick their mobile phones and then take a picture of this web address there, because there you can upload the original recordings from your PA catheterization from the cath lab, and the software will calculate you pressure volume curves and provide you with measurements of contractility. It's becoming so easy, but it's been a journey of 10 years of a whole working group around Rodatello to ultimately be able to provide clinical colleagues with the possibility to assess these functions directly in all of their patients. It may be also important for future investigations, including what we just heard about cadence, so also may be applicable and important for patients on the ICU and patients that have other cardiopulmonary morbidities. Which brings me, actually, Hazem, I'm sorry that it took so long to get to the actual topic of the meeting, to riociguat. And riociguat is a very important molecule. As you know, we've been working together with Bayer for many years in the preclinical setting to identify soluble guanylate cyclase stimulators and activators as potential therapies for cardiovascular diseases. And riociguat, after a long development, was identified as a potential candidate for the treatment of pulmonary arterial hypertension. And it was the first drug that was investigated in two parallel initiated phase 3 randomized controlled studies in PAH, where it would be a new class of drugs amongst other treatments that were already available, but also as a potential first drug ever available for the treatment of inoperable chronic thromboembolic pulmonary hypertension. At that time, the results were very impressive and very conclusive. Patients with or without background therapy of prostacyclins in the pulmonary arterial hypertension trial improved their primary endpoint, which was the change in 6-minute walking distance, in a clinically meaningful matter. They also showed for the first time in the field of PAH in sequential combination therapy patients that in fact the placebo group, once changed to the active drug, could catch up. So it actually contradicted the notion that was present at that time that they would never catch up. It contradicted it for the first time with riociguat. We saw the same pattern by the way, with sotatercept just recently. So with riociguat, they can catch up even if they are treated with a delay. And then we saw comparable results for the first time in inoperable CTEPH, where it became the first approved medical therapy for the treatment of these patients. I'd like to make a little detour through the hemodynamic data that we collected in those studies. And as an example, but the chest data look very much alike, I showed you the hemodynamic assessments in the patent trial. Remember, these were two trials in which it was mandatory to have right heart catheterization at baseline and follow-up. It was at times when the community would try and make us believe that hemodynamics don't play a role. And nowadays we know they play a role, and the role is becoming even more important. In this study, it was mandatory. It hampered the recruitment of patients, but it, after all, showed us very important information in that, first of all, this drug is highly efficacious. It is a very powerful drug, which dose-dependently reduces pulmonary vascular resistance and afterload. PVR was reduced by more than 30%. The absolute numbers are given here. It very prominently and proactively reduced NT-proBNP, more than the other drugs that have been tested at that time in that scenario. Mean PA pressure was reduced significantly. So it's not that it is only a driver of cardiac output, but also cardiac output was increased. Starting from a baseline cardiac output of 4 liters, it normalized to 5 liters. So this is clearly something you could attribute also and mainly to afterload reduction. And the right ventricle in these patients with this drug behaved like a compromised right ventricle should behave. If you unload a compromised and loaded right ventricle, it should return to its normal state. It should normalize and function, opposite to what we see with sotatercept nowadays in some of these patients. So here, the behavior was as anticipated. And then there was a post hoc analysis of more broader parameters regarding the effect of riociguat on right ventricular function, summarizing data or collecting data from both trials, from CHEST and PATENT, and then looking at them extensively and also correlating them to clinical risk assessment tools, mainly the REVEAL risk score at that time. The study was led by Ray Benza, and you can retrieve the papers online. There were certain additional parameters analyzed, such as stroke volume and stroke volume index. The cardiac efficiency was assessed by means of stroke volume ratio to mean PA pressure. The pulmonary artery elastance was calculated. RV work index, and RV power were calculated. And there was an interesting idea about paired marker analysis of combining signals from stroke volume index and right atrial pressure. And generally speaking, and I won't take you through everything that is listed here, generally speaking, all of those parameters in the majority of patients improved, which drove the positive signals that were observed. So patients were then categorized whether or not they had a favorable response, which was defined by a combination of a stroke volume index equal or greater than 31, combined with a right atrial pressure of lower than 10 millimeters of mercury. Then there were intermediate patients where one or the other was beneficially influenced with no beneficial change in the respective counterpart. And there were patients categorized by stroke volume index of less than 31, combined with a right atrial pressure of 10 millimeters of mercury and more. And then they were set into relation with the REVEAL risk score. What could be clearly shown was that stroke volume index was negatively correlated with the REVEAL risk score, and right atrial pressure was positively correlated with the risk score, meaning the higher the right atrial pressure went, the worse the REVEAL risk score got. Patients with a favorable SVI-RAP ratio had significantly lower risk scores, and this trend held regardless of other parameters. So it was an independent indicator of outcome. And this is also what you can derive when you apply those categories to the survival of the patients. You can actually mimic the clinical risk assessments by this hemodynamic ratio very nicely with a good resolution and good granularity between low risk, intermediate, and high risk patients. There was long-term survival insights from this study as well, from patent and chest, where several RV functional parameters at baseline and follow-up were associated with survival and clinical worsening-free survival in both studies, and stroke volume index at baseline and follow-up was also significantly associated with clinical worsening-free survival. And this is my last slide to this topic, just showing you that we recently also investigated on patients with classical oral upfront dual combination therapy, in this case a combination of an endothelin receptor antagonist with riociguat using the conductance catheterization, showing you that at baseline the EES to EA ratio in the middle part was 0.92 with an EES of 0.92, and then upon follow-up catheterization after initiation of this dual combination therapy, cardiac output was maintained while pulmonary vascular resistance was dramatically reduced. The EES, which is the contractility, was also reduced, so you might think this is a drug with negative cardiac output. However, the EES to EA ratio that was increased shows you that EA, the afterload, was more significantly reduced than EES, so showing you that part of the contractility of the right ventricle is in fact dependent on the afterload. However, there's an independent part of RV contractility that is not related to the afterload and shows you that the drugs at least did not compromise contractility in this combination. So allow me to summarize that the prognosis of PAH patients is closely correlated to RV function. There is emerging evidence that we should focus more on RV dimensions, on RV function, earlier assess RV-PA coupling, and there are now means to also indirectly calculate contractility with the nice software that I showed you from normal catheter data that you generate in your cath labs. The invasive assessment of cardiopulmonary hemodynamics is essential both for the confirmation of the diagnosis but also for follow-up risk assessment, and the hemodynamic assessment becomes more and more important the more complex our treatments become and the more combinations of therapy we are using that sometimes do not allow us anymore to assess efficacy by means of clinical parameters or subjective well-being of our patients, but demands to assess the invasive hemodynamics as a guidance for future treatment decisions. Echo and right heart cath have so far provided load-dependent insights into RV function and dimensions, and the gold standard, however, for the assessment of direct contractility, load-independent contractility of the ventricle remains conductance catheterization. However, and this is owing to the great work from Rodatello and his group from Gießen, there are now validated surrogates for conductance catheterization, and you can even download them from the web. Riociguat improves RV function, that's without a doubt, by a meaningful afterload reduction. Stroke volume index and right atrial pressure correlate with a REVEAL risk score, showing you that at least for this combination of drugs or these drugs outside the sotatercept world, the correlation of risk scores and hemodynamics still apply. And riociguat maintains cardiac contractility, as I showed you in the last case example, proportional to the PVR reduction, and this is something that we should expect from our drugs. I don't want to close before acknowledging the great work of some people from my own institution, Roda and Niels, mainly responsible for all the work that has been done on the clinical side that provided us with a much more in-depth and
more granular understanding of the right ventricle, of right ventricular to left ventricular interactions, and also about the impact of our pH drugs on the myocardium. And obviously, Ralf Schermoli, Norbert Weissmann, from the preclinical side of the translational development group. You've already got to know Soni Pulamsetti, who is our superstar in the preclinical labs, and also acknowledgements, shout out to Fritz Grimminger and Werner Seeger, who are the heads of our research group, and Werner for a long time was the head also of the university hospital. At the end of the day, the importance of international networks, of large groups that join their efforts together to decipher diseases and maybe ultimately come up with cure of complex diseases is very well exemplified by the colleagues from the German Center of Lung Research, who I'm happy to forward their cordial greetings to you. Join us in our efforts to, at some time and some stage, maybe cure this devastating disease which we call pulmonary hypertension. Thanks for your attention.
Thank you, RD. Great talk, great work for yourself and your colleague in Gießen. And it's a great talk complementing a fantastic session we had earlier on RV and the effect of sotatercept. Now, questions from the audience on the work that has been presented, hemodynamics, RV, coupling and uncoupling, and Reyes-Goat effect. Kelly?
Hi. Hi, Ardy, a fantastic presentation, and great to see the fantastic work coming out of Gießen. I have been sitting with your supervisor, Hoder Tello, who congratulates you on your presentation today, so thank you very much.
He should have given the talk, by the way, so.
I thought it was fantastic. So, I mean, the question really, I guess, is do you think we are getting very close now to having surrogates of those non-load dependent markers, moving away from conductance catheters for centers like ourselves who don't have access to this fancy technology? Do you think we're nearly there yet?
I think that we have useful surrogates for conductance derived measurements. I don't think that they have reached the precision of conductance catheterization, but balancing out the widespread applicability, for instance, of this little software, and also the very strong data coming from a cardiac MRI, combining it with other means of pressure measurement, enriches our knowledge, continuously. I think that if it becomes subtle, and if we really want to have a true proof of whatever hypothesis we're currently investigating, we might need to further utilize conductance catheterization, but in real world and in our clinical field, I think we can apply most of those surrogates that were suggested by Hoder and his group.
I mean, I do like your discussion as well about the collaborative nature of research, and I think one of the values in doing this type of work in a very particular way with the conductance catheters, when you have a surrogate, it then allows you to hypothesis generate and then validate in very, very large cohorts of patients. So thanks very much, Ardy.
Sarah?
Thank you so much, Dr. Ghofrani. It's very good to see you here today. I have two questions. First one, regarding the cardiomems. Do you think cardiomems can be also useful? I know we were able only to measure the mean pulmonary arterial pressure and the wedge pressure, but do you think is now with the complexity of the disease and a lot of medication can be used as a targeted for our treatment or response for therapy? The second one about the researches or clinical trials in pulmonary hypertension. Previously, we used the primary endpoint as a 6-minute walk test, and later on we measured the secondary endpoints. But my question is, should we from now and then assessing the multi-component improvement, such as the studies in the sotatercept, because we know the pulmonary hypertension, it's a complex disease. It's a syndrome with affecting a multi-organs, not only the heart, not only the right side of the heart. And only targeting one measurement, it's maybe less helpful comparing to multi-component improvement. where it is more influencing or more suggestive of the survival rate rather than just one variable?
Thank you. Both very relevant questions. I may start with answering your second first. The 6-minute walking distance is the hurdle that the US FDA has given us to jump over when we want to prove that a drug could be beneficial for patients with PAH. So far, it remains their minimum requirement to grant you an approval for a new investigational drug. You have to show, an improvement, a statistically significant improvement in 6-minute walking distance, and then you're fine. You get an approval. And many people in the FDA tell you, don't make your life too hard by suggesting new endpoints, which still need to be validated, are experimental. We don't trust in them. And the FDA, unfortunately, does not trust in hemodynamics at all, which shows us that they don't understand pulmonary hypertension at all. But they use 6-minute walking distance. They tell you, jump over this hurdle, we grant you approval. Don't make your life more complicated than needed. Maybe there will be a change in mindset if the community is able to provide other reliable surrogates of survival, of field function and survive. That is important for the agencies, and it's our obligation as a scientific community to provide those reliable surrogates, which are still missing. But I like the multi-component improvement endpoint. Your first question regarding CardioMEMS has just been in part answered by the study our UK colleagues did with individual dose finding with imatinib, just highlighting how important it can be to have an online measurement or a continuous measurement of pulmonary hemodynamics when you administer an experimental drug. And what they did was to use CardioMEMS in all of the patients that were receiving incremental doses of imatinib, and they could nicely show individual responses just by means of measuring the pressure, but nevertheless that was very important information. So leave out the limitations of CardioMEMS. It doesn't provide you with wedge pressure. It doesn't reliably measure cardiac output, but it measures pressure, and it is important, and it is an important additional information which can add to our understanding when we conduct clinical trials. Not so sure about the utility in the real-world setting, so I would not necessarily give every patient with PAH either the CardioMEMS or the QuarLock. It's more for investigational purposes, but it provides very useful information.
Thank you so much. Very helpful. Thank you.
So again, good afternoon. So thank you for this elegant talk. Just my question about volume pressure loop. I mean, it has been there in congenital heart disease. Sorry. So
no worries.
There in congenital heart disease in like early 80s with Professor Andrew Reddington. I mean, try to reproduce in a substrate of patient whom they are very unique. It's called congenitally corrected transposition, where your RV is the subaortic, the LV is subpulmonary, and then you try to retrain the left ventricle by doing a pressure volume loop. But then that all faded out because the number of patients are very, very, very minimal. He tried even to take this study to Toronto when he was in Toronto Sick Kids. Scientifically, it is a very, very promising. So my question now, is this, you think, would be the future to really get those conductance catheter in practice and then start to do it or not?
Well, I can clearly answer that conductance catheterization is not for routine clinical workup of our patients. It's much too demanding. There are even fewer and fewer companies which will produce the catheters. So even catheters are becoming rare. But now that it's been used extensively in the work of Hoda and his group to validate other less invasive or even entirely non-invasive surrogates, I think we can rely on those other parameters, all with the intention to assess contractility in these patients. So yes, we should not use conductance catheter regularly in our patient, apart from research and research questions. But also, yes, we should focus more and more on the assessment of EES to EA by means of the less invasive technologies or routine clinical technologies, right, like right heart catheterization, and then calculating from the normal pressure curves that you get with the Swan GANs using the software.
Ardi, although we had an advisory board meeting yesterday, and although it was not about Reisagwat, obviously, but you mentioned a statement. You said Reisagwat is undervalued drug or under-promoted drug in the history of pulmonary hypertension. Why is that?
This is slipping around in thin ice. So I think it was a combination of many things. The drug was evaluated at a historic time when medications that were tested were using 6-minute walking distance for good reasons that were just mentioned, and the trials were small, they were of short duration, they only had to show improvements in walking distance over 12 or 16 weeks, and it would grant you approval. At the same time, there was a change in philosophy in parts of the community who said, at least for second or third in class drugs, we don't consider it important enough to show that a given, third in class endothelin receptor antagonist improves walking distance by 2 or 3 meters more. That doesn't tell us whether or not it is really useful as a new addition to the treatment armamentarium. So the importance of short-term trials was deliberately downgraded by parts of the community in favor of using clinical endpoints, which were useful for second and third in class to show that they have added value, but riociguat is a class of its own. So another reason was that it was mistaken as a other form of PDE5 inhibitor, which it isn't. It's an own molecular entity, different from PDE5 inhibitors, which only amplify an existing NO signaling axis. These drugs do not depend on the presence of endogenous nitric oxide signaling or even the intactness of the soluble guanylate cyclase. The stimulators can directly stimulate soluble guanylate cyclase to produce the second messenger cyclic GMP in a dose-dependent manner, whether or not nitric oxide is present or not. So this makes it considerably different from PDE5 inhibitors. You are aware of the replace study, for instance, and the respite study in which PDE5 inhibitor failures then received riociguat and improved. They were uncontrolled, but it was a clear signal that we are speaking of different classes of drugs. So in this whole historic, let's say, time frame where the importance of clinical endpoints as endpoints for clinical trials was highlighted, basically there was not enough noise to make riociguat as popular as it would deserve it. It's a mainstay of therapy in CTEPH because, it is without discussion, it's a very efficacious and in many parts of the world only treatment for these patients. In PAH, it is undervalued, and I hope that with reminding people how strong this actually is, this medication, it is one way to revive its popularity again.
Thank you so much. Any questions from the audience? We had a fantastic morning session. Great. Thank you so much, Dr. Ofrani. And it's a time now to close the morning session for today. And we have a long break for the Jum'ah prayer. The lunch will be served in the lobby, just downstairs. Use the escalator to your right when you go out. And we will restart our afternoon session at 1:30 sharp. 1:30, we'll see you then. Bye-bye.
So, welcome back to this new session, continuing focusing on pH treatments. Before giving the floor to my friend Paul Yu, I want to start thanking the chairman of the conference, Professor Abdullah Al-Dalan, for the kind invitation. It's my first time on the SAP conference, and very happy to be part and join this wonderful meeting that has been built up over years. So please, Paul, you're welcome.
Thanks so much, Roberto. And welcome to this afternoon's first session, session 4, pulmonary arterial hypertension treatments. It's my pleasure now to introduce HQ Han, who's going to speak about targeting SMAD2/3 signaling pathway, a potential therapeutic molecule with less side effect.
Oh, thank you very much, Dr. Yu, for your introduction. And first of all, I want to thank the organizing committee, especially Professor Adlan and Dr. Alarini for your kind invitation. It is a beautiful city, and it's a great conference. So the first time I'm here, I feel very honored. So let me, in this talk a little bit about signaling, because if anything, this pathway is the one that I can claim my team has worked the longest. We worked almost 30 years on activin signaling, so we know a few things. But specifically here, talking about a goal, objective, if you will, focusing on this pathway called SMAD2/3 signaling pathway, with a goal to identify, hopefully one day, a highly effective and safe molecule, okay? So now we have a molecule called ALG-801. So let me talk a little bit about that, because that molecule is pretty good in many ways. And let me start by showing this, this data slide. And then you can see that there are 3 points I want to make here. And this molecule, first of all, is by structure, is a novel molecule that actually we pioneered this design by making a hybrid molecule between the two receptors, ActR2B and ActR2A. So this way we make a less immunogenic molecule, if you will. And the technical objective is to get a molecule that can selectively inhibit SMAD2/3 signaling. And here, we would like to be able to neutralize a set of a molecule that is a little bit more complete. And the several ligands listed in there, actually Dr. Yu's early work referred these molecules as high affinity binders or high affinity molecules. Indeed, they bind very, very tightly to the ActR2A, ActR2B receptors, and the receptor complex involving the BMP receptor as well. But I'm not going to go to that level of detail, but rather say that our molecule has a hallmark in that it does not bind or neutralize BMP9. And you can see here on the left, on the right side, I'm sorry, in there, you can see that that's the solution-based binding, and which is kind of unique in its own way, because it's not a sort of a regular binding assay. It's a solution-based binding assay. So with that, you get a more physiological binding measurement. So you can see that the 801, as I mentioned, is a hybrid molecule. It's two parental molecules, ActR2A and ActR2B, in the FC form, the trap, the wild type trap, compared to 801, you can see the difference. The 801 no longer bind to BMP9, and in a cell-based assay, you can see that there is no neutralizing activity. Why that's important? Because we believe that by completely avoid the BMP9 binding, you can avoid some major side effect as listed in there. So now, I think, in fact, in the literature, it's already reported, a long time ago, actually, that loss of function of BMP9 has some pretty serious consequence causing bleeding syndrome in humans. And also, if you neutralize BMP9 with an antibody, you grow red cells very effectively. So with that, you can see that the BMP9 sparing that we achieved is a complete sparing, as a hallmark of this molecule, means something. Okay, so now I want to give you an update here with our clinical progress. 801 has has successfully completed its MAD study in postmenopausal women and achieved proof of concept in both safety and efficacy. I probably should skip the details in here, but say that it's safe and well tolerated. It does not have a high hemoglobin liability. It has no effect on platelets. So that's the good start. And with that, at the same time, we saw a very nice PD response as listed in there. And you can see that there's FSH change the right direction. There's considerable muscle gain. So this is very different compared to sotatercept, because sotatercept does not have muscle growth effect. So now we also have fat loss, we have insulin sensitization, we have a bone anabolism. So there's outstanding metabolic benefit with 801. So let me show the data here on FSH first. You can see that we achieved in this MAD study a nice dose-dependent response in a statistically significant manner. The 3 doses elicited the FSH suppression remarkably well, up to 60%. So now, this is the PD data, the pharmacodynamic response in muscle mass measured by MRI, and in this is abdominal fat, the fat mass. You can see that muscle gained, fat lost. So this is the metabolic effect in the postmenopausal women that we measured. And this is important. Okay, so now we plotted this spaghetti plot on hemoglobin and platelets. And here you can see that, essentially the treatment has no impact on these parameters. And that kind of suggests that, our molecule really differentiate if you compare with sotatercept, is a different profile. And in fact, we early on, we performed extensive research studies, including the erythropoiesis assays in animals, and you can see that in this head-to-head animal study, compared to the, let me see here, compared to the ActR2B FC, this is the wild type ActR2B FC, And this is the wild-type ActR2A-Fc, or sotatercept analog, if you will. And you can see that our molecule, 801, compared to control, no change. But these two molecules had a profound effect stimulating red cell growth. So that data actually corroborated with our clinical data. So now I want to just switch to some of the, I would say, more relevant disease model studies for this audience. And I presented this data recently in the European Respiratory Conference. And essentially, I wanted to, just present part of the data that are presented in there, focusing on the head-to-head treatment efficacy comparison study, ALG 801 versus ActR2A-Fc. So, the study design is as follows. So basically, the animal get induced, get treated with this Sugen compound, but at 10-fold higher level. So this is a severe condition purposely used to give you a sort of a sicker animal, if you will, so that you can test the treatment. Three weeks later, the treatment started. So we actually have two molecules we gave to Core Dynamics in a blinded manner. So it's a blinded study, okay, head-to-head. So then you can see that, the hypoxia was also continued. So it's like a 6 hours continued hypoxic exposure. So the Core Dynamics team actually recommended that paradigm, and we accepted it. After completing the study, the compound was unblinded. Then we got the result. And here you see that the PAP on the left, on this side, and let me point this, the MPAP, and the IVSP, and Fulton index. You can see that the blue bar represents the effect of 801. And this dark gray bar is sotatercept analog. And you can see that our molecule 801 did better efficacy-wise in those parameters. You can see that all these three parameters appear to be better. And looking at the vasculature, the small arterioles, when they were analyzed, again, the data showed that 801 did better in reversing the remodeling, if you will. And then this is the histopath analysis. You can see the blue bar compared to the gray bar actually showed a better improvement in this pathology. The macrovascular or capillary damages were treated better. And finally, I wanted to show the biomarker, the serum BNP level. And you can see the difference is pretty remarkable in that 801 virtually normalized the BNP, and compared to the sotatercept equivalent is a big difference in there. And in terms of survival, I think 801 was also able to score better. So this is at the end of the study, 90% of the animals survived versus nearly 70% with the sotatercept equivalent. So I think, we have at least one hypothesis that we can try to use to explain why we're seeing this difference, because our molecule is able to, in fact, more broadly intercept these the Paul-Use high affinity ligand for SMAD2/3 signaling, if you will, activin A, activin B, activin AB, GDF8 and GDF11. The most important thing is that for the GDFs, the sotatercept just doesn't do a good job because the binding affinity is lower, and as a result, in vivo, it doesn't neutralize. For instance, if you put sotatercept in a mouse, it does not stimulate muscle growth. And that's because the GDF8 neutralization is not strong enough. And our molecule grows muscle so well in animals and in human. So that's one difference. And the other difference, of course, is still the sparing of some of the BMPs. BMP9 is one. We have some other BMPs that can also be spared by 801. So now let me just summarize what I presented. So I basically showed you data that suggests that 801 had a validated, improved safety profile and a robust PD response at the same time, as shown in phase 1 studies. And we also got data in head-to-head comparison. The treatment efficacy of 801 is better than that of sotatercept. Okay, so now the molecule now is ready, really ready to enter phase 2. So that's a pretty exciting scenario that we're facing. And FDA, the US FDA, has granted ODD to ALG 801. So that's the update there. I do wanted to take this opportunity to thank my co-worker, Xiaolan, who is in the audience, who is instrumental to the project, and also the core dynamic team, Michael and his team members. So I won't go through each one of them, but I say that I really owe them for their hard work. And finally, Dr. Sehe and Fremking gave us a lot of clinical insight and guidance that kept us going, because this is important. Why we're doing this, right? So I wanted to thank all of them. Thank you for your attention. Thank you.
Jean-Luc Vachery, that will talk about treprostinil palmitil inhalation. Please, Jean-Luc.
No, I'm good. Thank you. No, they had to fix the screen. Thank you, Roberto and Paul and colleagues. Welcome back. So yes, I'm going to touch upon treprostinil palmitil inhalation powder in patients with PH. And when talking about a new derivative of a prostacyclin pathway agent, it's a little bit, it's probably a little bit important to go back slightly in history. You've seen that in my previous talk. It's the treatment algorithm change. And when looking at it, you would think that everything started in 1998. But in fact, we all know it started way before that with the use of calcium channel blockers. And I got back to this article by Lou Rubin in New England Journal of Medicine in 1997, after there were several trials that were done with intravenous treprostinil that provided one of the very first treatment algorithms before the Third World Symposium. And this is what it was looking like. So basically, The proposal was, before we have all the drugs we have today, to use intravenous epoprostenol in patients with functional class 3-4 who are not responders to nitric oxide or IV epoprostenol. It was the pre-Sidbon paper, the pre-Sidbon era, where it's marked that there would be a moderate or minimal fall in pulmonary vascular resistance without very significant definition. So this is where we're coming from with intravenous epoprostenol. And then if you look at the milestone randomized controlled trials, the ones that really paved the way of PPAs, prostacyclin pathway agents. In PH, we had a large multicenter trial in 1996 with IV epoprostenol. Then we had oral baraprost and treprostinil subcutaneously that were published in 2002. Oral treprostinil in 2013, and then oral selexipag in 2015. So you can see here that the story of prostacyclin agents or prostacyclin pathway agents lasted probably for the very early days of what we called at that time primary pulmonary hypertension. Now, of course, what's missing on that slide is this. Those two drugs that were delivered by inhalation. One is iloprost. Again, 2002, it was a great year for pulmonary hypertension, bringing 3 new drugs on the market, 3 new drugs available to patients, and inhaled treprostinil in 2010. And one of the things that is interesting with those drugs delivered by inhalation is that the devices that were used started from something that was super bulky, super complex for patients with iloprost and the early days of the treprostinil, to smaller and smaller device. So the idea here was to try to find solutions that would decrease the burden on patients and improve the tolerability of the compounds. In the most recent treatment algorithm, if you look at it though, the space of prostacyclin has been significantly reduced. At baseline, it is recommended in patients with high risk, and the first-line recommendation would be a combination of oral therapies with intravenous or subcutaneous prostacyclin pathway agents. And then at first follow-up, only for patients with intermediate low risk, you would have sequential combination with oral or inhaled PPAs, and intermediate high or high risk recommendation is to use parenteral prostacyclin. So the space of inhalation of prostacyclins in our current treatment algorithm seems to be fairly limited to those intermediate low risk patients at first follow-up after initial therapy. And of course, it comes back to what I was saying a minute before, which is that when you're making a decision to treat a patient with prostacyclins or with any treatment, it's always a shared decision, and you need to understand, at least this is what we try to do, what my nurses are doing, is trying to understand the patient's perspective, understand the burden of disease, and it goes with the burden of therapy. So of course, intravenous is going to be higher burden than taking a pill. So by just looking at this, you can imagine that there's room for improvement to find new agents that may find their ways between the oral agents and the intravenous or subcutaneous delivery modes. And this is where treprostinil palmitil inhalation powder, or TPIP, as I will refer to in the next slides, comes in the play. It's actually it's a dry powder of treprostinil palmitil, which is an ester prodrug of treprostinil. So this is important. It's a prodrug. It has prolonged drug exposure, lung exposure, and it's potentially a long-acting vasodilator that may last for enough time to allow for a once-daily delivery, so once-daily inhalation. To look at this in a little bit more details, this is roughly how it works. The drug, the powder, is delivered by a commercially capsule-based dry powder inhaler, so DPI, similar than the ones you would do in some lung disorders. Then it goes to the lung where it's absorbed, and then there's an hydrolyzation of the inactive prodrug TP by lung esterase that leads to delivery of active treprostinil into the lung. Then treprostinil, of course, binds to the receptors. This is giving the the pulmonary effects and also the potential systemic effect. However, because it's given by inhalation and because the drug is transformed into treprostinil in the lungs, it is supposed to be limiting the systemic effect that you may see with other formulations of prostacyclins. And in fact, this is what was studied in this phase 1, and I'm going to present to you the phase 1 and the phase 2 TPIP studies. It was done in healthy volunteers. What is interesting here is that it was either a single dose study of several doses of TPIP, but also there was a multiple ascending dose showing from a pharmacokinetical point of view that you can achieve levels of prostacyclins that were enough to be associated with a biological effect. So when you use 85 micrograms, you have an 11-fold lower plasma concentration as compared to inhaled treprostinil. So for a higher concentration, slightly higher concentration of TPIP, you have a lower exposure than with inhaled treprostinil. Tolerance was supposed to be good enough to move on to the next level. And again, the PK of treprostinil delivered by TPIP were found to be good enough to support a once daily dosage regimen, which is, of course, you can imagine having a once daily dosage in 1 DPI. That might be very convenient for patients. I'm going to show you here, and I'm not going to discuss the PH-ILD study that was done and completed last year. I'm going to focus on the phase 2b inhaled treprostinil, which is a drug licensed TPIP, which is a drug developed by Insmed. Actually, this was presented by Eckhard Grünig on behalf of the steering committee at the last ERS meeting. So it's a randomized controlled trial, obviously, where patients were randomized to TPIP versus placebo in a 2-to-1 fashion. Stratification before randomization was done by baseline functional class 2 versus 3, the number of PH therapies from 1 to 2. Then you had 4 weeks lag time for screening time before randomization, as I said, 2-to-1. You see here that the idea was to get an ascending dose of TPIP from 80 micrograms to 640 micrograms. That was the maximal dose, so to be effective. It was done during a 3-week titration period, but some patients were allowed to have an additional 2 weeks if they couldn't reach the 640 microgram, but the investigators had the impression that the dose was ramping up enough. Then there was a steady state period for 13 weeks before the end of treatment and primary analysis was done, then followed by a follow-up safety analysis. The primary endpoint was the change in PVR at week 16, approximately 24 hours post-dose. So this is important. The measurements were done at trough level. At the time, the concentration of TPIP or treprostinil was supposed to be the lowest. There were secondary endpoint, important ones, walking distance, NT-proBNP, and there were also exploratory endpoints, and I will show you a little bit on the hemodynamic variables. And I must say that I like what Ardi was saying before about invasive hemodynamics. It remains important in PH, and unless we get something better, we will probably still keep on relying on invasive hemodynamics. So these are the patient's characteristics. As you can see, it's a very typical population for a PH trial, at least for phase 2b. The majority of patients were in class 2, 65% in both groups. Most of them, 3/4 of the patients, were already on double therapy. So this is important because we're targeting here the third pathway. The breakdown by pH subtypes is unsurprisingly dominated by idiopathic pH, and you can see the walking distance, 340 meters in the TPIP group as opposed to 371. The NT-proBNP levels were high enough to support the fact that the patient were still in need for an additional therapy. These are the hemodynamic variables. PVR, as you can see in Wood units, around 10, so that's really significant. Pulmonary vascular disease, the wedge pressure was normal. Right atrial pressure was in the expected range of 7 to 9 or 8 to 9 millimeters of mercury. Cardiac index was maintained, and of course cardiac output was maintained as well, 2.6, 2.7 in both groups. So again, a typically stable population, but in need of an additional treatment. This is the patient disposition. We had 165 screening, 102 being enrolled, 2-to-1 TPIP versus placebo. The rate of discontinuation was about 10% in the TPIP group. We had 89% or 90% completers in the TPIP group as opposed to 100% in the placebo group. What is remarkable is that 75.4% of the patient in the TPIP group reached the maximal dose of 640 micrograms compared to 82% in the placebo group. So not that much of a difference between the treated group and the placebo. This is the result on the primary endpoint, and all the slides are going to, the next slides are going to look the same. You have the a variable that is a variable of interest with a conclusion at the bottom, with the table showing the least square, or sorry, the figure showing the least square changes versus placebo on your left-hand side and the right-hand side, the absolute values for change. And you can see that obviously there was a drop in pulmonary vascular resistance by about 35% placebo adjusted at baseline from at week 16 from baseline. That was highly significant. The same observation was made with 6-minute walking distance, with an improvement on a mean of 35.5 meters. Again, that's at trough level. We saw this phenomenon that we see more and more commonly in PH trials, which is that even the placebo group go up in the initial phase and then go down at the time and the point of interest. So there was a plus 11.55 at week 16 in the placebo group, as opposed to 50 meters or 51 meters in the treated group. So again, that was highly significant. A second important endpoint was a change in NT-proBNP. So there was a 60% drop in placebo-corrected NT-proBNP compared from baseline. And that was quite significant because most of the patients on average reached the 340 or close to the 300 bar that would be associated with a better prognosis. Again, the p-value was positive. This is a broad representation of the hemodynamic variables that was exploratory, but you can see that most of those variables, including mean PA pressure and right atrial pressure, improved at week 16 without significant change in systemic vascular resistance, which supports the idea that there was a minimal systemic effect of the drug. What is remarkable is the decrease in right atrial pressure. It's uncommon, even in phase 2s with PH, to have a drop in right atrial pressure, but also you had a drop in mean PA pressure and an improvement in cardiac output. So in other words, the drop in PVR was accounted for by a change in pressure and an improvement of cardiac output, and neither one in isolation. That's clearly something we feel is indeed important. The adverse event, treatment emergent adverse events, 80% in the TPIP group, 76% in the placebo group. That was kind of even. The most common side effects, as expected, was cough. It affected 41% in the TPIP versus 21% in the placebo group. This is interesting, of course, because it's an adverse event of interest with all inhalation therapy. But if you look at the rate of, or let's say the severity or the intensity of cough, it was considered to be mild or moderate, whether in the TPIP group or in the placebo group. Study discontinuation were study drug discontinuation were cough, dyspnea, chest discomfort, increased transaminase in one case that was ruled out as being caused by the drug, and there was no death reported in the study. So if I may conclude now, this trial was the first one to look at the treprostinil palmitil inhalation powder in patients with PAH in 16 weeks. TPIP was associated with an improvement of pulmonary vascular resistance, so a significant improvement by 35% of pulmonary vascular resistance. But not only that, there was also an improvement in cardiac output, an improvement in walking distance, and a decrease in NT-proBNP with a broad effect across the border of the points of interest. Unsurprisingly, cough was considered, or was seen as the treatment emergent adverse event, 40%, followed by fatigue, by headache and fatigue, which is very consistent with not only the delivery mode, but also potentially some side effects of prostacyclins, and cough level are reported as mild or moderate. There was only one of the 69 patients who had to drop off and stop medication because of the intensity of the cough. So altogether, in this phase 2, we're able to demonstrate more than signals for efficacy. And as was mentioned yesterday by Sam, we are moving now to the phase 3 trial that has already been, that is going to be conducted in a much larger population with PH, hoping for the best and preparing for the worst, but I think hoping for the best. Thank you very much.
I'm delighted to introduce our next speaker of the session. Majdi Idris is going to speak to us about insights from PATH-GCC, PH treatment patterns and outcomes across the Gulf countries.
Thank you, Paul. Thank you so much. Assalamu alaikum, rahmatullah, salatu wassalam ala sayyidi rasulillah. So yeah, we're going to talk about the PATH-GCC trial. It's not yet published, but it's about in the publication phase. And before I start, I just need to share with you a very sad news that happened last night, where basically Barcelona team lost. So that was very devastating to me. So even the great, basically in Spanish they say, really, el mejor equipo del mundo. So definitely that's Barcelona, but they lost yesterday. So if you are a fan of Real Madrid, don't ask any question. I don't want to talk to you tonight. Please, thank you. 4-0, yeah. So that was tragedy. So anyhow, so let's talk about the study, the PATH-GCC study, which is the largest comprehensive PAH registry data across the region, highlighting many important parts of really pulmonary hypertension, burden, treatment, and management. Now, if you look at the primary really objective of that study was to really describe what's happening in the region. Basically, the patient's demographics, clinical characteristic, and the management and the outcome of those patients, looking as well, not only for the outcome, but for the healthcare resources utilization. The data collection period was between February 2024 until October 2024. So when you're looking at the methodology of the study, so this patient, basically this study was a multi-center, retrospective, non-interventional study really conducted in 9 PAH centers in the Gulf really countries, 4 Gulf countries, Saudi Arabia, United Arab Emirates, Qatar, and Oman. So basically we captured the patient from really 1st of January 2018 until 31st of December 2022. But we also allowed then 1 year follow-up period to really follow up those patients that were captured late. So we have a minimal, basically information really captured for 1 year and maximum really capturing information for up to 6 years. Now, inclusion criteria, we were looking clearly for basically adult patient, group 1 PAH, diagnosed by right heart cath. So really right heart cath was mandatory to make the diagnosis and to make the patients eligible to be part of the group. And of course, they should be fallen within the period of January 1st, 2018 until December 2022. Of course, we ruled out any patients who really do not belong to group 1 or those who were involved in other studies. So if you look at the centers that were contributed, we have 6 centers from Saudi Arabia, 4 from Riyadh, 1 from the Western Province, and 1 from the Eastern Province. We have 1 center from Qatar, 1 center from United Arab Emirates, and 1 center from Oman, with a total population of patients of 250 patients. Now, looking at the patients' demographies, 82% of the patients were female versus 18% were male. Of course, when I was reading the initial manuscript, they said patients were only male and female, and I said, were there any kind of other gender? So anyhow, I'm not going to go there now, but that's fine. So 92% were Arabs, and only 5% were either active smoker or former smoker, and about 69%, especially from those patients coming from United Arab Emirates, were actually insured. So this is actually, if you look at the age of our patient, were 40, so that's probably 1 to 2 decades younger from the Western population. But if you compare that to the initial publication that came from really Saudi Arabia, the first study was on 2014 by us, showing that the age at that time was about 32. Dr. Dalan, about 6 years later, reported a little bit younger, but now we are getting a little bit more older patients. So now probably we are heading towards what's the Western population, but so far we are about at least a decade younger, our patient, comparing to the really Western data. So let's look at the result of this study. So the first important information that we captured was that our patient, it took them about 1.6 years to get diagnosed. Now, I'm going to show you that this is much better than our previous data, but it's still long enough time, and it did not matter how much the patient was symptomatic. So even those who were at high risk initially, intermediate or low, everybody took around the same time before diagnosis is made. So really, the more symptom the patient have did not shorten the duration for diagnosis. And when we try to look at the etiology of those patients, about 41% belong to the idiopathic PAH, which is, again, consistent with the international and our previous data. But looking at the second important one, which is the congenital heart disease, now it's followed very closely by the connective tissue diseases. Another difference compared to the Western data, that systemic sclerosis is not the most common cause of CTD PAH. In our population, it seemed that SLE followed by mixed connective tissue diseases are more common cause for PAH compared to systemic sclerosis. So we need to keep that basically in mind. Looking at vasoreactivity tested, that was tested in about 90 patients who belong to the idiopathic, heritable, or really drug-induced, and those were about 12% were vasoreactive. All patients who were vasoreactive in our cohort came from the idiopathic PAH group. Now, let's try to look at the patient characteristic. So those patients, very interestingly, when they came in, about 55% were non-function class 3 or 4. So they were function class 1 or 2. You would, of course, argue with me if there is anything called function class 1 in PAH, but that's what was really captured. So, but at least 55% of the 55% of the patient belong to non-function class 3 or 4. And that was extremely important, because when we compare this data with our initial data that was really published about a decade ago, 1 year ago, you can see at that time about 75% of the patient were function class 3 or 4. Nowadays, it seems that we are getting better, and now only 45% of the patients are in function class 3 or 4 when they were captured. Also, you can see that at that time, it took us about 27 months to make the diagnosis, and now that is really down to about 18 months. So it seemed that there is signal that we are now capturing patient a bit earlier than what we used to do in 2014. What about the other parameters? If you look apart from the function class, the 6-minute walk distance, you can see that the majority of patients, about 62% of the patients were really walking in about between 165 to 440, keeping them in the yellow, really in the yellow zone. And that was indeed, if you look at the really European risk classification, about 60% of the patients were in the yellow zone, the intermediate risk, about 25% in the green zone, and about 17% in the high-risk group. Now, let's look about the diagnosis of the PH in those patients. Now, of course, I would remind you that all of those patients had right heart cath to make the diagnosis, and you can see that only the minority of patients fall within the new definition, i.e., basically pressure, mean pressure between 20 and 25. So about 4% of the patients fall in that category, and about 95% were more than really 25. Very similarly, when you look at the PVR, only 5% of the patients had PVR between 2 and 3. The majority of patients having PVR more than 3, again emphasizing that what's been really observed with other registry, that only the minority of patients are really captured when they have very, very mild pulmonary hypertension. The mean wedge pressure was really 11.7, and that was achieved in the majority of patients. The really author sometimes accepted some patient with a little bit of higher pressure when the clinical case was going with pre-capillary pulmonary hypertension, and that probably there might be some problem in really wedging. If you look at the hemodynamic data, the mean right atrial pressure was about 9.7. Mean pulmonary artery pressure was 53, so that's again tell you that those patients have decent disease, and PVR is in the range of 12. Now, keep that in mind, and correlate that with the symptomatology, that significant number of those patients were actually in function class 1 or 2, but just like tells you that even those who have milder symptoms, they were still suffering from severe hemodynamic disease. What about the comorbidities? Comorbidity was quite common, despite the fact that those are much younger population compared to the data from Compara and from Review. And from American data, you can see that about significant number of patients, about 70%, they had comorbidities. And very interestingly, the pulmonary comorbidities were the most common in our series, followed by the endocrine, to great extent thyroid disease and bone disease, osteoporosis, but other disease, and then followed by really cardiology. And if you look at the number of really these really comorbidities. From the cardiac point of view, systemic hypertension was the most common really cardiac comorbidity, while the obstructive lung disease, mild obstructive lung disease, asthma or so, were the most common really pulmonary comorbidities. What about treatment and treatment behavior? So again, we noticed very, very significant observation that really at the beginning, when we started the study, you can see that about 50% of the patients were on monotherapy. Of course, this was against all the guidelines, but that was the reality. 50% of the patients in these countries were on monotherapy, about 40% on upfront combination, and less than 10% were on triple therapy. Now, by the end of the observation, you can see that now we get much better, that only 18% were on monotherapy, and the vast majority of patients were either really on dual or in triple therapy. So that at least gave us some reassurance that the awareness programs, etc., is working, and now more and more patients are getting treated really with a combination therapy. And when you distribute that among the risk status, you can see that low-risk patient, 60% at the beginning were at the baseline were on monotherapy. And that, if you look at it, even what happened with those who were at high risk, it did not make a difference. Very similar to the pickup rate. Even those who have more higher risk stratification, at the beginning, most patients were on monotherapy. And that was something that we felt really relieved when we found that the behavior was getting better and more patient was shifted to either double or basically triple therapy. Now, if you look at the end of the observation, that 70% monotherapy at the beginning dropped significantly, and you can see that those who were at high risk, now only a minority of those patients, I mean, you can really argue that still 10% is high to have 10% of patients with high risk on monotherapy, but I can argue that 10% is far better than 70% at the beginning. But it was clear that the more severe the cases, when you follow those patients and we learn more about those patients, were treated with combination therapy. Finally, about the end of the observation, and we look at what happened at the end of the observation and how patients move from one risk status to another risk status, and we see that significant number of those patients who were really at low risk, about 60% of those patients stayed in the low risk, while those patients who were at really higher risk, they moved significantly to more lower risk group with the treatment. So it seemed that we were doing a little bit okay when it comes to, I will not say that this is ideal situation, but clearly that we are moving forward and we are following the guidelines in a better way. Overall survival in this cohort was about 94% looking at that treatment period. And you need to understand that those patients were really followed from at least 1 year to the maximum of 6 years. And you can see that survival correlate very well with the really risk status of the patient. No one died in the low risk group, and about 10 died in those patients who belong to the high risk group. Now the healthcare resources was another important one. When you look at the patient who were requiring hospital admission, about 45% of those patients required hospital admission, and the causes of that admission in the majority of patients, about 90%, were related to the pulmonary hypertension. And you can see clearly that the higher the risk of the patient, the more likely that that patient will be admitted and will stay longer basically in the hospital. What about the ICU admission? Again, that was correlated well with the risk stratification, and those patients who were admitted to the ICU, about 20% of the population stayed longer in the ICU, up to 25 days, if they were at the higher risk status. Emergency room visit followed the same pattern. The more severe the cases, the more likelihood that those patients were actually admitted to the emergency room. So to wrap up the discussion, I think we really observed very interesting phenomenon information when you look at our own data compared to the Western data. Number one, much younger really population, 40 years of age compared to about 60. So we're looking for 1 to 2 decades different. Now, when you look at the other interesting observation, that when we look at those patients in particular with connective tissue diseases, we noticed 2 important things. In the earlier publication from Abdullah and read our data, congenital heart disease was much, much higher compared to the CTDs. Now they are close to each other. So now we are capturing more CTD patients really in our patients. But the other important one, that's now, it's not the systemic sclerosis, it seems, most like SLE or mixed connective tissue diseases in our population. Congenital heart disease is still quite common in our region, and really it constitutes about 25% of really our cases. Female predominance, it seems to be consistent really all over the place. Favorable initial risk was really shown that the majority of our patients, when they were diagnosed, belonged to function class 1 or 2, about 55%, compared to less than 20% in our data and in the Western data. So that was maybe because our patient was younger, we didn't know, but that's what we really observe in those patients. We still have problem, I think, with the monotherapy at the treatment, although it seems that we are getting much, much better really over time. And now the minority of patients, especially those in the intermediate and high risk, are treated only with monotherapy. Comorbidity, despite the younger age group, it's still there. 70% of the patients, they have comorbidities. The pulmonary comorbidity seems to be number 1, followed by the endocrine, and then followed by cardiology, where systemic hypertension basically was the most common. Again, at the end of the really observation, we felt that the trend in the region is getting better. More and more patients were moving to the lower treatment group, and the overall survival was acceptable in this cohort of patients, about 94% really survival rate. Still, we have major problem with the healthcare resources. Those patients are admitted to the hospital, to the ER, to the ICU, and definitely that need to be as well understood and really taken care of. Thanks to the basically principal investigators, we all look here much younger than what we are now, and we look much better there in the image compared to now, but that's, I really got the best images there. And definitely special thanks to the MSD and Dr. Mohammed Safwat for their great work with us, and to our team, really the fellows, the nurses, the pharmacists, the technicians that work with us. I would like to really thank them all sincerely. And remember, despite the defeat yesterday, Barcelona is el mejor equipo. Thank you.
Thank you, Majdi. Please come and join the panel. So now it's really my great pleasure to introduce Marc Humbert. Unfortunately, he will not be here in the room with us. He will have a talk virtually, but I think he doesn't need any further introduction, but I would say, let me say, he is really very well known to all of us, and I would say one of the most important leaders in the field of pH in the research community. So Marc, please.
Thank you very much, Roberto, Paul. It's my great pleasure, great honor to be with you, sadly remotely, but I hope the communication will work fine. So congratulations on this outstanding 90th meeting of SAPH. It's getting bigger and bigger with excellent program and communication, so well done. So I'm going to share my screen. I hope you can see it. I'm going to put it full screen. Let me know if you cannot see it. So my name is Marc Humbert. I work in Paris at the Université Paris-Saclay, and I lead the French Pulmonary Hypertension Reference Center with many colleagues you know very well, including Olivier Sidbon with you in Jeddah today. I wanted to indicate some disclosures. And clearly, it's a privilege to work with so many companies, and it's a privilege to present today at SAPH 2026. So, of course, we are fortunate in pulmonary arterial hypertension to have so many drugs approved. It is certainly the rare disease with the most important approved agents. And historically, derived from great quality research, we have, together with pharma, scientists, clinicians, developed quite a lot of drugs targeting the 3 major pathways. And for a very long time, these 3 pathways remain stable. And we did not have more than that, but we could combine drugs. We could combine drugs according to risk. We just saw very good data from your local sites. But of course, our wish was to develop new drugs. And we borrowed this slide from Martin Wilkins from the UK. And of course, endothelial cell dysfunction is critical in PH, and that's an obvious statement. But targeting only some pathways is certainly not sufficient, and we need to think outside the box and try to have more targets, more testing, more drug development in order to achieve what is our ambition, remission or cure. And we are very far from that for most patients with PH. Of course, we learned a lot from genetics, from the understanding of growth factors, of the mechanism related to inflammation, metabolism, neurohumoral factors, and oxidative stress. And the idea is to reverse remodeling. I know you had great talks in the last 2 days with masterpiece from Paul Yu and others. So I'm not going to repeat too many things, but I will then walk you through new pathways. But I just wanted to start with one statement. And of course, clinical trials in PH are many. We are very, very fortunate to have such a strong community. But something I really don't like is that many studies have not been reported in publications. And really, my ambition today in 2026 is that every single study found on clinicaltrials.gov should be reported in a medical journal. And that's something we really need to do. Of course, you see here on that slide that half of the studies were reported in a publication, studies which are positive or negative, by the way, but half which are negative have not been published, and that's, I think, a concern. We don't have a shortage of drug targets, and we can have drugs developed exclusively for PH, but many are repurposed or developed for many indications. And of course, in our field, the genetic background is important. The growth factor superfamily are very important. Inflammation, metabolism, estrogen signaling, humoral DNA repair, epigenetics, serotonin, dopamine, VIP. So all these mechanisms have been tested somehow or are in the pipeline. So that's beautiful. And one of our major concern is how can we prioritize one drug development versus another one for a condition which remains rare. And the prevalence of PH ranges between 15 and 50 per million. In France, it must be more or less the same thing globally. So here we have a concern on how to develop the right pathway, the right pipeline. But from the start, I think there should be, we should have a commitment of publication of positive or negative studies, because negative studies are not negative per se. They give information because we do design good quality studies. So a negative result is an important result. So our target is pulmonary vascular remodeling. We can try to dilate these narrow vessel, but we should have an active development of drug which could be reverse remodeling or which could prevent the progression of remodeling. And obviously, you have heard a lot about the TGF-beta superfamily. It's a very complex pathway, but we understand it better and better thanks to scientists like Paul Yu, Christophe Guignabert, and many others. And we are very, very lucky to have such a good quality of the understanding of this pathway. And what is very important is that you have a balance between the SMAD158 phosphorylation and the SMAD23 phosphorylation with two, it's a very, very simplified message, but two major pathways one which is pro-proliferative and one which is anti-proliferative. And you can try to interact with these two pathways as best as we can, with a major focus on safety and efficacy. And of course, we don't want to eliminate what has been beautifully developed in the past. So our thinking is more add-on therapy rather than replacement, but we can discuss that later. So the TGF-beta pathway is extremely interesting, and you have heard a lot about it, so I'm not going to be too long on that, but you can try to stimulate the BMPR2 pathway with the idea to improve the SMAD158 phosphorylation signaling. And you could use recombinant protein. It has not been very successful until now. You can consider activators of this pathway. You can consider gene therapy. And you have the pro-proliferative pathway in red, which is TGF-beta activin driven. And here you can consider neutralizing antibodies, ligand traps, inhibitors, and antisense. And obviously, the great achievement of sotatercept is based on activin signaling inhibition, with an idea that it could act as a reverse remodeling. But finally, the speed of action of this drug and then the plateau makes the reverse remodeling more as an aspiration than a real fact, at least in human. And clearly, we have achieved a lot with sotatercept, but it's stage 1 of the TGF-beta superfamily targeting, and there will be more interactions on this pathway very soon. But of course, I'm not going to develop the sotatercept because now sotatercept is an established therapy. We no longer have 3 pathways. For me, it's a great relief because I didn't want to spend all my career on 3 pathways. So now we have a 4th approved pathways. It has been a privilege to be a strong player in the drug development with many people in the room today. And well, we now have 4 pathways. But targeting the TGF-beta superfamily cannot be summarized to sotatercept. With Olivier Sidbon and many colleagues, we published a few months ago this review article which complements the World Symposium proceedings, because in the World Symposium proceeding, there was no dedicated chapter on novel pathways and drug development on these pathways. So I will show you some tables which are difficult to read, and then some slides which are easier to read with very simple messages. And in the next 10 minutes, I will walk you through many pathways very, very superficially, but I will just show you the energy, the passion of our community to develop new solutions. So sotatercept, I don't comment it. It is now available, and the drug development certainly is on other groups of PH and also, of course, in pediatric patients. Then other drugs have been developed targeting the same pathway. The Keros company developed one agent, and 35 Pharma developed another agent, and there are many other agents available. Pfizer as an agent also targeting BMP9, and Novartis as a SMURF1 inhibitor, and of course FK506 with the major work from Stanford University is also available for drug testing and drug development. So one word on cibotercept, so the Keros compound. It was a beautifully designed drug development with what is a novel, modified, investigational at that time, activin receptor type 2B ligand trap. So different from sotatercept type 2B ligand trap. And it was designed to inhibit select TGF-beta superfamily ligands to inhibit pro-proliferative activins and GDF without increasing red blood cells. And it would spare, at least in part, BMP binding. And the idea was to minimize safety signals. But finally, the drug development had to be terminated because of unexpected pericardial effusions occurring at highest doses. And then after a month of supervision, it was found also at the lowest dose. So this development has been terminated. And here you realize that, of course, when you interfere with these very, very important pathways, such as TGF-beta, you can have side effects, even when you have done a very careful drug development program with an attempt to limit adverse events. But when you want to limit adverse events, you should also be careful with the dosage. And of course, it's a great lesson learned, and we will do our best to publish this information this year. So other drug development and the SMURF1 inhibition has a lot of interest. There was a paper last year published by Alex Rothman in Cell describing the therapeutic potential of SMURF1 inhibitors. And clearly, SMURF1 inhibitor is now available for randomized controlled trial. The idea is to propose a drug which will stimulate the SMAD158 phosphorylation and thus the BMPR2, BMP9, BMP10 pathway. So by blocking SMURF1, you would restore BMP signaling through SMURF inhibition. And an agent developed currently by Novartis, LTP-001, is going to be tested. There were early testing at very low dose, and certainly these very low doses were not efficient. And now there is another attempt to test this pathway. Interestingly, this drug is an oral drug, and it's a first-in-class SMURF1 inhibitor. It will be evaluated for PH. And I mentioned before the Stanford studies by Eda and Marlene and many colleagues, and these SAPH2026 studies will attempt to activate BMPR2, rescue endothelial cell dysfunction, and possibly reverse pulmonary hypertension. So this is currently still in development for PH. But of course, you have heard a lot about PDGF. It's very hot right now. So TGF-beta is very important, but this pathway, PDGF, is extremely important. I know that some of you, Olivier Sidbon, Paul, you and others have discussed, and we are eagerly waiting for the results of new studies coming very soon. But when you look at PDGF pathway, of course, imatinib is a major drug which had very, very difficult development in PAH, either by oral route or by inhaled route. But we shall see in the future, and seralutinib is another one. So the inhaled imatinib trial is now published, and those of you who know me, I want to publish every single study. And this one is published in a good journal, the Blue Journal, and you can see that there is no difference between placebo and different doses of this inhaled AV101 drug. So negative study, and this drug development has been terminated. However, Alex Rothman, again from Sheffield, has shown that you can treat patients with oral imatinib. There is the problem of tolerability, but the target dose of 200 milligrams seemed to be good to reduce TPR by around 20% with CardioMEMS with reasonable tolerability. And other companies try to develop new formulations of imatinib, which would be better tolerated, at least at the gastrointestinal level with, for example, this IKT-001 drug, which is currently in drug development and which will be very soon tested in humans, and Regeneron as a monoclonal antibody to PDGFB. So a lot of work on PDGF. Now I'm going to speed on. Targeting inflammation has always been a very, very great ambition. Sotatercept has an inflammatory aspect which should not be ignored. And of course, all the drugs recently tested, which are per se anti-inflammatory, like tocilizumab and others, have failed. Rituximab, tocilizumab, they have failed in PAH. There were some trends in connective tissue disease, but this is not that clear. So neither anti-IL-6 receptor or anti-CD20 have been successfully developed in PAH. You have other pathways. The hormonal pathways are very important, and many drugs have been repurposed for to target oxidative stress in PAH, including allopurinol, spironolactone, DHEA, and clearly these drug development have to be followed very carefully. Targeting metabolism, we have all read the papers which are very, very preclinical or very, very early in the field. And once again, I think that the bardoxolone data, which have been presented in 2015, should be published, because it's impossible to make our mind without clear proposal on the results of the already performed studies. Epigenetic is getting bigger and bigger, and epigenetics will target both the lungs and the right heart. We know that DNA methylation, non-coding RNA, and histone modification play a great role in the heart and lung of patients with PAH, and HDAC inhibitor clearly will be tested very soon in good quality studies, with the idea that it could reverse remodeling, reverse fibrosis, be anti-inflammatory, reduce pressure, and be antithrombotic. But of course, all these assumptions are hypotheses. Then you will see that we have a long list of targets. I will not discuss them now, but I would like just to emphasize that serotonin has been around for many, many years. Many of us have been active players in the appetite suppressant outbreak of PAH, pointing to the serotonin pathway as a possible player in the pathobiology of PAH. And for sure, serotonin plays a role in PAH, and because there is an enzyme, TPH1, which is upregulated in PAH, it could be targeted to decrease serotonin levels in patients with PAH. And together with Olivier Sidbon and many others, we have launched the ELEVATE-2 study. And it was a study to evaluate the effect of lodastrestat ethyl, a first-in-class drug lowering peripheral and lung serotonin levels. And we published that study, which is very negative, by the way, but it was published in one of the best journals, Lancet Respiratory Medicine, because there is a message. You see here, the placebo arm has no change in PVR, while the low dose and high dose of lodastrestat increased PVR. So when you have a study like that, if you don't publish it, I mean, it's not possible. These studies have to be published, and the Lancet editors were very supportive of that. So my final slide is that it's not over. We will remain very active in the field. There is a lot of room for improvement for our patients, and I'm very pleased to be with you remotely to discuss this data. Thank you very much.
Mark, thanks for a great overview of the new therapies. Glad to see you remotely. Sorry you couldn't be here in person. Thanks for your leadership and for advocating for full disclosure of our positive and negative results. I think that's an important theme. I'd like to start us off with the question and answers right now. And Dr. Aldalan, please.
Thank you all. Again, fascinating session. And thank you, Mark, for being with us from too far. Since being away, let me start with you. A philosophical question. Now, given the complexity of these hundreds of inflammatory cascades and pathogenesis pathways, I think developing a drug that has a magic action to stop all of this is probably almost impossible. So unless you start from way high in the pathogenesis pathway to stop the very first eliciting trigger, otherwise we will not, have a single drug that covers everything. Unless, of course, there are multiple drugs that covers multiple pathways in combination, this is probably another possibility. So I know it's a philosophical question, but I'd like to hear your comment on this.
Thank you very much, Abdullah, and it's a great question. I like philosophy. So indeed, I think when you challenge the field, we intervene too late. We intervene too late, and as we have just seen from your own registry, we intervene late because the condition is difficult to diagnose early. That being said, we know that there are some patients at very high risk of PH, like the BMPR2 mutation carriers, the scleroderma patients, etc. So we have an opportunity to make preventive or very early intervention. And I think the earlier, the better. But of course, it's very difficult to develop. Then in established PH with multiple therapies, I agree with you. It's very complicated, but that's the vast majority of the patients. So our mission is to take care of them and to try to find a way to at least reach remission. And the definition of remission will have to be proposed by experts and world symposia and guidelines. But I think with what we have right now, one of my conclusions is that we should not consider replacing too many of the drugs. We should consider adding on and aim for remission. And then the philosophy is that it's very difficult to prioritize one drug versus another one because you have financial issues, you have also leadership issues. People believe more in what they have developed themselves. But we have to be very open to discovery and innovation. I don't know if I addressed your question, but thanks for making me think.
You did. Thank you so much.
Okay. Luke has a question.
My question also is for Mark. The first one is, are you looking forward to March the 14th? But the second one is around the fact that you talked about there are so many potential targets. Can we really afford to go with the same size studies like in phase 2? I mean, I remember there was some discussion around PULSAR, 100 patients, phase 2. Was this too risky to go in with this completely unknown entity? Fortunately, the story worked out extremely well. But, if we'd done, say, cards on the table as a Brit, obviously I'm a fan of small studies with CardioMEMS, had we done that with Redigistrat, then we would have picked up an early signal for adverse effects. So how would you potentially design smaller studies for faster go, no-go decisions?
Yeah, thank you, Luke. It's very important. The pool of patients is very limited, very limited. And if we want the pool of patients to be larger, then we navigate in very, very stormy waters with elderly patients with multiple comorbidities. So I'm a strong believer that we have to make proof of concept studies of the highest quality in small collective of patients. If you look at the bosentan drug development, the phase 2 was only 30 patients, but they had all the measures made. in terms of safety and efficacy. We knew after the phase 2 that there was a dose-dependent safety signal and a dose-dependent debatable efficacy signal. So then the phase 3 needs to be of a good size. But your question relates to very early phases. And I'm in favor of small group of patients, and we need, as a community, with the World Symposium, with PVRI, with our national and international societies, we need to build strong networks. And for example, in Europe and in France, we have networks. And we have the French network, we have the European Reference Network, and there are international networks led by many of you. So then the big thing is prioritization, and don't take too many risks with unknown targets. Sometimes you hit something very sensitive, but I think what you did with imatinib in the UK with CardioMEMS indicates that there is a tolerability issue for sure, but also a safety signal. So that's the reason why I think many of us should try to say whether yes or no imatinib is the right target. So I think we have sufficient preclinical data, and the Rothman UK data are very important for that, because all the others are very, very concerning. And now we will try to develop, but maybe monoclonal antibody will be more better tolerated, etc. So I'm with you. Small group of patients. It can be open label if the quality of the measure is perfect.
And I look forward to seeing you in a month for Le Crunch.
Okay, the rugby game, huh?
Thank you for the whole speakers. It was a wonderful session, actually. It seems that the horizon is coming with many mechanisms of action, and many drugs are coming. And this morning we were talking about some people who do not respond, for example, to sotatercept, and we are looking for some surrogates that tell us which drug for which patient. So do you think that we need to look and phenotype more the patients that we have? I mean, the pulmonary hypertension patient, idiopathic, for example. And for example, in sotatercept, which patient is going to respond more? From my point of view, which I believe it's naive because I don't have a good experience with a drug. For example, theoretically, I feel the cardiac comorbidity patients are going to be the best to respond. So with the other mechanisms of action, don't you think that we need some other surrogates like genetics, like epigenetics, the metabolics, in order to predict which patient needs which drug?
Maybe I can take that one. The point of responders versus non-responders with sotatercept has not been elucidated. So we don't know what makes the difference between those patients. And in fact, it's also probably true with the other drugs. There may be a broad effect, and we saw that in Triton, the Triton trial, where you're giving 3 drugs versus 2 drugs, you don't do much better. But still, it seems that down the road, there may be a difference in outcome, but after 6 months, you see basically no difference. So perhaps that the 2 questions would be which drug should be started first, and whether those TGF-beta signaling, PDGF signaling pathways should be started earlier as opposed to later as some sort of, I wouldn't say rescue, but later in the course of the disease. So far, we're just using the tools that we see. Echocardiography, walking distance, of course, right heart cath, MRI, whatever the kind of imaging you're using. And we know it's not enough because there's no way to make the distinction. And that was one of the main interests in Triton, which is that none of the predictors of response work because they just were diluted by the potential effect of the drug. So getting towards more precision medicine and having let's say, a biological signature, not only of the disease, but also on the potential response, would probably spare times and resources. It's not only important for patients, but surely for clinical research. But at this stage, I don't think it's completely ready for prime time. But the data that is accumulated from the TGF-beta, PDGF pathway are really novel and going to help us to find our way through all these maze of potential targets.
All right, thank you.
Thank you so much. I have a question. Anyone of the panel can answer this regarding the sotatercept and as a HHT mimicker. My question is, we know that sotatercept have a good response with regard to the pulmonary hypertension. And on the same side, we know that HHT can cause itself pulmonary hypertension. Still, I didn't find an answer how we can balance between that. I mean, why those patients who receive sotatercept and they have signs with HHT, but still their pulmonary hypertension is controlled and they didn't relapse or they didn't progress. Anyone of the panel can answer this question, but I remember Dr. Paul last year you presented a very nice lecture and you brought a table about these mimickers and one of them is sotatercept. And one of the things that you highlight is sotatercept is not blocking the, it's not targeting the ENG or endogelin genes. So this for the first one. The second one or we can answer this one first, then we'll go to the second question.
Okay, thanks for that nice question. And thanks for bringing it up. I think our first speaker, HQ, is working on a program that is designed to alleviate some of that. And so the idea, and you can elaborate, is on finding ligand traps that have refined selectivity that won't affect the ligands that are known to provoke HHT. But I'll let HQ talk about his program.
Thank you for the question. I think I'm not the best one to answer the complex biology, but we know that we're trying the best we can and to come up with a more selective ligand trap and hopefully more effective as well. So I presented our data, and I think that it's just the tip of an iceberg, because when you look at the hemoglobin response, it's abundantly clear that in both preclinical and clinical settings, our molecule that we engineered to get rid of at least some of the BMPs, the binding to the BMP9 and some other BMPs, and we're able to actually create a molecule that has a totally benign profile for the hematopoietic response. And I think that alone predict some of the, hopefully, some good safety to come. That's my answer.
So for the second question, Prof. Idris, regarding the data that you present for the registry, the age onset or the average onset of diagnosis increased now up to 40. So still Western country is like 70 and so on. So my question is I always think about, are we missing those patients, those population? Are we overlook them? And regarding those with who have a positive vasodilator, what's the percentage of patients who have already a long-term responder?
Okay, thank you so much. So regarding the age group, I think we are not the only nation. We know that, for example, Chinese, they have the same basically age around 40. So it seems that there is some really ethnic differences among the groups. I don't think we are really overdiagnosed. Those were diagnosed by right heart cath, and we still believe that we are actually underdiagnosing PAH in the region. Still, we think that there is a lot of patients who were really underdiagnosed. So we need to get better, and it seems that we are getting better. Now we have shortened basically the diagnosis time from 28 months to about 18 months. So we are getting better slowly. Now, regarding the responders, we don't know. We did not capture those patients. We did not follow them in particular to see who did lose basically the bronchodilator, the visual activity testing versus those who do not. So we did not capture that in this registry. But it will be interesting to really look at those patients and see what they are doing.
Thank you so much.
Thank you.
If I may, last one. For Dr. Mark, thank you so much, Dr. Mark, for being here today. Last week I read a paper from Japanese group. It's for a rheumatological disease patient. They were comparing two biologic medication, and they used something very interesting, which is the targeted trial emulsion. They use AI to compare head-to-head the two medications by AI, not by clinical. And they used it by a help of a company. Do you think it will be feasible, especially that it's difficult to have a clinically comparing two drugs and pulmonary hypertension patients?
Yeah, thank you very much. It's a very good point. I mean, when I replied Luke Howard's question, I did not mention AI, but AI can be a great addition to our understanding of the results, and you can make, of course, indirect comparisons. You can make emulated trials. So certainly in the next future, the AI tools will be implemented in our community. And I don't say it will be the solution, but it's a great tool to treat multiple data, high quantity of data, big data. So at the end of the day, you're right. I think we need to work with AI, but we need to clearly state the question before using AI without any strong ID. And no, your point is well taken. I've not read this Japanese paper, but in oncology, you have a lot of papers using AI and emulated trials, etc. So thanks for your comment, and we should work on that.
So, Joanna, the last question.
Last question, going back to the TGF-beta pathway experts. I understand now you're refining, the ligands, the traps, the avoidance of different receptors to better design a more tolerable compound. But how can we test these compounds at the physiological level in animal studies before we find a signal of increased risk of side effects in humans? Do we have different models or combined animal models to answer these questions?
I think that's a great question, but it's always a tough one to try to come up with an animal model that can predict all kinds of toxicological responses. Certain assays in animal are better than others. And especially when we talk about large molecules, it's tough because immune response is one that so far cannot be predicted by any animal model, as you know. So I would say that there are certain biology you can try to test out, but eventually I think primate is better for TGF-beta because you have a fast-growing rat that doubles every month, and the growth factor will be there, like give you some exaggerated response. So primate, unfortunately, very expensive, very difficult to use, but that's what we're counting on. And for human testing, and I think that's the ultimate safety response that we need. Yeah. Thank you.
So very tough question.
Can I just have a quick question to Jean-Luc? Because now it seems that we are going to the era of the inhaled therapy, which is definitely attractive when it comes to the selectivity and to decrease the basically systemic side effects. However, if you look at really our journey with bronchial asthma, we have failed to convince our asthma patient to stick to their inhalers, even severe asthmatic. So even severe asthmatic, only 40% are actually adherent to their treatment. So despite, yes, okay, it's very good to have really selective drug going to the lung, But what are we doing with this compliance issue and really adherence and the local side effect? For example, we have patients on the inhaled Tyvaso, and really the problem is the cough, so you need to sit with those patients. So I'm not sure where are we going with that?
That's a very good point, Majid. Thank you very much for asking. It's amazing, of course, if you move out from the move away from the clinical trials where you have kind of a good adherence. The issue is when you pass 6 months and you pass that bar, then you see problems in adherence. And this is something, by the way, that we know very little in pulmonary arterial hypertension. It seems to be, the legend is that all our patients are super adherent to treatment, but it turns out that it's not really the case. So the idea to have a less complex delivery system for a deadly disease perhaps is a way to convince patients to keep on the drug, one, and B, the tolerance that we saw in TPIP, but also with other inhaled compound in PAH, is much better than what you may see in PH-ILD, for example, where cough is an issue. Although even there, coughing with the drugs that are currently developed, like mostly, for example, or TPIP, the rate of cough seems to be fairly acceptable. Now, of course, I don't have a crystal ball to look into the future, but I think the more convenient, the better tolerated in a deadly disease, that's something that will make the difference.
So thank you, Jean-Luc. Paul, I think you can close the session.
Okay. This was a great overview of some really interesting anecdotes from the Gulf PH experience. Very instructive and probably with important implications on many other populations in the world. HQ, thanks for talking to us about your developments of new improved next generation inhibitors. And Jean-Luc, thanks so much for this exciting story about TPIP. Very encouraging efficacy. Congratulations to you and the team. And thank you all for your attention in the session. For this next break, we're going to extend it by 15 minutes, so we're going to ask people to reconvene at 3:45. Thank you.
Assalamu alaikum wa rahmatullahi wa barakatuh. On behalf of
Assalamu alaikum wa rahmatullahi wa barakatuh. Good evening, everybody. It's my pleasure to start the last session of today. We have challenging real-life cases. I know people like filled with a lot of talks today about basic science and treatment of pulmonary hypertension. A lot of things has been discussed today. So now we'll go to something else. Maybe we'd like make things a bit more exciting to talk about cases. So I would like first to introduce my colleague in this session. The first one is Dr. Professor Ushiro Fukumoto. He's a professor and chairman of the Division of Cardiovascular Medicine, Department of Internal Medicine, Kourmei, correct? University School from Japan, as well as Professor Sean Gain. He's a director of National Pulmonary Hypertension Unit, Dublin, in Ireland. So the first case, we are going to talk about it today. First of all, thank you, Dr. Noor Taif. So we are going to cancel your case today. I'm sure you put some time there to review. However, we'll go to 3 cases instead of 4. So the first one will be a rare case of subacute pulmonary arterial hypertension with a dear friend, Dr. Faiz Al-Ahmadi. He's a pulmonologist, pulmonary hypertension and balloon angioplasty from King Faisal Specialist Hospital in Riyadh, Saudi Arabia. I'm sure Dr. Faiz will give us really a rare case. We are talking today about how rare the pulmonary hypertension. So what about your case? So Dr. Faiz.
Good afternoon, everyone. I'm Dr. Al-Ahmadi from King Faisal Specialist Hospital in Riyadh. Thank you, Dr. Shaya, for the nice introduction, and thank you for the organizing committee for the invitation and this chance to present this case. So I really have, I will present first of all 2 cases. It's really challenging. So thank you for your attendance and to staying late to the end of the day. Where is Prof. Majdi Idris? I'm supporting Real Madrid, so please don't leave the room. I have really 2 interesting cases for you. So the first case, I chose the title of, Is It Just a Cough? So my patient is a 53-year-old gentleman who was referred to the clinic as a case of chronic dry cough for 8 weeks. He presented to the clinic back in June 2021 when he suffered from mainly dry cough for 8 weeks. That is non-progressive and intermittent. You know, the most important thing in cough is the history. So he denied everything. So no diurnal variation, his cough not affected by position, supine or sitting, no difference during the weekdays or weekends, and his cough mainly during the daytime rather than the morning or night. That cough actually was associated with mild exertional dyspnea. Having said that, he was able to walk 3 kilometers and he was able to climb multiple flights of stairs. He got a history of TB exposure back in 2017 when his maid had an active pulmonary TB that was later treated. His BBD was positive according to him, but he didn't take any prophylaxis. 2 AFBs in 2017 and 19 were negative, and 3 of his sons had positive BBDs, and they took prophylaxis. He reported night sweats for 8 months and unintentional weight loss of 4 kgs. No fever. He denied any fever, syncope, lower limb edema. He was not on ACE inhibitors. No symptoms suggestive of GERD, postnasal drip, or sinusitis. and other systemic review were unremarkable. So he had an ischemic heart disease history with, for that he underwent PCI back in 2021. His known dyslipidemia, no surgical history, not known to have any allergies, no pets at home, and he's working an office job, mainly in the housing management. He's an ex-smoker with 10-pack year smoking history. These are his medications. He's on aspirin, ticagrelor, Losec, atorvastatin, ezetimibe, and bisoprolol. So, upon examination, his vitals were okay. He was satting 95% in room air. Apart from this, his ENT, cardiovascular, respiratory exam, and GI exam all were unremarkable. So, in summary, we have a 53-year-old gentleman presented with a history of chronic dry cough, weight loss, with no other significant symptoms. He underwent a bunch of investigations and imaging, so the routine labs were unremarkable. His proBNP was 210, LFTs were normal. His CRB and ESR inflammatory markers were normal, and quantiferon was negative, and the IgE level was negative. So this is his chest X-ray, the first chest X-ray. And as you can see, apart from mild diffuse bilateral interstitial thickening, the lungs are clear. This is his PFT, which showed obstructive pattern with positive reversibility and normal DLCO. The first echocardiogram was back in May 2021. The left side was normal, and the right side, the RV, was normal in size and function. And the Doppler findings didn't suggest the presence of pulmonary hypertension. So the first CT scan, it showed, as you can see, faint ground glass opacities. These actually was associated with tree-in-bud appearance, so the provisional diagnosis was hinting toward an infectious inflammatory process. His microbiology were negative, including 3 samples of AFP, sputum culture, Legionella, rapid multiplex respiratory viral screen, including pertussis, were negative, and repeated inflammatory markers were negative. So during his follow-up, he went to the family medicine and ER several times. He received multiple courses of antibiotics, steroids, inhalers, without significant improvement, and his cough had worsened to the point that he cannot finish a sentence. August 2021, he came again. By the way, all his investigations and workup was with the general pulmonary, not with the PH clinic. So he went to the pulmonary clinic, and his cough actually was persistent and worsening. It's affecting his daily activities and his sleep pattern. Therefore, the decision was to admit him and do further evaluation. ENT exam was unremarkable, including CT sinuses and otoscopy. And during the admission, his inhalers were upgraded and singular added with no improvement. Ticagrelor was changed to Blavix, as there were some reports of cases of cough with ticagrelor, and bronchoscopy was attempted. However, it was aborted due to desaturation during the procedure. He had a D-dimer, and his CTBE study was done. It showed no acute PE. And as you can see, there is, again, ground glass opacities, but it's more worse now. And here, for the first time, we can see smooth septal thickening, suggesting of edema. And during this CT, we started to see enlargement of the right side of the heart. So for that reason, he underwent another echo. So his echo in August, he started to have a reduction in the RV function. And as you can see, his RVSP now is 77. And if you remember his echo back in May, so 3 months prior, it was totally normal. So again, part of his workup, the routine care sets of pulmonary hypertension, his autoimmune panel were negative. Viral screen, including hepatitis and HIV, was negative. And for that, he was started on IV Lasix and showed improvement in his shortness of breath and oxygenation. And actually, his oxygenation went back to normal, so he was in room air. However, his cough persisted. So, we did a PET scan just to look for any hidden malignancy. The PET actually showed ground glass opacity and with prominent right paratracheal lymph node. And again, it's hinting toward an infectious process. However, no neoplastic process. So again, the patient presented to the pulmonary clinic with worsening symptoms. He was on a wheelchair, oxygen connected to him with significant worsening of his respiratory symptoms, a repeated CT scan. As you can see, there is progression of the size of a pulmonary artery. It's more dilated now. And for the first time, they reported this CT, as you can see, increase in the interlocular septal thickening with focal ground glass opacities. And again, in this report, they reported a significant enlargement of the mediastinal, hilar, and lymph nodes. So they give us a provisional diagnosis of PVOD, pulmonary veno-occlusive disease. So in September, he underwent a right heart cath. As you can see, his mean RA pressure was 9, very severe mean pulmonary artery pressure of 68 pre-capillary. His cardiac index was 1.8, output 3.2, and PVR 16.4 Wood unit. So the patient still has had persistently coughing. He was functional class 4 at the time. He was on 2 liters of oxygen. So our thought actually, PVOD, it's less likely to present like this acute presentation. Having said that, he was started on monotherapy cautiously with close observation, and his ProBNP dropped down in 1 week from 3,000 range down to 300. However, again, no improvement in his cough. So what to do next? We did MDT meeting. To be honest, there was no clear consensus about the diagnosis, and we raised the question that does the patient had an exposure that led to accelerated ILD with secondary pulmonary hypertension? It's a possibility. So the decision was to start him on pulse steroid, 500 for 3 days. Then he was continued on 60 milligram. The repeated echo actually after the monotherapy and the steroid showed improvement in the right side function, and his RVSP now 30 to 40. In the previous echo was 77. And then he presented to the clinic after the pulse steroid. He got again progression of his symptoms, mainly the cough. He was on tapering dose of steroid till off. And for the first time, he reported a neck swelling. So CT neck was arranged, and the CT neck basically showed enlarged lymph nodes in the triangle of the neck and the deep neck spaces and in the superior mediastinum. And as you can see, we have a lymph nodes, lymphadenopathy with some lung changes. Again, the thought process was towards infectious inflammatory process, mainly pneumonitis. So who would perform a lymph node biopsy? Raise hands. Okay, 50%. Okay, so he underwent a lymph node biopsy, and unfortunately, it was reported as metastatic adenocarcinoma consistent with pancreatic origin. As a follow-up, he was, of course, electively admitted. The cuff didn't improve. He was functional class 3, oxygen dependent on 2 liters. And we did part of the malignancy screening. We did the CT abdomen, which showed focal area of enhancement in the pancreas. However, no definitive pancreatic lesions, no abdominal metastatic disease. However, he got persistent mediastinal hilar axillary lymph nodes enlargement. And again, it showed the CT multifocal pulmonary disease process. And on the CT, unfortunately, it showed sclerotic metastatic disease, multiple. Followed by MRI. The MRI actually confirmed the diagnosis of pancreatic neck mass. And he was, at the time, complaining of back pain. So again, the MRI showed, unfortunately, a widespread osseous metastasis. In December, he got bicytopenia, so a bone marrow biopsy was done, and it showed infiltration by the malignancy. So, any hint about the diagnosis? So, we diagnosed him as a metastatic pancreatic cancer with pulmonary tumor emboli and lymphangitic carcinomatosis. So back to our patients, he was evaluated by the oncology team, and he was started on a palliative chemotherapy and radiation palliative radiotherapy to the hip and spine. And unfortunately, despite that, he progressed clinically and radiologically, and he was signed DNR, and unfortunately, he passed away soon after. This case was published for your reference by my colleague, Dr. Abdurrahman Harbi and Dr. Muhannad Hawari. That was for the first patient. So let's go for a much happier scenario. So for the second patient, I chose the title of Rapidly Progressive Dyspnea. So we had a 50-year-old lady who's known with breast cancer. She underwent chemotherapy followed by mastectomy, and she was in remission for 5 years. And after 5 years, she started to develop progressive dyspnea on exertion. So she presented again to the general pulmonary clinic with progressive dyspnea for 2 months. And actually, she correlated this dyspnea to the recent H1N1 pneumonia that she had 3 months ago. She denied any previous radiation therapy or steroid use. In the clinic, she presented on a wheelchair, and she was sitting 93% in room air, and her chest exam was unremarkable. So her initial CT scan, again, it showed a new focal ground glass obesity. So we labeled this as a consequences of her H1N1 pneumonia. We did an echocardiogram, which was unremarkable with no evidence of pulmonary hypertension. This is her CT scan. As you can see, as a follow-up, she got a new focal ground glass densities, central and subpleural, and again, it was hinting toward organizing pneumonia, a post-viral infection. She had a PFT, which showed moderate restrictive pattern, and given her persistent dyspnea post-H1N1, the final diagnosis was a post-viral organizing pneumonia, and she was started on prednisone 30 milligram. 2 weeks later, her symptoms worsened, unfortunately. She suffered from severe functional limitations, In the clinic, actually, she walked in the corridor and her saturation dropped down to 77%. So she was admitted for electively for further workup. And because of the rapid deterioration, she underwent a CTPE study. The CTPE actually showed, again, peribronchivascular obesities, faint ground glass densities, however, no PE. And in the CT, they pointed toward that she had enlargement of the right side of the heart. So her labs were unremarkable except for elevated LFTs that reached up to triple the upper normal of normal limits. So she underwent ultrasound of the liver, and it showed complete thrombosis of the portal vein and its intrahepatic branches. Her viral and autoimmune hepatitis were negative. Thrombophilia and lupus antibodies were negative. So she was started on therapeutic anticoagulation. The repeated echocardiogram, now she has evidence of pulmonary hypertension. Her RVSP 40 to 50 millimeter mercury, however, with normal size and function of the RV. Part of pH investigations, she underwent a VQ scan, which showed multiple bilateral mismatch perfusion defects, raising the high probability of pulmonary embolism. So the differential diagnoses in our mind that during the admission, she got worsening respiratory status and she requiring high flow nasal cannula now. So as I mentioned, because of the impression of post-viral organizing pneumonia, she was started on a steroid with higher doses of methylbrid up to 50 milligram daily. And we did a right heart cath, and at that time they involved the pulmonary hypertension team, which showed a mean pulmonary artery pressure of 25, RA pressure of 4, which is 5, and PVR by FIC was 2.9, normal cardiac index of 3.8, and cardiac output of 6.7, and no significant shunt with a QP/QS of 1.1. During the right heart cath, and I believe the case was evaluated by Dr. Noura and Dr. Dal'an, they requested for a wedge sample. Luckily, they asked our cardiologist, Dr. Masoud, to do a wedge sample during the right heart cath. And although it's expected to have a low diagnostic yield, however, it was done and sent for analysis. And the cytology, again, it showed malignant cells consistent with metastatic breast cancer. And the markers, it goes with breast in origin. So after that, we did a PET scan, which showed enlargement and avidity in the lymph nodes, supraclavicular, mediastinal, and hilar lymph nodes. She underwent FNA of the right supraclavicular lymph nodes, and that's actually confirmed the diagnosis of metastatic breast cancer. So given the right heart cath findings, she was started on monotherapy, sildenafil, and she was started on Lasix, and oncology team were involved. And in the view of precapillary pulmonary hypertension and the picture of similar picture of CTIF by the VQ scan and the blood sample, the wedge blood sample that came positive for metastatic breast cancer. So we confirmed her diagnosis as PTTM. So what is PTTM? Is pulmonary tumor thrombotic microangiopathy. It's first reported by von Erbey in 1990. And the types of tumoral pulmonary hypertension, we have different types. We have the macro embolism that results of proximal tumor PE. We have the microvascular disease, the pulmonary tumor microembolism, and the PTTM. The PTTM actually carries poor prognosis, and it's the aggressive type of tumor emboli. And usually these tumor emboli are really small. It goes to the peripheral pulmonary arteries. It causes activation of the coagulation cascade and fibrocellular intimal proliferation that subsequently results in high pulmonary pressure and PVR. And as you know, now the PTTM is classified under group 5 of pulmonary hypertension. As I mentioned, different involvement of the tumor cells in the pulmonary vasculature. It can go to the pulmonary artery, like proximal emboli. It causes lymphangitic carcinomatosis. It's very common. Patients with malignancy, and pure microvascular disease, or combination of these. So a high percentage, looking in the literature, a high percentage of cases were reported in autopsy, unfortunately, and the most common malignancies, it's the large cell carcinoma and adenocarcinoma that associated with tumor emboli, and it's higher in the gastric, lung, breast, and ovary, kidney, and liver tumor sites. The prevalence is 3 to 26%. It carries, as I mentioned, poor prognosis. The most common symptoms is dyspnea, 71%, cough around 8%, and as I mentioned, it's involved the hepatocellular, it's associated with the hepatocellular carcinoma in 29% and breast cancer in 18%.
Dr. Faiz, 1 minute.
Okay. So, as I mentioned, it's really difficult to get a diagnosis from alive patients. It's by the wedge sample, and most of the cases they diagnosed very late when the patients already have metastasis. And this is the sample from the wedge position. The pathophysiology is similar to group 1 PH, however, no plexiform lesions. And as I mentioned, usually the CTPE is negative. You have positive VQ scan and evidence of pulmonary hypertension by cath. And unfortunately, most of the patients, they present late. We don't have definitive treatment for PTTM. Imatinib and bevacizumab, it's studied, and anticoagulation is considered not of value in such cases, so it's not recommended. So back to our patient. Our patient, she received trastuzumab, so she went off from high nasal cannula to nasal cannula, and then they added another chemotherapy, bevacizumab. She was starting well after the bevacizumab, and she was with no oxygen supplementation. She was on sildenafil and Lasix, and her repeated right heart cath 4 months later showed normalization of the mean pulmonary artery pressure and PVR. As I mentioned, it's a rare disease. It remains underdiagnosed, and you have to have a high suspicion to diagnose it, and timely diagnosis is essential for treatment, as it's prevent the mortality. And by this, I conclude. Thank you.
Thank you, Dr. Faiz. If you can stay there, please. We'll give like one question. It seems to be King Faisal Specialist Hospital specialized in pulmonary tumor microangiopathies.
Yeah.
So if you have one question, for the sake of time, we'll take only one question, then we'll go to the next case. Any questions? Okay, I have a question for you.
Yeah.
So I will go to the first case. So the first case sadly died because of pancreatic cancer. We know that pancreatic cancer have hereditary diseases. However, from the beginning, there is some ground glass opacities. What about the VQ scan? Do you have like a VQ scan can tells you about like similar to CTEPH?
Exactly.
Or let's say DLCO goes down to like less than 40 or something like that. Can give you a hint about something else going on?
His VQ scan, again, it was positive for peripheral disease, but the CTPE study was negative. The DLCO was normal. So, and to be honest, initially we didn't have any clue for his diagnosis. We thought about ILD. Then he developed this neck mass. Then we have a hint, and we did a biopsy, and it came positive. But apart from this, during the investigation, we didn't find anything.
Interesting. Yeah. Okay, thank you, Dr. Faiz. Thank you so much. We'll go to the next case.
Great, thank you very much.
Okay, thank you.
It's a great pleasure now to introduce our next speaker, who needs no introduction, Dr. Hani Sabor, who's a cardiologist and electrophysiologist in Abu Dhabi. And he is going to also start with a presentation that involves cough. So today is the day of cough. So it's cough and severe ILD, a case of an unexplained progressive dyspnea.
The organizing committee for inviting me. This is really the most outstanding meeting we have here, literally in the Middle East. The level is so impressive, and it's better every year, which is even more amazing how you can better yourself when it was outstanding last year. I'm very proud to be part of this program. So the objective of this case is to talk about a patient. This patient, he's actually a senior professor of medicine at the university. I did take his permission to disclose some of this information because it could identify him. And he has a chronic cough and severe ILD, unexplained dyspnea, hidden within ILD. So this is the QR code. If you want a copy of the slides, I'll be more than happy to provide them to you. So he's a 67-year-old male professor of medicine at the University Teaching Hospital. He had a history of COVID in April 2020 with persistent cough, treated with steroids for ILD, not improving. Recent pulmonary evaluation, nintedanib was added. He's an ex-smoker, he has mild hyperlipidemia, he has a little bit of osteoarthritis, but was never tested for connective tissue disease. Ex-tobacco smoker, more than 5 years, no anorectic drugs, and on steroids, 15 milligrams of prednisolone when I saw him, nintedanib, and Lasix was started 2 days before the cardiology clinic visit. So the problem really is not just the cough, it's the worsening dyspnea for 1.5 years, 3 weeks of rapid heart rate and palpitations, lower limb edema for about a week, and near syncope. At this point, he saw a cardiologist. So the key problem, new symptom, is palpitations and concern about edema, but what about the progressive dyspnea 1.5 years, which was remarkably assumed by many specialists to be just due to the ILD itself. So the 2-year history, frequent exacerbations and progressive deterioration. This is literally written in the notes. 47 courses of steroids were given to this professor since 27. Mycophenolate was started in June 2025. And the main problem is worsening dyspnea, exertional limitation, dizziness, and syncope. He also noted that his resting heart rate was increasing with palpitations at rest. So I don't know if you can see this. This is a screenshot from our EMR. They basically describe organizing pneumonia versus eosinophilic pneumonia. Started on prednisone. This is the very first note I could find. And this is the progressive worsening of his DLCO. Worse, worse, worse. And unfortunately, and this is the most telling problem, is this echo. This terrible echo tricked everybody. April 2024. What does it say? No pulmonary hypertension, good systolic RV function, and of course, a cursory echo is a lethal error, and it misled everybody in this unfortunate case. So, he's functional class 3, he has positive JVD and lower limb edema, he's losing weight progressively, he's a tall man, blood pressure was on the low side, and his heart rate was 120 beats per minute at rest with a saturation of 92%. This is on Lasix. Now this is his x-ray. Are there some clues about a cardiopulmonary presentation more than just the lung? So you can clearly see some chronic changes, but there's also something wrong here, which I'm going to highlight in a bit. There's cardiomegaly, the lungs are basically oligemic, and the pulmonary arteries show sort of a 3 sign, which I'll highlight in a minute. Now this is his ECG when he came to see me. So he actually already had a murmur, and the ST depressions across the anterior leads are not ischemia. This is right ventricular hypertrophy with RV, not LV strain. So it's incomplete right bundle branch block, right axis deviation, and RV hypertrophy. So the tachycardia only triggered a cardiac evaluation after 1.5 years of symptoms. He was seen by a cardiology professor in the same teaching hospital. That's the ECG. An echo was done the same day, and of course there was severe pulmonary hypertension at this point, pericardial effusion and RVSP over 100. I got an urgent phone call, and I saw him the next morning at our center, and I immediately admitted him to the ICU. He was in frank right ventricular failure for IV milrinone and IV furosemide for 24 hours, and we did the next day the right heart cath, and we initiated treatment. So this is the actual echo. This is a properly done echo, and you see the right ventricle and the right atrium are collapsing the left ventricle. The peak velocity is about 7 or 8, so about 123 millimeters of mercury, and a hugely dilated right atrium. There's also reduced right ventricular function. TAPSE is low at 14 with an S wave of 9. Pericardial effusion. We did the bubble echo. There's no evidence of right to left shunt. You can see here what's also dilated is the pulmonary artery. So this is the kind of patient that needed to be fast-tracked. So fortunately, he was fast-tracked by the cardiologist. Worsening symptoms, functional oxygen 3 or 4, signs of RV failure, hypotension and tachycardia, and that's really the fastest way to approach the patient. So here's the right heart cath. So the mean PAP is 67, 118 over 36, cardiac index of 1.65, 13 Woods units. And as a routine, we do nitric oxide in every patient. There's really a minimal or non-respondence fact. So just so you can believe me, this is actually his cardiac index, 1.83. So we do both FIC and thermal, and this is it. And you see the RV pressure here, how incredibly high it is. So the NT-proBNP the day we saw him was 1,800, and now a more detailed evaluation looking at the anti-nuclear antibodies, so SCL70 plus SSA and SSB. Pulmonary function test was 47%. Now, remarkably, he was able to walk, but desaturated quite a bit during that and had to stop. And in fact, he was telling me he was basically planning to retire and stop working. So, of course, this is a CT angiogram. You see how dilated the pulmonary artery is, massively dilated and pancaked left ventricle and pericardial effusion. VQ scan is unimpressive. His IVC is also dilated, so there's really right ventricular failure. We didn't do genetic testing. So if you do the standard risk assessment, this guy is high risk of 1-year mortality. And as I mentioned, there was a 1.5 years of preceding symptoms and imaging focused mostly on the lung, not the heart. So when you look at the high resolution CT, and I think this is most telling, does not appear very extensive, does not explain the severe progressive symptoms. But we found this report from June. It again shows ground glass densities. And what they mentioned, they compared it to a previous CT scan, stationary disease course. So if it's stationary on CT, why is the patient progressively worsening? This should have been a clue to everybody. And he's a professor in the university with a lot of friends. So this is the July 2025 high resolution CT. Now this is my notation. You see how big the pulmonary artery is? You can't miss this, even on a non-contrast high resolution CT. And again, there's ground glass opacification and some scarring, but it's really not telling us the whole story. This is 43 millimeters end-to-end on the high-resolution CT. So to summarize, a 67-year-old, very senior physician who taught basically half the country, physicians, misdiagnosed as only ILD for 1.5 years in spite of actually a second opinion. So what's really interesting, he got a remote visit in a UK center of ILD excellence, but they never saw him. They just saw some of his images. And it's an ILD center, not a PH center. So this is the disintegration and fractured nature of care we have in healthcare. So reality is he has CTD, PH, ILD, with lung changes maybe 25%, maybe more, maybe less. Very low progressive DLCO. And of course, non-vasoreactive, severe hemodynamics, high mortality risk, with severely limiting symptoms and a 6-minute walk of 163 with hypoxia. So we do use combination therapy in these patients. This is the old study that we found, which is the Triumph study. Remember, this was in patients with group 1, not group 3. So we sort of extrapolated some of the data. But those patients who were given inhaled treprostinil benefit a lot more than patients who started even 12 weeks later. And this is a high-risk patient. So what we did was upfront triple therapy with inhaled treprostinil and endothelial antagonist tadalafil. He was given portable oxygen, Lasix-40, spironolactone, and we started on the treprostinil 4 times daily at 3 puffs with gradual escalation. So he did have some side effects, including headache, GI, and bone pain, which we managed with multiple things like analgesics and antiemetics. The cough was due to treprostinil inhaler. We had to manage that. Oral steroids now were initially a slow taper, and of course he has GI side effects from nintedanib, and there's a discussion ongoing. So follow-up in November. He continues on the triple therapy. We're now down titrating the furosemide to 3 times weekly. His BNP was 1,800, it's come down to 321. The 6-minute walk distance extremely increased, 423 with a saturation of 94%. And reverse remodeling now, the SPAP, which was 124, is about 62. And he's now in the intermediate risk zone with functional class 2. He's now canceled his retirement plans and is off ambulatory oxygen. So this is his prior to triple therapy with inhaled, and this is his most recent echo in November. You'll see there's a significant improvement, although the right ventricle has still remodeled at 62 millimeters of mercury. Now, as part of this detective work, he actually found that old echo from April 2026. He brought it to us on some images and a CD, and you can clearly see this is the dilated right atrium. This is not a normal RV function, not a normal RV size, and even if they relied on his TR velocity, this was a mistake. This was also his old ECG from 2024, which was retrieved. These T-wave inversions, which became much more prominent, were still there. This is, again, not non-specific T-wave inversion, but again, the cardiologist reading this in a busy ECG clinic wasn't really focused on his symptoms. So again, this is the missed clue. See his left atrium, here's his right atrium, clearly dilated and not normal. So all of the clues were there, including the D-shaped septum, but was missed because they completely depended on the RVSP only and the TR velocity. I think this is a very bad error in this patient. So I wanted to show you this data because we did look a little bit about ILD pH and group 3 pH in our registry. This is my friend Khalid Saleh. Unfortunately, he couldn't come here. He's the first author on this paper. And we have a significant number of patients in our registry who were followed for up to 6-7 years with group 3 pulmonary hypertension. Now, group 3, due to lung disease, of course, these patients are going to have dyspnea and hypoxia. This is obvious. And there are many causes of group 3, but many lung diseases can coexist with pulmonary hypertension and significantly increase mortality. And this is the bottom line. So the definition, pH due to chronic lung disease, is having a severe pH with a high pulmonary vascular resistance. And in fact, the high pulmonary vascular resistance itself is the marker of severe mortality. But if you don't do the right heart cath, you're never going to pick this up. And hemodynamic decompensation is clearly fatal. So there's many different patients that you see with different extents and severities of the lung disease. Unfortunately, is the dyspnea due to the lung disease or a high PVR or a little bit of both? So it's really an assumption that we have to be careful about. Don't assume that the only reason for dyspnea is the lung involvement, because the lethal overlap with pH is a much more serious complication. So these are some simple examples of extent and severity in patients who have different degrees of disease. Now, there's clearly pulmonary vascular involvement in different ILD pathologies, and when they looked at biopsy specimens and from post-transplant patients, the remodeling in the blood vessels in the patients with ILD parallels the progressive fibrotic nature. So always think in the background, the fibrosis in the interstitium can clearly affect the vessels. And these are diseases in whom it is more likely to have pulmonary hypertension, particularly patients with scleroderma, mixed connective tissue disease, other forms of pulmonary vascular disease, including PE and CTEPH. So think about the overlap, because what is common is ILD, but what is lethal is PH-ILD. Now, this is the severity and extent of connective tissue disease in relationship to mortality and morbidity. So patient's survival also depends on the degree of the disease on the CT, but there's another component as well, and that is scarring has greater survival, but is it also due to increased coexistence of pulmonary hypertension? So in the 2 studies that looked at RV dysfunction in patients with lung disease, RV dysfunction is generally the cause of progression and death, but it is RV dysfunction that we are trying to prevent. So in the increased study, I'm sorry it's very small, patients with very high PVR, more than 4 Woods units, are the ones that had the greatest benefit. And I think that's the key point. We have to look for the vascular component and treat it in order to save the right ventricle. The objective is to save the right ventricle, which is increasingly the more lethal issue. So this is a paper that we published with the same group, and Khalid again is the co-author, is the first author. We looked at vasodilator therapy in patients with pulmonary arterial hypertension, connective tissue disease, and lung disease. So this is the triple extravaganza, if you would. And how this is complicated, because in these cases sometimes we're not sure. Is it group 1, is it group 3, or a combination? but we did look at the patients on treatment. And the investigation was basically to look at patients with mild to moderate fibrosis, subgroup A, less than 30%, or subgroup B, more than 30%, how did they respond to vasodilator therapy? So if you look at the two groups, A and B, there's really no significant difference between them except maybe the DLCO percentage, which you would expect, that makes sense. But the PVR was elevated in both groups. The cardiac index was within this range, 2.8 and 2.5, and the saturation was not really different. So with ICE, ILD, CTD, and PH, and the vascular phenotype, we found worse hemodynamics and higher NT-proBNP than our primary group, but both had improvement of the mean pulmonary artery pressure and PVR on combination therapy. There was no statistical difference between both 6-minute walk distance, NT-proBNP, cardiac index, and median follow-up was about 12 to 20 months. So both groups improved hemodynamically without worsening from a lung disease or hypoxia standpoint. So when you come back to how the guidelines position this, because it is complicated, right? You have patients where their pulmonary vascular disease is predominant, and you have patients with chronic lung disease is predominant, and you have patients in the middle, and it's physiological as well as morphological changes. When the classification is unclear, the recommendation is to refer to expert PH and ILD center, and if severe with high PVR, individualized therapy can be reasonably tested. I think I've just highlighted this because this is something that's really important. These people need expertise on both PH and ILD and an MDT for individual decision making. So PH-ILD is a progressive disease. Clearly, at the time of initial diagnosis and basically within 8.6 months, 38 had PH at baseline, 86% developed worsening pulmonary hypertension at follow-up. So if it's not there this year, maybe it's there next year. And when the patients are progressing, like our patient, you need to have this really clear in the differential diagnosis. So the determinants of poor prognosis are a low DLCO, a saturation less than 88% during 6-minute walk increases death by 4-fold, an SPAF of 36 versus an SPAF of 56, progressive disease has a 3 times hazard ratio, high pulmonary vascular resistance, and high mean pulmonary artery pressure. So all 3 of these are hemodynamic parameters, and you're never going to order a right heart cath unless you think of this or get the echo. And of course, high resolution CT findings as well. Clearly, you have to save and recover the right ventricle. These patients do respond to vasodilator therapy, and we actually have the PVRI paper that published recently with the group in Gießen, where we saw this number of patients on treatment, and their survival is different depending on the presence of RV dysfunction. You really have to look at the RV as determinant for risk. So if there's moderate to severe PH on right heart cath, PAH risk factors present, for example, the presence of connective disease biomarkers, then PH therapy may be considered at expert centers. And of course, we have to consider transplantation in these patients. Finally, in the last couple of minutes, why is PH missed in the setting of ILD? This is obviously a university professor seen by a lot of experts. This should have been picked up fairly early. So when to suspect PH in ILD patient avoiding a lethal oversight? So these patients, when you think about it, why do they need supplemental oxygen? Everybody assumes it's infection or progression of disease. Why are they having more acute exacerbations? Maybe it's pulmonary vascular. Why are they having decreased exercise capacity and palpitations? And of course, the prognosis is significantly worse. So these are progression, but don't assume it's all due to the lungs. And of course, if they have PH, their survival is a lot more miserable than patients without, in the red line. So if you detect it, you're going to find a very valuable thing in saving these patients' lives. So this paper was a consensus paper published in CHEST, looking at how to do screening and what are the highest risk groups of patients. So first of all, look at some clues. Even on the high-resolution CT, you see this patient has a 3 sign, an oligemic lungs. On this high-resolution CT, non-contrast, where is the septum? It's right in the middle. And clearly, if this is not being reported to the radiologist or asked to the radiologist, what does the RV look like? It's not a contrast study. They're not going to routinely order it. So don't miss these common imaging clues which are not properly interpreted or flagged, i.e. you have to look at these scans yourself, not just rely on the reports. So here is a patient with IPF on an antifibrotic, slow decline. The TLC is dropping from 58% from a year ago. This is the CT scan, but what's missing? What's missing is this. We didn't do a right heart cath. If you don't do a right heart cath, PVR more than 5 Woods units, the prognosis is vastly different in this patient who is on antifibrotic and standard of care. So see these clues, and this was also derived from that paper. Some high-risk symptoms, especially syncope and edema, are very important. Syncope increases the hazard ratio 3 times. Jugular venous distension, edema of any kind, altered heart sounds, dizziness and palpitations, as was triggered in our patients. Cardiopulmonary symptoms and physical examination can't be ignored in the ILD patient. Next, red flags on non-contrast CT have a huge impact on your likelihood of diagnosis. So the presence of right ventricular enlargement, a dilated pulmonary artery, or an aorta ratio of more than 1, and all of these things can actually be seen on a non-contrast CT if it's done properly. So ask the radiologist to look and measure. Now this is really where they come back to sort of a conclusion of things that can be used in the clinic. A drop in the DLCO more than 15%. DLCO less than 40% at any stage. Worsening ratio where the FVC is generally preserved, so it's more vascular than interstitial. Worsening need for oxygenation, especially on a 6-minute walk, and desaturation disproportional to what is called the static ILD severity. And on a CT scan, again, any big ventricle. Now, what's also useful if you use BNP liberally. Yes, the patient has dyspnea due to lung disease. Doctor, he's on oxygen. Why should I get a BNP? Because it's going to give you a clue of something that's hiding. Next, get a proper echo. A proper echo with focus on the RV, not the LV. Tell your cardiologist, I need to see the RV in detail, not just the TR velocity. You have to remind them they're stuck on this TR velocity story and it's lethal. So high probability findings of a dilated RV, a big RA, and higher pressures. So this is really simple, but sometimes a busy echocardiology lab, they're going to read 100 echoes. They're going to blast through it and they won't really look at this point. And finally, if the suspicion is high, refer to right heart cath. This is a scoring system coming from this paper. It's very nice. A poor 6-minute walk distance, physical examination findings, but these are higher value. DLCO less than 40, worsening need for supplemental oxygen, cardiac symptoms, and PA enlargement. You can easily get the score clinically, but if you don't have to remember the score, just think of that. Big chambers, worsening hypoxia, same ILD patient, looks static, maybe think about it. I'd like to thank my colleagues who helped me prepare this presentation. Dr. Khalid, of course, with his work on the research itself, and my 2 research fellows who actually helped save this presentation and make it available today. Thank you.
Thank you so much for that presentation and tour de force. I think it was the term used yesterday. I'd use it again today. Any questions from the floor? I'm just looking down there. Maybe if anyone is getting bravery before, I just throw into anyway. But this gentleman, I mean, congratulations on really saving him from cardiogenic shock, essentially, when you saw him at the end. His response to treatment is really dramatic, you know, and actually probably even more dramatic than you would ever anticipated when you saw him in that particular circumstance. And I'm intrigued by the addition of inhaled treprostinil in that setting, because not everybody has the potential to do that.
Sure.
Do you see a situation in the future where you roll on and that he'll get intravenous therapy, or would you consider him also for sotatercept, etc.? Do you see him going down the other pathway, given how responsive he is to the treatments he gave?
Sure. So obviously the rapid response, as we've always heard, including from yourself, the first visit is really the determinant visit. So this guy has changed his trajectory completely. As a matter of fact, he traveled back to the UK to visit with his family. He wasn't on oxygen. So we are watching him carefully because by definition, he's still intermediate. You know, his BNP is still hanging around those thresholds we don't quite accept. And anticipating that we increase the treprostinil utilization a little bit more, but clearly, this is and of course, there's another component. Now the rheumatologist is getting involved and they're talking about additional therapy for the underlying Sjogren scleroderma spectrum. So I am very, very, happy about his progress, but again, the intermediate risk is not something we accept lightly. I mean, there's a lot of left on the table here and there's a lot of remodeling that has happened. So I am planning to reassess him again with a right heart cath, actually very soon, probably when he comes back from the UK.
I might just, in the interest of time then, thank you very much, Hani, for that. Fantastic. We look forward to hearing again about his progress, and I'm going to hand you over.
Well, now we move on to the last speaker, Dr. Ragda Arif, who's going to talk about multifactorial hemoptysis.
Assalamu alaikum warahmatullahi wabarakatuh. Masā al-khair. Thank you so much, everyone, for staying late with us. I know it's becoming difficult at the end of the day. So I will present a case today that we share the care of the patient between King Abdulaziz University Hospital and King Faisal Specialized Research Hospital. So first, I would like to thank Professor Abdullah Dal'an and the organizing committee, the scientific committee, for always giving me this great opportunity to speak on the SAPH. It's my great pleasure to be here and to speak with the PH experts. So I'm going to talk about a multifactorial hemoptysis case, a case unrevealed. So this is a 29-year-old gentleman. This patient, he came to our emergency department as a case of antiphospholipid syndrome. So he had a previous diagnosis of ABS that was made in 2013. Based on some positive antibodies, and at that time he was started on warfarin, and of course he had a multiple venous thromboembolic disease events. He was started on hydroxychloroquine. There was a provisional diagnosis that he might actually have lupus at that time. And also he was started in Imuran. However, he stopped all of his medicine and lost the follow-up by 2017. His first episode of a venous thromboembolic disease was a DVT that he got in 2010. And at that time, he was anticoagulated for a short period, only 6 months, thinking that this is just something provoked or acute. And then he had another episode on 2013, and at that time he had a DVT, received anticoagulant, and at that time he received the diagnosis of the APS. Then he lost the follow-up, and he had a third episode in 2022 with a bilateral pulmonary embolism, started in warfarin, and again, he was not compliant with that warfarin. So he landed in our emergency department in Jeddah, King Abdulaziz University Hospital with the progressive symptoms of shortness of breath that started about 2 weeks ago from the presentation. He progressed from WHO functional class 3 to 4, and he had so much hemoptysis, medium volume hemoptysis, at least for the last few weeks, and it was increasing in amount. He was having palpitation, pleuritic chest pain. He was having exertional dyspnea and some pre-syncopal episodes. So he didn't have any infectious symptoms at the time, no constitutional symptoms. He did not have any symptom to suggest any connective tissue disease because of this diagnosis of the lupus. He did not have any feature to suggest any chronic lung disease or chronic liver disease. He was not a smoker. He worked as a salesman. He didn't have any family history of CTD. He did not have any risk factor for HIV, and he did not have any symptoms or previous surgery or any symptoms of congenital heart disease. So we did the whole history taking by the help of our residents, and then this is his initial presentation examination. So he was hypoxic. He was satting 94.5 liters of oxygen. He was coherent and he was fully conscious. However, he was clubbed and he had mild cyanosis and he has no stigmata of chronic liver disease or autoimmune disease. This is his initial investigation. So we can see he has severe thrombocytopenia. So he had platelets of 5,000. His kidney function was mildly elevated. His INR was therapeutic 2.8, and he has a proBNP of 3,600, so sky high. The rest of the investigation were fine. This is his initial chest X-ray. So we can see there was a sign of for cardiomegaly, enlarged right cardiac chambers. There was no features of pleural effusion or pulmonary congestion at the time, and there was some pruning of the peripheral vessels. And because he presented very hypoxic, we decided to send the patient in the emergency department for a CTPA to be assessed for pulmonary embolism. So this is the CT scan. I hope this works. So basically, he had demonstrated bilateral, what we call mosaic attenuation that we can see with patients with pulmonary vascular problems or severe pulmonary hypertension or SAPH. And he did not have any other feature to suggest any interstitial lung disease, fibrosis, scarring, a bit of atelectasis in the left lower lobe. So this is the soft tissue. So he had multiple bilateral filling defects that was seen. There was significant filling defect in the right main pulmonary artery and also on the segmental and the subsegmental branches. They were actually also concerned about a left pulmonary artery segmental and subsegmental filling defects. You can see the mass is coming. There is an infiltrating mass was seen in the right hilum, and this mass was large, causing obstruction to the pulmonary vascularity. So at that time, the radiologist called this a mass in the right pulmonary, in the right hilum, causing an obstructive effect, and they were not sure what's the nature of this mass. So at this point, we were thinking about the differential diagnosis. Is this patient having sarcoma? Is he having angiosarcoma? Is he having malignant tumor? What's going on with this mass infiltrating the pulmonary vascularity? So he was admitted at the case of multifactorial hemoptysis. Is he a case of SAPH? Is this an acute or subacute PE? Is this an ABS? Is this anticoagulation related? Or does he have an ITP? Also, he had this obstructing soft tissue mass to the pulmonary vascularity, especially in the right side. We were not sure what's going on. And this patient required very long stay in our center, up to 65 days. So, of course, he got an echocardiogram that showed that patient had mild reduction of the LV. However, he has severely radiated right ventricle, severely enlarged right ventricle with signs of severe pulmonary hypertension and dilated right cardiac chamber on the echo, raising the possibility, very high possibility of pulmonary hypertension. So his platelets were very low, 5,000. His autoimmune workup came back negative. And his hepatitis serology came back negative. The blood film showed thrombocytopenia, and the DIC workup was done, and all the septic workup was done, came also negative. So we sent him for the lung ventilation perfusion scan, and this one showed severe reduce in the perfusion of the right upper lung, and also compressive effect on the right lower lobe of the lungs, also reducing the perfusion. So the whole right lung was not perfusing at that time. So I discussed this case with our interventional pulmonologist, and I said, we don't know what's going on. There is a mass, and we know that there is acute and subacute PEs, but please stick a needle and take a biopsy. So he was brave enough and took this patient to the bronchoscopy suite. So he had severe thrombocytopenia that we needed to transfuse him with platelets. We gave him IVIG, and we increased the platelets. So he had a lavage that came back negative, and the mass was biopsy. And he called me saying, I think I biopsied a clot. And I said, what? There is a mass on the CT scan. How come you biopsy a clot? He said, I'm sure this is a clot. And I said, okay, I didn't believe him. Let's go and do a cardiac MR. And we did. So we did the chest cardiac MR. And at that time, the radiologist read our MRI and said that we actually dealing with a chronic large organizing thrombus causing obstruction of the right pulmonary artery. Of course, he had a lot of features suggesting of SAPH and enlarged cardiac chambers. So at this time, we were positive for dealing with a SAPH with a large obstructing mass related to a large organizing thrombus. So the hematology team was involved now because we have a case of SAPH with ABS and thrombocytopenia. And I would like to say thank you to the hematology team. And we have here Dr. Adil Marzougi. He is the leader of the team. And he will present a couple slides giving the view from the hematology regarding the case. So please welcome Dr. Adil on stage.
Thank you very much. It was. We're
going to just get hematology first, I think.
The hematology will speak, and then we will conclude the case.
Thank you, the SAPH committee for this opportunity. Thank you, Dr. Agda, for sharing this wonderful case from our part. I know it's somewhat difficult to deal with. And being the only hematologist here among those sea of pulmonologists, I feel like it's easier and safer to take a deep breath. Thank you all. So from a hematology side, this is actually our absolute nightmare. You have a patient with acute/subacute thrombosis, also with thrombocytopenia. And we find this scenario usually, I would say, not uncommon. And we always scratch our head and say, what can we do for this patient? And especially with a patient that is going to do a lot of procedure like our patient here. So at first, we have to know exactly what's his diagnosis. So we diagnosed him as antiphospholipid syndrome with also secondary thrombocytopenia due to his autoimmune process with his antiphospholipid. And we have two parts of his management. First, we have to deal with the acute event, and later on, we have to deal with the chronic event. So what can we do later on? So at first, acutely, we tried with him IVIG, which is by itself slightly thrombogenic, but we pray for the best, and we gave him IVIG. We started with IVIG just to raise up his platelets count to a point that we can do all kinds of procedure, including cardiac cath, bronchoscopy, or any other procedure that we can help this patient with. And once his platelets reach a certain level, usually we try and we like to start therapeutic anticoagulation where the platelet is around 50. But for such a patient, we can sometimes decrease our threshold for platelets count and we make it around, I would say, above 30 and 40 because it's always immune-related. When you have an immune-related thrombocytopenia, actually the platelets, the number that you see on your CBC, it's actually not the actual number. Actually, those platelets are more functioning and more active and more potent, not just like a bone marrow suppression platelets. So in this patient, we started to resume his therapeutic anticoagulants where we reach around 50, and we said if the count is above 20 and he's not showing any signs of bleeding, we can go with some prophylactic dose or intermediate dose of anticoagulants. In our facility, we like to use enoxaparin, which is the drug of choice in such cases. And later on, we have the issue of we want to maintain this platelets count to a certain degree and certain level. So for such patient, it's very difficult when we have a patient that we are giving him steroids and he received steroids several times before. And unfortunately for our patient, he was steroid refractory. So whenever we are giving him a steroid, his platelets count is not improving. Other lines for such cases is usually thrombogenic. So we always try to find some solution. Even we try to use the old guidelines and using some medications. So in his case, we tried also MFM, mycophenolate in such cases, and without any success. Steroid wasn't successful in his case. So we opted out after careful considerations with the pulmonology team and also after the approval with the patient to use thrombopoietin, such as eltrombopag. The eltrombopag problem is that it is a thrombogenic, known to be a thrombogenic process because it's activate the thrombopoietin, increases the megakaryocytes, and will increase the platelets count, and it might lead to another thrombosis in an acutely thrombotic patient. But fortunately for our patient, we started with a higher dose of eltrombopag, 50 milligrams, and with God willing, his platelets count almost returned to normal, and we kept monitoring its platelet count until we found a proper dosing for him. Unfortunately for our patient, he didn't have any side effects. He didn't have any new thrombosis. His bleeding was controlled. His platelet counts returned to normal. And we ask our pulmonologist to continue his management for his pulmonary hypertension. Thank you all.
يعطيك العافية. Thank you, Adel. So back to the room to the pulmonology management. So we took the patient to the right heart cath. And again, this time I was able to convince another cardiologist, to stick a needle again, and this time to get the right hemodynamic. And he had a severe precapillary pulmonary arterial hypertension with a very high severe BVR with reduction in the cardiac output and the cardiac index with very low mixed venous saturation. So at this stage, clearly we have a diagnosis of ABS, SAPH, secondary ITB, severe thrombocytopenia. The patient was started in Lasix infusion and required ICU admission for inotropes and support, pressor supports. He was actually achieving euvolemic status, and his proBNP, as you can see, was trending down. Because of the severity of the hemodynamic, he was actually started pre-op on Riociguat and Macitentan, and at that time he was also referred to King Faisal Specialized Research Hospital for pulmonary endoarthrectomy as the center of excellence in the country. And of course the case was accepted by Professor Abdullah, and we were just waiting for the surgeon to come to do the surgery. So during that time he was on our floor, and then successfully he was transferred and the surgery was done late January. And I remember Dr. Abdullah texting me saying the patient is doing fine out of the OR, and his immediate numbers were impressive from the Swan-Ganz catheter after the surgery. He had a very smooth recovery, and then he continued to follow up with the team there. And echo repeated after 3 months, and this is the protocol there, and we can see that he's showing significant signs of improvement. He was off treatment, off PH treatment. He was on anticoagulation, switched back to warfarin, doing great. He was on small doses of prednisone, and this is his post-op right heart cath, and you can see the BVR improved from 13 wood unit to 2.5. This is impressive. So he's actually fixed. The patient had a VQ scan, showed complete improvement with only very residual subsegmental mismatch defect, and improvement of all of that obstructive mass was gone by the surgery. And then he was seen by our beautiful doctor, also Dr. Noura at King Faisal yesterday, actually, and the patient is doing great. He is back to work. He is not in any pulmonary hypertension medication. He is on anticoagulation, doing fine. His platelets count is back to almost normal. He has WHO functional class 1, and he was able to walk for more than 500 meters. There was no signs of RV dysfunction, and he had a very minimally loud B2 with very acceptable blood work. This is how his proBNP was trending down from 1,000 to almost less than 100. And so the case illustrated the established well-known risk factor of CTEPH, including younger age, male patient, primary APS, recurrent venous thromboembolic disease, and someone with subtherapeutic INR not adequately anticoagulated. The presence of severe refractory immune thrombocytopenia complicated the picture because of the risk of the bleeding and the risk of the thrombosis, which led to the interfere of or the management plan from our hematology team. Despite these challenges, we believe that this patient needs to go for the surgery as a curable, and this was done, and the patient received the pulmonary endoarthrectomy, and we have seen the impressive result of his right heart cath and clinical improvement. So we would just like to emphasize the early identification of high-risk patient group and early referral to the specialized center, and without the help of the King Faisal team and the KAH team and the collaboration between the two centers, this patient would not make it today. And the multidisciplinary approach, especially in cases with this complexity. With this, I would like to thank you. This is our patient. He gave the permission to put his picture. This is him in our floor. He was sick, hopeless, with recurrent hemoptysis. And mashallah, after he finished the surgery, he's working in jewelry now. And I asked him that we're going to put his picture or photo today, and he said yes, because he wants to be famous. With this, I will conclude my talk. Thank you so much for listening and for your attention.
Thank you very much. It was a very tough case. Well, is there some comments or questions from the audience?
Sorry, thank you for the great case. And I think it was like an emotional roller coaster for you with this case. One last question. At the end, you put his hemodynamics, or the PVR is a bit on the higher side, 2.5, MPAP is 29, is slightly on the higher side. Would you really consider like balloon pulmonary angioplasty for the residual CTEPH, or maybe monotherapy with RESIG? What do you think? Especially that the patient, although he's in a low risk category, but he's still requiring a bit of diuresis to keep his anti-proBNP on the normal side. What do you think about that?
So, as I said, he's following down with King Faisal. I mean, clinically, he is doing great. He's like class 1. His proBNP is less than 100. His echo improved significantly. I think we're following him, and then we will decide later on whether this patient will have a residual pH, whether he will need a balloon pulmonary. I mean, now we are in the multidisciplinary team. We decide about doing multiple procedures together. But for him, so I can see from where he was and where he is now, I think I'm just gonna observe him until there is any clinical deterioration, then I might consider. But I don't know if Prof. Abdullah have different opinion.
No, obviously. But thank you for referring such a very complex case. And in our cohort, we have more than 40% of our CTEPH cohorts are actually antiphospholipid syndrome. Very complex. Of course, wide spectrum. Some of them are as complex as your case, having severe thrombocytopenia and major clots. So you anticoagulate or you don't anticoagulate. So very, very complex situations. And some patients, before we take them to OR, sometimes we had to do actually plasma exchange. Beside all these fancy medication from our hematology colleagues, he had very good. So to answer Fatima's question, there is no clear consensus if when to treat patient with some residual pulmonary hypertension. As Dr. Raghda mentioned, he made a great outcome. So we're following him. But the behavior of antiphospholipid syndrome is actually different than other CTEPH patients. So we expect some of them will progress with time. So that's why we are having a close follow-up with them. We tend not to treat patients if they are completely asymptomatic and having PA pressure, mean pulmonary pressure less than 30, and a normal RV function by by echo, and normal proBNP, normal 6-minute walk test. Now, once their mean pulmonary arterial pressure goes more than 30, or starting having some echo findings, or trending into proBNP, of course, we intervene at that time. Yes. So that he needs a close follow-up. Yes, I think yesterday was his first follow-up with Nora.
Thank you for this great case. Have you looked for HIT, heparin-induced thrombocytopenia? And how is his further course with regard to the platelets? How do you manage this? And third, is he on Coumadin, or have you started to use a DOAC? Thank you.
Okay, so his thrombocytopenia was very challenging since he came. And this led to multiple times to the interruption of the anticoagulation. He actually had a bone marrow biopsy also to look why. And eventually we decided that this is an immune-mediated process. He responded a bit to the steroid in the beginning, and then he became refractory. He was responding a bit to the IVIG, and that especially was given before any procedure, but he will go again into severe thrombocytopenia, sometimes less than 20,000, and we had to stop his anticoagulation. The only agent to work with him was the thrombobiotic receptor agonist, but that was also very challenging because you can push your patient to other side of the spectrum where he can develop thrombotic event. So that was very challenging. And in between, he was having intermittent hemoptysis, where we needed to use tranexamic acid and all of this. So he was very challenging, but we end up controlling his thrombocytopenia and putting him in low molecular weight heparin until he made it to the surgery. And then after the surgery, he was also in low molecular weight heparin, and then the team in Riyadh was able to switch him back to the warfarin. So now he's in warfarin, off medications. He's not in any platelets transfusion or any platelets treatment, and his platelets count, as of Dr. Noor said yesterday, was almost to 200,000. So he's almost at the back to the baseline. But that's, I think, the nature of the antiphospholipid, that the disease now is in remission, and he's in some immunosuppression and some prednisone, but the disease can wake up anytime, and he can develop either thrombotic or he will go back to thrombocytopenia. So we're watching him very closely.
Well said, Doctor. And HIT is negative.
HIT was negative,
yeah.
yes, sorry if that question, HIT was negative. All the DIC and HIT workup were negative.
Well, listen, thank you very much. That was a fantastic presentation. In fact, I'd like to take the opportunity of thanking all of our presenters. And in fact, if you don't mind, presenters, if you might come up for a photo at the end, but I might take the opportunity to thank my co-chairs, to thank the presenters for a lovely afternoon of clinical cases that were challenging. And we'll finish for the day, a wonderful day 2, and we look forward to an even better day 3. Thank you.
I have 2 announcements, by the way. Just 2 quick announcements. So 7 p.m. sharp in the lobby. So all the buses will be waiting there. So please don't be come late. So the second one, please, all of you, we need to take a 1 and photo for everybody. So we will have 2 lines, one here, the other one and down there. So please, we can do it quickly.